ASRB net plant pathology 2024. So this is last minute preparation, like those who don't have a time to prepare from the beginning or from the day one. So this video will be helpful for the people who are preparing in the last minutes or just uh, 1 week away to national eligibility tests, or 1 week away to various other kinds of preparation exams. So this video will be definitely helpful, but we cannot assure you 100% that the same kind of questions we may expect in the exams as well. But this is just like a recap, like what are all the different kinds of contents we may expect from various kinds of sections. uh, this is helpful for not only to have an idea from where we can expect the questions, but also it is helpful to guide your juniors or maybe other kinds of people who are all expecting help from you guys. Okay, so this is the last minute preparation video. I'm Manoj Shankar Reddy, PhD plant pathology. Let's start with origin of plant pathological words. There are a couple of pathology words will be there where we may also expect like the terminology is actually derived from what is the meaning of the terminology. Sometimes we may also expect such kind of questions. Let's start with phytopathology. Phytopathology, also known as plant pathology, which is a Greek word. So whatever that mostly ends with logy, mycology, pathology, like whatever it ends with logy, mostly they are Greek words only, derived from Greek only. So phytopathology or plant pathology generally deals with the study of plant diseases and their management aspects as well, right? So fungi, which is a Latin word, bacteria, virus. Fungi, bacteria, virus, these three are actually a Latin words. Fungi means a mushroom, bacteria means a staff cane or little stick, virus means slimy liquid or poison or venom or infectious matter. Such kind of meanings will come. But lichen is actually derived from Greek word which means to lick, the meaning. And fungicide is also a Latin word. There are four Latin words we have we just saw now: fungi, bacteria, virus and fungicide. These all are Latin words. Okay, and tungro, it is a little bit special, like rice tungro virus, where we observe rice tungro, right? So rice tungro is caused by rice tungro virus, where other two viruses are actually associated with rice tungro bacilliform virus, rice tungro spiricalform viruses, right? So generally this tungro is a mean actually originated from a Philippines word which means a stunted growth, or we can call it a degenerative growth. Since it is a Philippines word, the meaning of tungro in Philippines is in Philippines language is a degenerative growth or stunted growth. Quarantine is actually a Italian word which is means 40 days period. Anabiroga is actually a Kannada word which means a plant disease caused by mushroom. Tristeza, which is a Portuguese and a Spanish word which means sadness. Generally, this tristeza, tristeza virus we might have studied in disease of citrus, right? So this tristeza means sadness, which is actually derived from Portuguese or Spanish word. Kresek is an Indonesian word which means crackle. Kadang means again it's a Philippines word. This tungro and kadang both are Philippines word. So this kadang means dying. So okay, so generally kavaka is a Sanskrit word which which generally means fungi. In a couple of years before, it's a very long back, one of our professor used to tell there is a journal called kavaka. The kavaka means fungi. They used to publish articles, various kinds of articles related to fungi. Now I don't know whether this kavaka still exists or not, but generally kavaka is a Sanskrit word which means fungi. So from this origin of pathological words or origin of terminologies, we may expect like fungi is derived from which language, or tungro is derived from which language. What is the meaning of tungro, or what is the meaning of tristeza? Kadang means. So such kind of questions we may expect. The next one is the books. This is a very important section when it comes to books, because every year there is we may expect one or two books from in all the exams, not only net exam but also you can see other entrance exams as well. We definitely expect one or two books. So here we have just listed out some couple of important books and their the scientist who wrote the book, or the publisher, I mean the person who wrote the book, and along with the year of publication as well. So let's start with the Gaspard Bachin. He wrote a book called Phoenix Theatri Botanici, so which was published in the year 1623. And the next one is Michelli, P.A. Michelli. He wrote a book called Nova Plantarum Genera, and generally this Michelli is considered as a father of mycology, founder and father of mycology. But modern mycology generally it goes to Anton de Barry, but generally Michelli is considered as a founder of mycology, and also we can consider as a father of mycology, only mycology, okay, not plant pathology. So he's the first person who told that disease is caused by spores. So spores are the main reasons for causing the diseases, and he wrote a book called Nova Plantarum Genera. The book is very very important, which was published in the year 1729. The next one is the Linnaeus, Carolus van Linnaeus. So he wrote a book, not one book, he wrote two books, Species Plantarum, and uh, one more book he wrote regarding this taxonomy and all those things. Species Plantarum is one among the very important book, which was published in the year 1753. Species Plantarum and one more book I couldn't able to recall. The next one is C.H. Persoon. He wrote a book called Synopsis Methodica Fungorum. He published in the year 1801. And next one is E.M. Fries. E.M. Fries, he wrote a book called Systema Mycologicum, which was published in the year 1821. So this C.H. Persoon and E.M. Fries are both are the students of Carolus van Linnaeus. Actually, these two people are actually guided by this Carolus van Linnaeus. Later, they become the greatest contributors of mycology as well. This E.M. Fries is generally considered as uh, is generally considered as I forgot something reconstruct. No, not reconstructor of mycology. Reconstruct Tulsen brothers. Linnaeus of mycology. Yes, I think so. He is considered as a Linnaeus of mycology. E.M. Fries. His book Systema Mycologicum, and J.G. Kuhn. This is a very important. And J.G. Kuhn is the first person. He wrote a book that is completely devoted to plant pathology. Then you can ask me, what about the book that was published previous? Of course, they mentioned a little bit about plant pathology, but those books are not completely meant for plant pathology. But the book which was written by J.G. Kuhn, this book, that is Diseases of Cultivated Crops, Their Causes and Their Control, which was published in the 1858, is the first book that is completely devoted to plant pathology. So this book is a very important book. Like in exams we may expect, like, what is the first book that is completely devoted to plant pathology? Diseases of Cultivated Crops and Their Causes and Their Control. Sometimes we may expect years also, like 1858, such kind of things also. Okay, then Charles Darwin, he wrote a book called Origin of Species, 1859. Anton de Barry, we know very well Anton de Barry, father of plant pathology, father of experimental plant pathology. So many terms introduced, so many greatest scientists and who are actually contributed, those all are students of Anton de Barry. That's why we call Anton de Barry as the father of plant pathology. And recently I have published a good a newsletter on Anton de Barry, how actually Anton de Barry started his legacy and where it actually leads, and what he have done exactly contributed to plant pathology, why he's called as father of plant pathology, and all those things I have published in a newsletter. So interested candidates, just go and take a look into. Anton de Barry wrote a book called Comparative Morphology and Biology of the Fungi, Mycetiosa and Bacteria. Actually, the book was published in German version in 1866, but it is translated to English in 1887 by Garson and Beflor. But the book was originally published in German language in 1866. After almost 21 years, it is translated to English. The book is Comparative Morphology and Biology of the Fungi, Mycetiosa and Bacteria. And Tulsen brothers, they wrote a book called Selecta Fungorum Carpologica. They are considered as the reconstructors of mycology. Actually, everyone contributed something, right? But they are the one who put all the stuff into an order, like which one comes first and what is the next thing, and also the whatever the kinds of the diagrams or the life cycle stuff that we are studying right now, everything was given by Tulsen brothers only. That's why we named them as, not we, that's why they called as reconstructors of mycology. And they published a book called Selecta Fungorum Carpologica, and one more book also they wrote, Fungi Hypogaea, I think 1854 probably, but this book is very famous, Selecta Fungorum Carpologica, which was published in between 61 to 65, probably 4 to 5 volumes included in that, I think so. Secado wrote a book called Silloge Fungorum. This is also a very famous book, almost comprises of 25 to 26 volumes, which was published in between 82 to 31. And E.J. Butler, we know very well E.J. Butler is considered as father of plant pathology and mycology when it comes to India. So he wrote a book called Fungi and Diseases in Plants. Okay, and another book was there, Fungi and Plant Disease, that was written by B.B. Munkur. But Fungi and Diseases in Plants was written by E.J. Butler, which was published in the year 1918. And M.J. Thrumlachar, he wrote a book called Antibiotics and Plant Disease Control. This is one of the very famous book, and he is considered as the father of Indian antibiotics. And probably the book was published in 1960s. I'm not sure in detail about. Please let me know if you guys know. And B.B. Munkur, he wrote a book called Fungi and Plant Disease. He's the person who actually evoked this IPS, Indian Pathological Society, along with the chairmanship of SR Bose and some other greatest contributors in India. So he wrote a book called Fungi and Plant Disease, which was published in 1948. Years also very important. If you just take a look into the previous year question papers, years also repeatedly asked. So we cannot say where exactly the question will come from, so that it's better to go through each and every single aspect of plant pathology, right? So YL, Nina and PN Thapilial, Fungicides and Plant Disease Control, which was published in first version published in 1971, and recently fourth edition are running at, I think so. Paul Neergard is considered as the father of seed pathology, and he wrote a book called Seed Pathology also and he published it in the year 1977. The book is Seed Pathology. He is also the father of seed pathology. And Baker and Cook, they wrote a book called Biological Control of Plant Pathogens, which was published in the year 1980. So these are all just a couple of books. Other books also there. If you just take a look into the videos that we have published so far, around 40 plus videos we have published. If you take a look into, we have given the various kinds of books and their contributions, all those things we have given in detail about. We may expect so since why we are placed here this slide, just like we may expect from book names and so such kind of questions also we may expect. That's why we placed this slide. Coming to cell wall components of different plant pathogenic organisms, like what is their cell wall composition. So if you just take a look into the previous question papers, like fungal cell wall is made up of, or Womycota cell wall is made up of, like just giving the organism, following organism, their cell wall is composed of, such kind of questions we might have gone through. So let's see the organisms are different kinds of taxonomic groups and their cell wall composition. Generally, fungal cell wall is made up of chitin plus glucan, whereas a Zygomycota group of cell wall is composed of chitosan chitin or chitin chitosan. Womycota cell wall is made up of cellulose plus hydroxyproline, whereas Hypochytridiomycetes cell wall is made up of cellulose plus chitin. Yeast cell wall mannin beta glucan. Algae glucan plus cellulose. Trichomycetes polygalactosamine galactron. Bacterial cell wall is made up of peptidoglycan, are also known as mucopeptides. Virus generally contains no cell wall. Instead of cell wall, it contains a protein coat, generally act as a protecting structure. So the most commonly asked question is Womycota cell wall, Womycetes group of cell wall is made up of cellulose and a little amount of hydroxyproline also. In the options, sometimes they may mention cellulose, or sometimes they may not mention hydroxyproline also. If you see in 2021 exam, they have mentioned like Womycota cell wall is made up of chitosan, chitin, mannin and hydroxyproline. So since hydroxyproline is also one of the major component of Womycota cell wall, so the option only hydroxyproline was related to this Womycota. So the answer is hydroxyproline. So such kind of questions we may expect, right? So generally cell wall composition, these are all the important cell wall composition we just need to take a look into. And storage organ of plant pathogens, like however we have a humans, we have adipose tissue for storing the extra food material or food content during unfavorable time, or during a period or drought period where for their survival. The pathogens also have their food storage products, or the energy is stored in the form of other complex forms. So generally fungi stores the food in the form of glycogen and oil and lipids also. And Womycota generally food storage is a mycolaminar, whereas algae is a laminary. Yeast stores in the form of in volutin, whereas bacteria polybeta hydroxybutyrate or beta hydroxybutyrate or beta hydroxybutyrate. Such kind of questions we expect. If you see this bacterial cell wall is bacterial food storage organ or bacterial food storage product is polybeta hydroxybutyrate. So this is the important question in the if you see the previous year question papers in 2019 and 21, this question was asked. So generally bacterial food storage organs are bacteria store the food in the form of polybeta hydroxybutyrate or storage organ or storage products. Such kind of questions we may expect. And the next one is the anabiroga. Anabiroga means plant diseases are caused by mushrooms. There are a couple of terminologies: anabiroga, coliroga, polyroga, aracinaroga, chandiroga. What are all these terminologies? What are all the meaning of these terminologies? We'll see now. Anabiroga means plant diseases are caused by mushrooms. Coliroga means Mahali disease of areca nut, whereas polyroga means infection of pseudostem. For example, in banana bract mosaic virus, we can see the infection of pseudostem. So that pseudostem infection. So if the infection especially affects to this pseudostem region, that is called polyroga, especially we can see banana bract mosaic virus. Example, aracinaroga or chandiroga means if the disease is caused by the ganoderma, especially in areca nut we can see, for example, yellow leaf disease of areca nut which is caused by phytoplasma. So if it is caused by ganoderma, maybe sometimes in combination with other diseases as well. Generally if it is caused by ganoderma, it is called chandiroga or aracinaroga. This ganoderma disease especially we can observe in areca nut and Karnataka, where this disease was very prevalent. Later that is also identified in Kerala and as well as Tamil Nadu as well, right? So fruit rot of areca nut generally commonly called as coliroga in Kannada or Mahali disease in Malayalam. I repeat, fruit rot of areca nut commonly called as coliroga. Coliroga is a Kannada word, whereas we can also call it as Mahali disease of coconut. Mahali means it is a Malayalam word, whereas coliroga means it is a Kannada word. And I have mentioned a couple of first reports, like what is the first organism reported in fungi, who reported it, and what is the organism, what is the causal organism, scientific name we can call it, right? So bacteria, virus, and all those things. We have mentioned a couple of organisms and the scientist who first discovered it, what is the organism first reported by, what is the pathogen, and what is the first organism actually reported in India belonging to that particular organism, and the scientist involved. All those things we have given here. And the first one is the fungi. So before also, before Anton de Berry also, there are lots of fungi were but Anton de Berry were the first person who reported that plant diseases are caused by fungi, especially that much devastating form, especially lead blight of potato. So Anton de Berry was the first person who reported this fungi lead blight of potato, which is caused by Phytophthora infestans. In India, we couldn't able to find an exact proof. If you guys know, please let us know. And the second one is a bacteria. T.J. Burrill was the first person who identified the first plant bacterial disease, that is a fire blight of apple and pear, which is caused by Irvinia amylovora. But T.J. Burrill named this fire blight of apple is a Micrococcus amylovorus, later name changed into Irvinia amylovora. In India, bangle blight of potato, which is caused by Ralstonia solanacearum, is the first plant bacterial disease that was reported by E.J. Butler. When it comes to virus, M.J. Bijanic was the first person who reported a virus that can cause plant disease in tobacco mosaic disease, which is caused by tobacco mosaic virus. When it comes to India, the first reported virus in India is a sugarcane mosaic virus that was reported by J.F. Daster, probably 1913, I think so. And the next one is viroid. Diner and Raymer were the first person who reported this viroid in 1971. And the first reported viroid is a potato spindle tuber viroid. And in India, the first reported viroid is a tomato bunchet up viroid. And the report who reported this was still unknown. Please let us know if you guys know. Coming to virucide randless, valvet tobacco mottle virus, phytoplasma. The first phytoplasmal disease was reported by Doe et al. In 1967. Reported the disease is mulberry dwarf. It is known to be transmitted by leaf hopper Hishimonas Pycetis. Okay, when it comes to India, sandal spike is known to be the first phytoplasmal disease that was reported by Verma et al. In 1969. When it comes to spiroplasma, Davis et al. 1972, they reported a disease called constant. It is known to be the first pyroplasmal disease, and it is known to be transmitted by an insect vector called leafhopper Dulbellus maedis. If you see this phytoplasmal and spiroplasmal disease, majority of the phytoplasmal and spiroplasmal diseases are transmitted by an insect vector called leafhoppers. So when it comes to prions discovered by Stanley Prusner. Kuru disease in humans, scrappy disease in sheep were known to be the first reported prion disease. So for the discovery of a new causal agents, Stanley Prusner received a Nobel Prize in probably 1977, I think so. And the satellite virus that was reported by Casanis 1962, tobacco necrosis satellite virus is known to be the first satellite virus, and it is also known to be the smallest plant virus, which is around 70 nanometer. Mycoviruses was first reported by Hollings. Lala France disease of mushroom, that is reported in Agaricus bisporus, also known as X disease or water stripe disease, is known to be the first mycoviral disease, which means mycoviruses, virus that infect fungi, that is known to be the mycoviruses. Cyanophages, which was first reported by Safferman and Morris. LPP, Lingba, Plectonemanda, some things will be there, right? So the first name of the fungi as given as like LPP, like that. So cyanophages, that was given by Safferman and Morris. And bacteriophages was first reported by Twart and Herrelli. They reported T4 bacteriophage or T2 bacteriophage known to be, they worked on, and they identified bacteriophage individually, where Twart discovered in 1915, Herrelli discovered in 1917 individually. So they both got credit for this discovery of bacteriophage. Phage means eater. Bacteriophage means a bacterial eater. A virus that infects bacteria is known as bacteriophage. So Twart and Herrelli was the first person who reported this bacteriophages. Herrelli is known to be of course, they both discovered this bacteriophage, but Herrelli was the first person who named this as a bacteriophage. But Twart discovered it, but Twart didn't give any name. So they have identified a specialized organism or a new kind of organism with same kind of characters, whatever they have discovered. But Herrelli took one further step and named the organism as bacteriophages. So these are all the various kinds of first reports on organisms and who discovered it, and what are the first organism related to the discovery, and all those things we have mentioned here. We may expect, if we go through this table, it will be very easy to understand the overall concept rather than compiling in various other way. Just studying this table is very useful, I think so. And chemicals used in surface sterilizing agent. Here there are we do use various kinds of chemical for sterilization, and we have mentioned couple of surface sterilizing agents and their concentration and what exactly it is used for. And ethanol generally the concentration is available to 70 to 95%. Of course 99% is also available. So we use it for various kinds of purposes like disinfection of instruments and bench tops and all those things. When it comes to spirit, same ethanol only around 90 to 99% is available. Disinfecting and sterilizing the instruments and glasswares. So mercury chloride, it is a very important. This mercury chloride and sodium hypochlorite both are surface disinfectants only. But when it comes to mercury chloride, it is recommended at 0.1% concentration, whereas sodium hypochlorite is recommended at 1% concentration. In exams also they will ask what is the concentration of sodium hypochlorite, 1% or 0.1. If it is a mercury chloride is more toxic. That's why we are recommending at low concentration, 0.1. Sodium hypochlorite we can recommend up to 1% concentration. Both are used for surface disinfection. And I believe this mercury chloride was banned, and still it is available outside, but originally it is banned. In all the laboratories we are using sodium hypochlorite only at the rate of 1% concentration. 1 ml in 100 ml of water, you can make it 1% of sodium hypochlorite concentration. I repeat, 1 ml in 100 ml of water, it is equal to 1% concentration of sodium hypochlorite. If you want 5%, you can add 5 ml of sodium hypochlorite in 100 ml of water. That gives you 5% concentration. And propylene oxide also recommended 0.1% concentration, used for filter paper sterilizing. And formaldehyde we can recommend at 1:10%. Formaldehyde and water combination, generally used for laboratory, and if we are getting lots of contamination, we used this formaldehyde and water concentration to disinfect, especially laminar airflow chamber and the laboratory we used to disinfect by using this formaldehyde and water combination. And ultraviolet is generally in laminar airflow chamber, we have a specialized light called UV light, right? It just kills some kind of fungi to avoid contamination. Generally before working, if you are planning to work in laminar airflow chamber, you just switch on the UV light for at least 15 to 30 minutes before you're working, so that it can sterilize the area that you are planning to work. And we have given some couple of suitable media. Of course there are plenty of suitable media. If it is a fungi, generally oatmeal agar media and PDA is most recommended, but PD and oatmeal more or less similar, like they are the most recommended medium, but oatmeal medium preparation is a little bit difficult, so that every laboratory is using PDA medium, potato dextrose agar medium. For NA medium is bacteria, Kingsby pseudomonas fluorescens. Those are all common, and we have given a couple of other here. And azide rose Bengal, which is especially recommended Fusarium axisporum pharma species PC. Care medium Fusarium solani, all the Fusarium species we can recommend this care medium, the most suitable medium. Rapeseed agar medium and V8 juice agar medium, that is recommended for Phytophthora species. Apple leaf decoction medium Venturi anaequalis. Bean juice agar medium Coltotrichum lindimuthianum. CMRA medium Macrophomina fasciolina. These are all a couple of suitable medium that is especially recommended for the fungi given here. Apart from this, you can use PDA medium for almost all fungi, but these are all the specific medium, so where the fungi go very well at, so that we can see morphological characters or the spore production can happen well at this. But you can also go with the PDA medium as well, but in exams we can't say I will go with the PDA medium and all those things, right? So that's why, so these are all the specific medium for the fungi. And susceptible stages of for some important diseases, for at exact what stage there is a chance of infection of disease, that we will see. For rice sheath blight, the most susceptible stage is active tailoring stage, whereas brown leaf spot of rot vegetative growth, late blight of potato early adult stages, bean anthracnose pod formation stages. Fusarium wilt affects at all stages irrespective of tailoring vegetative, all the stages Fusarium wilt infects. Root rot generally at the first 45 days, exactly after 45 days of transplanting, initially where the root is developing, right at the stages, this root rot is highly dangerous. And cassava mosaic virus early stages of crop, pea root rot pod formation stage, and brown leaf spot of maize, it is highly destructive at the vegetative stage of the crops. These are all some susceptible stages of the crops or diseases. We have mentioned a couple of staining agents, how you stain, like in exams also endospore staining agent or plasmid staining agent or bacterial food storage organ staining agent. So bacterial food storage organ, poly beta hydroxybutyrate, we might have studied in the previous slide, right? So here I have given a couple of like what can I say, staining agents, along with what the staining agent is actually recommended for. So here endospore staining agent is malachite green. So malachite green is an agent which is recommended for the staining of this endospore. Plasmid staining agent acridine, capsule staining agent Indian ink, flagella staining agent carbol fission, lichens staining agent archin, inclusion body staining agent azure, cytoplasm and nucleus staining agent acetic dye, and bacterial food storage organ staining agent Sudan black bee. So these are all the various staining agents and what we are recommending for on the given here. So most commonly, if you see the previous exams, I personally see this endospore staining agent, bacterial food storage organ staining agent. These are all very important, and these questions were repeatedly asked in exam, but we can expect the same kind of questions will come in all the time. So that I have just pointed out a couple of agents or staining agents, that is what I actually recommended for. Please go through, definitely we may expect one or two kinds of questions from this. Here I have mentioned a couple of detection tests for plant pathogens, and what is the detection test, and the detection test is actually recommended for what pathogen I have given here. So sodium hydroxide NaOH, that is recommended for kernel bunt of wheat and ergot. For two diseases we can recommend NaOH test, and brain flotation technique is recommended for wheat tundu diseases. For bacterial leaf blight, not only bacterial leaf blight, for all the bacterial diseases, all the plant bacterial disease, Ouzo test is recommended. ELISA is for almost all plant viruses. Tetrazolium chloride test is see, this ELISA is not only meant for plant viruses but also known to be recommended human viruses as well. So this tetrazolium chloride test is recommended for banana bunchita virus. This was the repeatedly asked question, like banana bunchita virus, what is the detection techniques or identification technique used for the pathogen banana bunchita virus, tetrazolium chloride, right? So such kind of questions we may expect. So iodine test is recommended for rice tumor virus. Page or SDS phase or fluoroglucinol test is especially recommended for virids, because by using ELISA we cannot able to detect virids, so that page or SDS phase or fluoroglucinol test is recommended for identification of virids. DAPI technique, DAPI we can also call as a DAPI technique, especially recommended for Pytoplasma, whereas a Paraquat technique is recommended for post-harvest disease. So it is this slide is very important. Not only this slide, whatever the slides we have prepared, almost 225 slides every single slide is a very important. All the knowledge and experience I put together and prepared this fine preparation, and we are expecting a couple of questions from this in the last minute preparation as well. And so these are all the different identification tests. Along with that, and we have also given a couple of baiting techniques. Generally, carrot disc is used for the baiting of our agrobacterium tumefaciens. Cellophane is Rhizoctonia solani. Brassica seedlings is Pythium. Apple fruit is for Phytophthora infestans. So these are all the different kinds of material used for the baiting techniques of the pathogens. So generally, we mostly studied this Pythium and Phytophthora, right? Pythium generally Brassica seeds are used. Apple seeds are recommended for Phytophthora infestans. Just very simple. If you just want to know whether the soil or whether the particular field is infected with Pythium or maybe Phytophthora, just have the apple seed and put it there. If you observe this Phytophthora, that it means there is a inoculum, there is a load, inoculum load, or there will be inoculum present in such easy identification. So for easy identification, we call it as a bait, right? So bait is simply like to know the presence or to know whether it is there or not. So such kind of things we do recommend this kind of materials. Next one is the genomes of different plant pathogenic organisms. Here we have a couple of pathogens along with their genomes are very important. This question is also, as I told you, every single slide is very important. So mycoviruses generally contains a genome, double stranded RNA, hypovirulence. Just like reducing the virulence or capability of infecting nature by having this double-stranded RNA is known as hypovirulence. Generally, we can observe this phenomenon in Dutch-Elm disease, right? So hypovirulence is generally related to double-stranded RNA genome. Cyanophages contains double-stranded DNA. Bacteriophages contains double-stranded DNA. Gemini viruses contain single-stranded DNA. Lambda X, Pi X 174 contains single-stranded DNA. These are all some kind of genomes. So in the previous questions, Gemini viruses genome, or sometimes they may include a specific like cauliflower mosaic virus genomes. That kind of things also we may expect. But generally, these are all some kind of pathogenic groups and their genomes. And along with the model organism for genetic studies like fungi, what is the fungal organism that is used for genetic studies? There are fungal organisms like Neurospora crassa and Aspergillus species are generally recommended for the model organism. For example, when it comes to bacteria, Agrobacterium tumefaciens. For when it comes to virus, tobacco mosaic virus and cauliflower mosaic virus are the model organism. But when it comes to nematode, Chinorhabdis elegans is the model organism for nematodes. These are all couple of genomes and their model organisms. International Congress on Plant Pathology, generally called as ICPP. It is generally a non-profitable organization. Just generally they used to work on the recent problems and all those things. Every 5 years, all the big scientists all combined or joined together in a particular place, and they will discuss with all the problems and what are the future plans and all those things. So every year a particular venue will be there. It happens once in every 5 years, and the presidents and the tenure and all those things that are given here. The first International Congress on Plant Pathology happens in London, UK in 1968, and Ronald Calsey Starwood at the time he is the president. He continued his tenure between 1968 to 73. So tenure also 5 years for every president. I'm not telling the presidents and all those things. I'm just telling the venue and the year. So the second International Congress on Plant Pathology happens in 1973, exactly 5 years after the first conference. Minneapolis, USA. Third one is Munich, Germany, 78. Melbourne, Australia, 83. Fourth one, Kyoto, Japan, 88. Montreal, Canada, 93. Edinburgh, 98. Christian Church, New Zealand, 2003. Toronto, Italy, 2008. Beijing, China, 2020. This question was asked in 21, I think so. And Boston, USA, 2018. Lyon, France, in the previous year, 2023. The International Congress on Plant Pathology happened in Lyon, France, probably in August. It happens, and the next conference is going to happen in Australia, Queensland, probably in 2028. So far 12 international congresses on plant pathology happen, and the 13th International Congress on Plant Pathology is going to happen in Queensland, Australia in 2028. If you want, you can also book the tickets now itself by visiting the website of International Congress on Plant Pathology. And these are all the types of dispersal. I think you guys might have known this very well. Any agent that is dispersing through wind is known as anemochory. water hydrochory, animal means zoochory, insect means entomochory, birds means ornithochory, human means anthropochory. These are all the technical terms used for the dispersal. And some kind of blotting techniques, it is supposed to be discussed in uh, physiological and molecular plant pathology, since the molecular plant pathology, physiological molecular plant pathology is a very depthy subject. We can't able to discuss line by line. If you want to discuss a line by line, again it will take another very big videos, right? And we are also planning to publish this physiological and molecular plant pathology videos one by one. So just a couple of important things we have mentioned here. Northern blotting, that is detected for the identification of RNA. Southern that was first developed or discovered by Alvin 1971. Southern blotting is used for the identification of DNA. EM Southern, year 1973. Western blotting is recommended for protein, that was discovered by Bernetti and year of 1971. If you see news, N, north, south, east, west. So if you see, southern blotting is employed for DNA. Southern people are look more dark just because of having more DNA. You can remember in that way. That doesn't mean South Indian people are having more DNA. It is believed that more capable people are more strong. I can't say strong people, like more DNA, like the original kind of people having dark. They having more DNA or strong DNA content. Such kind of things will be there. I don't know whether it's true or not. That is secondary. So you can remember in that way. North Indian people are white, which contains RNA. And that doesn't mean they don't have DNA. Generally, I'm telling the way how we can remember. So Western blotting automatically, if you see West, Mumbai, so that is a hub for marketing. You can see all kinds of protein supplements or production, all kinds of marketing in that way, where they are producing from Mumbai. So you can remember in that way. Okay, that doesn't mean other people are not producing. Just for your remembrance, I am telling. Okay, northern blotting RNA, southern blotting DNA, automatically western blotting is a protein. So that their discoveries and all the years they have given here, that is also very important. Here some couple of culture collection centers were given here along with their full forms and located at ATCC, American Type Culture Collection, located at Maryland, USA. ITCC, Indian Type Culture Collection, very important, located at IRI, New Delhi. And NCCB, Netherlands Culture Collection of Bacteria, located in Netherlands. And CFBP, Collection Francis D. Bacteria Pathogens, collection of plant pathogenic bacteria, very simple, Angers, France. ICMP, International Collection of Microorganisms from Plants, Auckland, New Zealand. And IMTech or MTCC, it is a very important from Chandigarh, India, Indian Institute of Microbial Technology and Microbial Type Collection, located at Chandigarh, India. And BCCM LMG, located at Belgium Coordinated Collection of Microorganisms, located at Gent, Belgium. NCPP, National Collection of Plant Pathogenic Bacteria, United Kingdom. NRRL, Agriculture Research Service Culture, probably somewhere in India. So these are all a couple of culture collection centers and their locations. So generally the questions may fall from like expand ITCC, expand ATCC, or like IMTech is located at. Such kind of questions we may expect in exam. So these are all a couple of culture collection centers. First in genome sequencing of plant pathogens, so very important in the last couple of years. We may expect at least one. What is the first plant pathogenic fungi to be completely genome sequenced? Who sequenced it? And all those things. To have a good idea, we have mentioned the first in the sequencing. First fungi to be completely genome sequenced is Magnoporthe grigia, that is causes rice blast, sequenced by D. Natal in the year 2005. So this genome size is not that much important, but for our reference and further adding information, we just gave a genome size as well. But I would say you just remember the first four columns. The agent fungi, plant pathogen, sequenced by, and the year. So fungi was first sequenced is Magnoporthe grigia, D. Natal 2005. First plant pathogenic bacteria to be completely genome sequenced is Xylella fastidiosa, which causes Pierce disease of grapevine, citrus variegated chlorosis, and various other diseases are right. This Pierce disease of grapevine is caused by Xylella fastidiosa, is known to be the first plant pathogenic bacterium to be completely genome sequenced. That was sequenced by A.J.G. Simpson in the year of 2000. So there is a reason. Actually this Pierce disease become very especially in citrus and grapevine, this become very problematic in Brazil. So at the time A.J.G. Simpson was a person who are working in the Brazil with the help of Brazilian consortium, various kinds of other contributors. He just started sequencing this specific organism, that is Xylella fastidiosa, and now we know to be that this is the first plant pathogenic organism or bacteria to be completely genome sequenced. In 2021 they have asked a tricky question. What is the bacteria that is related to genome sequencing in the particular fields. For example, when it comes to plants, it's ZLF fastidios. What about the first plant pathogenic, human pathogenic bacteria, Haemophilus influenza? They have mentioned the two, what can I say, two options in a same question, and they just formed the question. So for further reference, I suggest you to go 2021 net question paper. We have published the previous 2021 net question paper, the model question paper. The same questions only they are asked, the memory based questions, whatever we remember, the same question papers we have mentioned there. You please go through and take a look into. You might have understand how they have asked. So when it comes to DNA virus, first DNA virus completely genome sequenced is cauliflower mosaic virus. It was sequenced by Frank et al. In the year of 1980. First RNA virus to be genome sequenced, tobacco mosaic virus, sequenced by Golet et al. In 1982. Here, if they are not mentioning DNA virus or RNA virus, which one we need to go? We need to go with cauliflower mosaic virus, since if they are asking what is the first plant pathogenic virus to be completely genome sequenced, we must go through the years first of all. So DNA virus or RNA virus, if they are not mentioning, that's not a big deal. Simply if they have asked the question like what is the first plant pathogenic virus to be completely genome sequenced, we will go with 1980, because 1980 is the year where cauliflower mosaic virus sequenced. After 2 years only TMV is sequenced, right? So if they are not mentioning DNA, RNA, no need to worry about it. The first virus to be completely genome sequenced is cauliflower mosaic virus. RNA virus to be completely genome sequenced means tobacco mosaic virus. Next one is a viride. First viride to be completely genome sequenced is a potato spindle tuber viride that was sequenced by Gross et al. 78 and Dixon 79 in the year of 1978 and 79 respectively. And the first yeast to be completely genome sequenced is Saccharomyces cerevisiae. The genome project of yeast was actually started in 1989 and it is continued up to 1995 and 1996. Okay. This is the single-celled organism first to be completely genome sequenced. And when it comes to nematode is the Chinorhabdus elegans. It is also the model nematode, and it was sequenced by Andrew Fire and Craig Mello in 1998. And of course, these are all the two scientists who discovered this RNA interference technology in nematodes, right? RNA interference or post-transcriptional gene silencing technology. Okay, red bread mold first genome sequenced is Neurospora crassa, which was sequenced by Gallagher in 2003. This table is very important. We may expect at least 1 or 2 questions from this table. So rather than compiling the information, so if you study it in this way, it will be very easier to remember. So this is about the genome sequencing of plant pathogens. Plant pathological societies. There are a couple of plant pathological societies was established to promote the plant pathology, and we also expect questions from these societies as well. And in 2019 probably this question was asked, what is the oldest plant pathological society? That is the Netherlands Society of Plant Pathology, which was started in 1891, and we have given like the year of establishment of the society, and what is the journal that we will publish, what is the frequency of publication that we given here. And the oldest society and the first society of plant pathology is the Netherlands Society of Plant Pathology, which was established in the year 1891. And the next one is the British Mycological Society 1896, and American Phytopathological Society, one of the largest society, which was established in the year 1908. They are publishing the two journals, Phytopathological 1911 and Plant Disease 1916. They are monthly journals. And 1916 Pathological Society of Japan established. They published a journal called Japanese Journal of Pathology from 1934 onwards, quarterly journal. And the next one is the Canadian Pathological Society, which established in 29, and the Chinese Society of Plant Pathology is also established in 1929. And when it comes to India established IPS, Indian Pathological Society, that was established by under the chairmanship of B.B. Munkur in 1947, February 28, I think so. They are producing a journal called Indian Pathology from 1948 onwards, which is a quarterly journal. And in annual reviews 1963 and annual review of pathology. And International Society of Plant Pathologists, which was started in 1968, which mainly they produces a journal called Food Security. And Australian Plant Pathological Society is from 1969, which they are producing a journal called Australasian Journal of Plant Pathology, which is a bi-monthly journal. So these are all couple of like what is the oldest society or oldest association of plant pathology we can expect, like Netherlands Society of Plant Pathology, and this American Society, Society of Japan, Chinese Society of Plant Pathology, Indian Society of Plant Pathology. These are all a couple of important societies where we can get very good knowledge on plant pathology. And other couple of things also there here. 1970 Society of Mycology and Plant Pathology 1970, and British Society of Mycology and Plant Pathology in 1981. International Society of Molecular Plant Microbe Interactions are producing from 1982. Association of Plant Pathologists in India 84. Indian Virologist Society 1984. Indian Society of Plant Pathologists 1984. Society of Plant Production Sciences 1992. So these are all the various societies and the journals that they are producing. We may expect sometimes um, not only for net, even for if you go for interview, ARS interview also such kind of questions. A couple of our seniors and our teachers told that we may also expect such kind of questions or this kind of questions as well. And the laboratory equipments along with their discoveries, who discovered it, what is the laboratory equipment. The first one is starts with the compound microscope. So compound microscope was first discovered by Zacharias Janssen in 1595, used to magnify the objects. And the simple microscope was discovered by Antony van Leeuwenhoek. So first compound microscope was discovered by Zacharias Janssen, but simple microscope was discovered by Antony van Leeuwenhoek in 1676, used to magnify the objects. Autoclave generally was discovered by or developed by Charles Chamberlain. Developed by Charles Chamberlain in 1879, used for sterilization of glasswares. Hot air oven, which was developed by Louis Pasteur to sterilize the glasswares, and the continuation comes. And electron microscope, which was developed by Knoll and Ruska in 1931, generally used to detect very small organisms like plant viruses. And electrophoresis, the technique was developed by Arne Tiselius 1931. Spectrophotometer was developed by Arnold Werwel Beckmann 39. Phase contrast microscope, Zernike 53. Laminar airflow chamber was developed by Whitefield 1962. He is also considered as Mr. Clean, sorry. So ELISA, enzyme linked immunosorbent assay, that was developed by Adam and Clark, which was developed in the year 1977, identification of plant viruses based on antigen antibody reactions. Incubator generally used to incubating the plant pathogens. Scanning tunnel electron microscopy STM, that was developed by Binning and Heinrich 1981, generally to observe the atomic images. And PCR Cali Mullis 1983, generally used to amplify DNA. And hemocytometer, which was developed by Louis Charles Malasage. Generally this hemocytometer is used to count the number of cells or spores in a given sample. So these are all about a couple of instruments along with their discoveries related to. And coming to mycology, Aprosorium, generally what is Aprosorium? Attachment organ of fungi, or like attachment organ, like what can I say, the supporting organ of fungus, that kind of things will be there. Hastoria means a nutrient absorbing organ or energy absorbing organ of fungi is called as Hastoria. Generally pathogens produces different shapes of Hastoria. Each and every pathogen has their own shapes of Hastoria. And if you see knob-like Hastoria is produced by albugo. If you see albugo, in albugo also we can see some kind of knobs. So such kind of knobs will be observed in albugo. A finger-like Hastoria is generally produced by Phytophthora, whereas branched or peg-like Hastoria is produced by Sclerospora. Bulb-like Hastoria is produced by Cuscuta. Wedge-shaped Hastoria is produced by Archithobium. Finger-shaped Hastoria is produced by downy mildew pathogens, whereas globose Hastoria is produced by powdery mildew pathogens. Pyriform Hastoria is produced by Basidiopora. So these are all the couple of shapes of Hastoria, and other pathogens also there, where we have mentioned just a couple of examples. That's it. So if you see this mechanism where this is the spore is coming and landing on the surface, and it is germinating when it gets the signals from PAMP and CAMS, and cyclic pathways are there, right? So the germination, it is known as germination tube, and again it's producing another structure called Aprosorium. This Aprosorium is the attachment organ of fungi. From that Aprosorium, where some other kind of a penetration, a peg is penetrating inside, the peg is known as penetration peg or penetration hyphae. From penetration hyphae, some small bulb-like structure, some kind of bulge-like structure is producing. Of course, it depends. Shapes may differ, like as I told you, knob-like Hastoria or finger-like Hastoria. So shapes may be different, but Hastoria, it enters inside the cells and inside the epidermal cells and starts to absorb the nutrients. The structure which absorb the nutrients is known as Hastoриum, or energy absorbing organ or nutrient absorbing organ of fungi is known as Hastoриum. This is about Aprosorium and Hastoриum and different shapes of Hastoриum. Now we'll see the types of flagella which is produced by the fungi. So generally fungi can locomote. Not only for fungi, you can see bacteria also produces flagella. So now since we are discussing only flagella in fungi, so in flagella in fungi generally like hair, with hair, without hair, such kind of things will be there. Now I will tell you what are all the different kinds of things, and in exams also what kind of questions we may expect. So let's start with the first one. First of all, we will go through some kind of terminologies. If the flagella is in upward direction, it is called anterior. If the flagella is a downward direction, it is called posterior. You remember these things. If the flagella is upward direction, it is anterior. If downward direction, posterior. And the second thing, if the flagella is having hair, it is called tinsel. Flagella with hair means tinsel. If the flagella is having no hair, it is called whiplash. I repeat, if the flagella is in upward direction, it is called anterior. If the flagella is in downward direction, it is called posterior. If the flagella is having hair, it is called tinsel. If the flagella is having no hair, it is called a whiplash type of flagella. Now, by having these pictures, the flagella is a downward direction. It is called posterior. No hair, it is called whiplash. So now let's say all combinedly together. Posterior whiplash. Posterior means the flagella is a downward direction. Whiplash means there is no hair. Posterior whiplash type of flagella is produced by Chytridiomycetes group of fungi. What are all the fungi that comes under Chytridiomycetes? They produce this kind of flagella. Now the next one is the flagella is upward direction. Upward means anterior. It having hair. Hair means tinsel. Anterior tinsel type of flagellum, it is produced by Hypochytridiomycetes. All Hypochytridiomycetes group of fungi produce anterior tinsel type of flagellum. Third one will come to the flagella is upward direction, but here two flagella is there, which means biflagellate. So one is a short, one is a long. We can say in this way, anterior biflagellate starts with biflagellate. Biflagellate with anterior whiplash type. One is short and one is long. I repeat, biflagellate, two flagella are there. Anterior type, anterior, the two whiplash type. Anterior whiplash means no hair, right? So biflagellate anterior whiplash type, one is short and one is long. This kind of flagella is produced by the Plasmodioporomacetes group of fungi. For example, clubroot of cabbage, such kind of group of fungi, they will produce this kind of flagella. And the last one is upward direction, one is downward direction. So upward means anterior. We will say anterior tinsel. Tinsel means flagella over there. Anterior tinsel. Posterior, there is no hair. Without hair, what we can call, what we used to call whiplash, right? Anterior tinsel. And posterior whiplash type of flagella is produced by Oomycota group of fungi. Very clear? Chytridiomycota group of fungi, all the fungi that comes under Chytridiomycota group of fungi, they produce is posterior whiplash type. Posterior means downward, whiplash means no hair. Hypochytridiomycetes produces a flagella called anterior tinsel. Anterior means upward direction, tinsel means with the hair. Plasmodioporomacetes biflagellate with two anterior whiplash. One is short and one is long. Oomycetes biflagellate, anterior tinsel and posterior whiplash. These are all the different types of flagella that is produced by the fungi. So in exams, what kind of questions we can expect? They used to mention like, they won't mention the class of fungi. They used to mention Phytophthora produces what kind of flagella. First of all, we must know what are the different kinds of fungi that comes under this classes of fungi. Oomycota, there are three important classes of fungi, or genus I would say. Pythium, Phytophthora, all downy mildews fungi comes under this Oomycota. Majority of the downy mildew, true downy mildews, not false downy mildews, okay? True downy mildews that comes under this Oomycota. All the Oomycota fungi, what all comes under the Oomycota fungi, they produce same kind of flagella, that is anterior tinsel and posterior whiplash. Biflagellate with anterior tinsel and posterior whiplash, Oomycetes. And Plasmodioporomaicota, there is only one fungi, that is a clubroot of cabbage, which is caused by Plasmodioporobrassicae. They produces the same kind of flagella, that is a biflagellate anterior whiplash type. One is short and one is long. So by mentioning examples also, they may ask questions. So generally these are all the types of flagella which we can observe in fungi. And sexual and asexual spores produced by fungi. First of all, we must know What is sexual spore? What are the different kinds of sexual spores are there? So in fungi can reproduce by means of both sexual and asexual reproduction. In sexual reproduction, sexual spores are produced. In asexual reproduction, asexual spores are produced. Now we will see what are all the different kinds of sexual spores are there in fungi and asexual spores. From this also we may expect questions. That's why we are mentioning here. Okay. So when it comes to oospores, oospore, zygospore, ascospore, basidiospore, these are all the sexual spores in fungi. Then you can ask me, there is no other sexual spores? No, these four are the sexual spores: oospore, zygospore, ascospore and basidiospore. Oospore is the sexual spore which is produced by Oomycota group of fungi. Zygospore is the sexual spore produced by the Zygomycota group of fungi. Whereas ascospore, ascomycota, basidiospore, basidiomycota group of fungi, where they produce this kind of sexual spores. Then, which means, so then all the Oomycota group of fungi, they will produce only oospores as a sexual spore? Yes, of course. What are all the fungi that comes under Oomycota group of fungi? They will produce oospore as a sexual spore. Here couple of other things also there, where oospore and zygospores are diploid sexual resting spores. I repeat, oospore and zygospores are diploid, which means 2n in condition, diploid sexual resting spores. They are only resting spores. Okay. When it comes to ascospores and basidiospores, ascospores and basidiospores are haploid non-resting sexual spores. All are sexual spores only. Oospore and zygospores are sexual diploid sexual resting spores, whereas ascospores and basidiospores are haploid non-resting sexual spore. Generally, ascospore in ascomycota group of fungi, they are producing inside a sac-like structure called ascus. Each ascus contains 8 ascus, which means all ascomycota group of fungi produces a ascus where 8 number of ascospores are producing inside. Generally 4 to 8 it ranges from, but majority of the ascomycota group of fungi contains 8 number of ascospore in their ascus. When it comes to basidiomycota, majority of the basidiomycota produces 4 basidiospore above a structure called basidium. Here they produce inside a structure called ascus, where the basidiospores are produced outside a bulb-like structure or a club-like structure called basidium. And in exams we can produce this, see here, produces spores endogenously. Endo means inside. They are producing inside a sac-like structure is called endospore, or endogenously production of spores, right? Exogenous, exo means they are producing outside, the spores are producing outside, right? So it's called exospores. Based on the options also we may go through. So the overall, this oospore and zygospores are the diploid resting spores. Ascospores and basidiospores are the haploid non-resting spores. Oospore is produced by Oomycota group of fungi. All the fungi comes under Oomycota produces oospore as a sexual spore. Zygospore is Zygomycota, ascospore, ascomycota, basidiospore, basidiomycota, and the diagrams how the spores look like I have mentioned on the right side corner. Asexual spores. So now we'll see asexual spores. Zoospores. Zoospores are generally motile spores. So we have already discussed the various kinds of flagellation, how the spores will be look like, right? If the spore having flagella, it is called as zoospore. So motile spores with flagella is known as zoospores. Zoospores are generally produced from is a type of sporangiosspores produced from sporangium, and they are produced in Oomycota group, lower fungi. Pythium and Phytophthora generally produces oospore, this kind of oospore. Aplanospores. Aplano means non-motile spores. The only difference between zoospores and aplanospores are zoospores are motile spores, aplanospores are non-motile spores, just because of flagella. And aplanospores are produced by the groups of fungi like Haematococcus, Chlamydomonas, Wickeraria. I would say just Zygomycota group of fungi produces this aplanospores. And sporangiospores, again same Zygomycota group of fungi, Rhizopus and all those produces a structure called sporangium. So inside the sporangium only sporangiospores are produced. If it is releasing outside with the flagella, we can call it as zoospore. If there is no flagella, it is called as aplanospore. Very simple. They are also non-motile haploid spores. Okay, they are also producing from a sac-like structure called sporangia. So this is a sporangia. We can right side we can see, right? And other spores are like Conidia and Conidiospores are which is produced by Penicillium and Aspergillus. Pycnidiospores are Heteromycota group of fungi produces, and Chlamydospores and the Chlamydospores and Sclerotia are the resting spores that is produced by the pathogens. For example, see if you see Chlamydospores and Sclerotia both are hard compact resting spore that is produced by the plant pathogens during unfavorable time only. But chlamydospore is produced by the plant pathogens like saprolegni and fusarium, whereas sclerosium is produced by the plant pathogens like Sclerotium ralphsi, macrophomina, rhizoctonia, and other kinds of fungi, and claviceps, other kinds of fungi. Okay, blastospores, the spores which are produced from budding or budding spores are known as generally blastospores, generally produced by the Ascomycetes group of fungi, Basidiomycetes and Jagomycetes group of fungi can produce this blastospores. And resting spores, and please add that is chlamydospores also in this section. Chlamydospore is also a resting spore. Okay, Sclerotia, just now I told you it is a hard resting structure that is produced by the plant pathogen during unfavorable conditions. So Macrophomina, Fasciolina, Sclerotium ralphsi, they produce this hard sesame shaped resting structure. It is a very small resting structure that is called Sclerotium. When it comes to rhizomorph is like a root kind of aggregations or root like aggregation that is produced by a couple of pathogens like Armillaria, Amelia, Ganoderma. Especially in forest trees we can observe this rhizomorph, simply like aggregation of roots. However, if you just pull the a plant roots, however the roots look like the same way the rhizomorph look like. This is also resting structure. So these are all the different kinds of sexual and asexual spores. In exams the questions will come in any way. If you have the knowledge, it will be very easy to understand the concept so that we can answer the question in the way that they are asking. So that's why we are mentioned this different kinds of sexual asexual spores along with the resting spores. Since I have mentioned this, ascospores and basidiospores are the haploid non-resting sexual spores. I repeat, haploid non-resting sexual spores, whereas oospores and zygospores are diploid sexual resting spore. All are sexual spores only. Ascospores and basidiospores are non-resting, whereas oospore and zygospores are resting. During unfavorable time, what they do, what this ascospores and zygospores, they simply survive in the soil. That's it. Whereas ascospores and basidiospores are the infecting spores, non-resting spore. They cause the infection. Okay. And these are all the different kinds of fruiting bodies. Just now we discussed the kind of sexual spores and asexual spores. Where these sexual and asexual spores are producing from, right? So asexual spores are producing from asexual fruiting bodies. Sexual spores are producing from sexual fruiting bodies. So let's discuss what are the different kinds of asexual fruiting bodies are there. So pycnidium, sporadicium, acervula, cinema. These are all the different kinds of sexual fruiting bodies. Okay. So pycnidium is a hollow flask-shaped globose fruiting body. Sporadicium is a hemispherical barrel-shaped. Acervula is a saucer-shaped depressed sauroparenchymatous hyphae. Cinema is aggregation of branched and unbranched conidiophore. Very simple. If you see here, it is a hollow flask-shaped fruiting body. In the pictures, just take a look at it. It is look like a, what is the shape? Flask-shaped. In exams, they may also ask what is the shape of the fruiting body. Something like such kind of questions also you may expect. Okay, and generally, if it comes to sporadicium or sporadicium, it is actually cushion shaped. Okay, and acervula is actually saucer shaped or saucer shaped. And when it comes to cinema, it is generally aggregation of all branched unbranched conidiophores, all are put together. And the most important thing we need to consider is examples. Pycnidium type of asexual fruiting body is produced by the plant pathogens like Macrophomina fasciolina, Diplodia. So these are all the different kinds of pathogens which produces pycnidium. Sporadicium is produced by Fusarium, Tubercularia and Epicoccum. These are all the pathogens comes under sporadicium. And Coltrotrichum, very important. Give an example of fungi that can produce acervula as a asexual fruiting body, Coltrotrichum. And one more example is Pathology. And Cinema, there are the examples of cinema producing fungi, Sarcosistis and Graphium. So it's a very important where we do expect examples or maybe shapes or maybe other kinds of things. So the overall this is very important. Just go through along with the examples as well. And other names of the fungi. Just now we discussed different groups of fungi, Ascomycota, Basidiomycota, Jagomycota and all those things. So what are all the other names? Each and every fungi has other names also. In exams we may expect other names also, right? So Womyscota group of fungi or Womystis group of fungi is also known as egg fungi, also known as pseudo fungi, also known as zoosporic fungi. So there is a reason behind the naming. So Womystis is called, why it is called egg fungi? It is produced egg like oospore or egg kind of reproductive structure is produced. That's why it's called as egg fungi. Pseudo fungi, because the Womystis group of fungi is not a true fungi. That's why it's called as pseudo fungi. Zoosporic fungi, because they are producing a zoospore. That's why it's called as a zoosporic fungi. And you come to the Zygomystis group of fungi. They also called as a yolk fungi or zygote fungi, where the zygospore is producing from a zygote. That's why it's called a zygote fungi or yolk fungi. Okay. Ascomycota group of fungi is called a crown fungi, because it is one of the largest phylum of fungi. The majority of the fungi, around 80,000 plus species of fungi, comes under this Ascomycetes, include mushrooms and other species of fungi as well. And the sac fungi, because all the ascospores are produced, a sac-like structure is called this sac. That's why it's called ascus. The sac-like structure is called ascus. That's why it's called a sac fungi. When it comes to Basidiomycota, it is called a cup fungi, also called as club fungi, where the basidiospores are produced above a club. That's why it's called as a club fungi. And in Ascomycota group of fungi, each ascus contains 8 number of ascospore. In Basidiomycota group of fungi, each basidium produces 4 number of basidiospore. It is a very important. Deuteromycetes group of fungi. Before Kirketal classification, up to 90 Ainsworth classification, this Deuteromycota was separate division, separate phylum. Now it is classified under Ascomycota group of fungi. Okay, so majority of the Basidiomycota contains mushrooms, and a couple of mushrooms also comes under this Ascomycota as well. But Deuteromycota, all, or why it is called as Deuteromycota, dustbin fungi or fungi imperfecti, because all the fungi comes under this Deuteromycota, which lacks a sexual stage. That's why it's called as a dustbin fungi. And downy mildew fungi are generally called as hairy fungi. Powdery mildew fungi is generally called as sac fungi. Actinomycetes fungi is called as ray fungi. Pisidomycetes is called a cup fungi, because all Pisidomycetes produce a cup-like structure. It is called as cup fungi. Okay, so these are all the other names of fungi and different groups. And other terminology is also used. Asexual stages, it is not necessary to mention the same kind of terminology. Like asexual stages is also have other names. Asexual stage is also known as anamorphic stage, also known as a mitotic stage, because mitosis will happen. The producing spores also known as mitospores. Okay, also known as the imperfect fungi, also known as the residual fungi, also known as secondary fungi. These are all the various kinds of terminologies. In exams we may expect any kind of terminology. Sexual stages are also known as anamorphic stage, also known as mitotic stage, because mitosis will happen, also known as imperfect stage, because asexual, because sexual stage is lacking, also known as a residual fungi, also known as secondary fungi. When it comes to sexual stage, fungi is known as teleomorphic stage. In sexual life cycle, meiosis will happen. That's why it's called as meiotic stage. The production of spores we can also call as meiospores, because it is producing through the process of meiosis, also known as a perfect stage. And the combination of a teleomorph, which means sexual stage, and anamorph, asexual stage, combinedly called as holomorph. And the sexual spores are also known as meiospores, because the spores are producing through the process of sexual reproduction. That's why it's called meiospores. Examples are oospore, zygospore, ascospore, basidiospore. These are all sexual spores. I think we have already seen in the previous slides. All the spores which are producing through the sexual process, we can call also as a meiospores, because they are producing through the process of meiosis. Examples of these meiospores are sexual spores are oospore, zygospore, ascospore, and basidiospore. When it comes to asexual spores, also known as mitospore, because they are developing through the process of mitosis. Examples are zoospores, conidia, oidea, spermatia, microconidia, macroconidia. All are asexual spore. You can also mention as sclerotium, chlamydospore. All those things are, even though they are resting spores, they are vegetative or asexual resting spores, so that it comes under asexual. Okay. Regarding Anton de Barry, we need to discuss a lot about this man. He's a truly phenomenal man. We generally called as the father of plant pathology. And what we know is he discovered the life cycle of lead blighter potato, and we introduced many terms. So since it comes to terminologies, let's see what are the different kinds of terminologies that he have introduced. Hastoria, also known as sinkers. Teleospores, he called it as teletospores. Heteroecious rust and autoecious rust. Symbiosis and symbionts. Sex hormones in fungi. Obligate parasitism. endophytes and other terminologies, facultative parasite, facultative saprophyte, various kinds of terminologies and new things that was introduced or coined by Anton de Berry only. He is considered as father of plant pathology, is also known as father of experimental plant pathology, and he discovered the life cycle of lead beta potato, and is also the first man who initiated the work on this botrytis cinerea, first physiological work, and lead beta potato, that is a Phytophthora infestans, and he also gave the term, right, Phytophthora. Phyto means plant, Phthora means destroyer. Generally Phytophthora means plant destroyer, and he also developed around 220 plus students. Later they become the greatest contributors of plant pathology. So in order to you know, to respect him, or the kinds of contribution that he have done, we respected him, we honored him with the name of father of plant pathology. And there are other things also there to discuss, but anyway, we'll see one by one. So these are all the different kinds of terminologies that was given by Anton de Berry. And in nomenclature of fungi, if you see the nomenclature of fungi, fungi ends with mycetes, mycota, and all those things. Let's see what this nomenclature actually trying to tell. If any phylum is always ends with mycota. You can take zygomycota, ascomycota, omycota, basidiomycota, whatever it is. If it ends with the mycota, it is a phylum. Subphylum always ends with the mycotina. Class always ends with the mycetes. Order always ends with the ales. Family always ends with the ACA. By using the standard endings, we can remember, or we can identify whether it is a order, whether it is a class, whether it is a family, all those things. In exams they will ask like class ends with, in taxonomic classification or in taxonomic or nomenclature, class always ends with, or mycetes is actually the ending that is actually denoted or recommended to class. So such kind of things are directly indirectly they may ask, but we remember in this way, like phylum always ends with mycota, subphylum mycotina, class mycetes, order ales, family ACA. In this way we will remember this nomenclature of fungi and their suffix endings. And these are all the different kinds of downy mildews we can observe. Of course, various kinds of downy mildews are there, but these are all generally considered as true downy mildews. Uh, and we'll see the sporangiopore characters. Generally, this downy mildews are characterized based on the sporangiopore characters and the where how they are hanging, like the branches and all those things. So the first one is the sclerospora. How can we identify the sclerospora genera? Very simple: swollen, long, stout, upright branches. If you see this is the why it is called downy mildew. We can generally see on the lower surface, down surface or downy surface. That's what it's called as downy mildew, because on the lower surface more amount of moisture will be there due to more amount of stomatal closure and opening, where more amount of moisture will be maintained at the lower side. This downy mildew pathogen is favored by high moisture, like 90 to 95, around 95 to 100%. So lower surface more moisture will be there, so that this downy mildew pathogen is favored by lower surface. That is first thing. So here, based on the sporangiosperm branching, how they are branching, we can identify which pathogen it is. So sclerospora generally it is a swollen, upright branches will be there. There we can see here, this is a swollen. The spores are like swollen, small swollen, right? Like I would say bulged, right? So long stout with upright branches, generally we can say. Examples are like Cambo downy mildew, which is caused by Sclerospora graminicola. So Peronospora dichotomously branched at acute angle. I repeat, dichotomously branched at acute angle, which is caused by onion downy mildew, Peronospora destructor. This is Peronospora. And Plasmopora, right angle with blunt tips. We can see, and every right angle the tips are like blunt. So right angle with the blunt tips, that's the identification factor for Plasmopora. Grapes downy mildew, which is caused by Plasmopora viticola. So Basidiopora, generally club shaped with a swollen head. We can see here, this is a club shaped with a swollen head. So this is this this club shaped swollen head is generally known as sterigmata, around where the sporangium is produced above. So this kind of thing is called Basidiospora. China Astra downy mildew is caused by Basidiospora entospora. And last one is a Bremia. So for the identification of Bremia, it is more or less similar to Peronospora only. If you see this, more or less similar to Peronospora only, but where the tips of the branches, each tip or cup shape contains four sterigmata. Four sterigma it contains. For example, lettuce downy mildew, which is caused by Bremia lactose. So these are all different kinds of downy mildews that we can observe in Oomycota group of fungi. These are all very, very, very important. They simply tell you the kinds of branching. By using the kinds of branching, we must identify which genera it is. I repeat once again: Sclerospora, long stout upright branches; Peronospora, dichotomously branched at acute angle; Plasmopora, right angle with blunt tips; Basidiospora, club shaped with swollen head; Bremia, tips of the branches are cup shaped with the 4 sterigma are present. So these are all the different kind of downy mildews along with the examples. And we have mentioned, like, since the dichotomous branch, all those things we mentioned, right? So now we will see what are the different kinds of pathogen which causes diseases. So Peronospora, what are the different Peronospora genus or species are there which causes disease? Onion downy mildew, which is caused by Peronospora destructor; blue mold of tobacco, Peronospora tabacina; spinach leaf mold, which is caused by Peronospora spinacinae; Anthracium downy mildew, Peronospora anthracium; downy mildew of carnation, Peronospora dianthicola; soybean downy mildew, Peronospora manchurica; pea downy mildew, Peronospora viceae; downy mildew of crucifers, which is caused by Peronospora parasitica. These are all the various kinds of downy mildews that is caused by Peronospora genus alone. These are just examples only. And Pseudoperonospora dichotomously branched at acute angle. Examples are cucurbit downy mildew, which is caused by Pseudoperonospora cubensis; hemp downy mildew, which is caused by Pseudoperonospora cannabina; and Plasmopora. Plasmopora is right angle with blunt tips, right? So couple of examples, right? Grapes downy mildew is caused by Plasmopora viticola; sunflower downy mildew is caused by Plasmopora halsteadi; pale millet downy mildew is caused by Plasmopora penicillin; carrot downy mildew is caused by Plasmopora novia. These are all the couple of examples. When it comes to Basidiospora, soul and head over sporangia, we have already discussed, right? This is a soul and head like structure will be there on the sporangia, right? So example is only one example: China aster downy mildew, which is caused by Basidiospora and Entospora. These are all the examples. And other things like Bremia. So Bremia, like tips of the branches are 4, like 4 steric is there, right? So downy mildew, lettuce downy mildew is a Bremia lactose. And the last one is Sclerospora. This is also before it is actually classified into some other like PTLs. Now it is not PTL, some other genera, but it is now it is classified under Peronosporals. So this is generally produces separate branches at the end. So downy mildew of pale millet, which is caused by Sclerospora graminicola, whereas downy mildew of oats, crazy top of corn or wheat and barley is caused by Scleroptera macrospora, previously called as Sclerospora microspora. So these are all the various types of downy mildews along with the examples and descriptions. We do expect questions from this. Sporangospore morphology of Phytophthora. How this sporangospore morphology are like simple or compound or umbellate. Various kinds of morphology are like simple or unbranched. This is how the simple or unbranched like. So this kind of sporangospores are produced by Phytophthora cactorum. So compound are irregularly branched, like 2 branches, like twin branches, like V-shaped. So such kind of branches can be observed: Phytophthora capsi, Phytophthora citropthora, Phytophthora colocaceae. Umbellate. This is umbrella, like umbellate, which is produced by Phytophthora capsica and Phytophthora morinde. And sympodial branching, like one by one, one after layers, like such kind of branching can be observed in Phytophthora caricae. And compound branching, we can see compound branching can be observed in Phytophthora infestans and Phytophthora lychee. So these are all the different kinds of sporangospores morphology. How these sporangospores are producing, like what is the kind of morphology. On the right side we have given easy identification. On the left side we have given what can I say, the pictures of micrograph photos, so for easy identification. So sometimes the pictures what we are observing in the microscope is not that much clear or identical to what exactly we are looking. So generally for identification I have given here, just take a look into. These are all the different kinds of sporangospore morphology and especially Phytophthora species. And we have discussed about asexual fruiting bodies. Now it's time to sexual fruiting bodies. So there are 4 types of sexual fruiting bodies are there: Clistothecium, Apothecium, Perithecium, and naked assay. Actually, naked assay are there. Sir, anyway, Clistothecium means completely closed ascocarp is known as Clistothecium. Completely opened ascocarp is known as Apothecium. Partially opened ascocarp with a narrow opening or mouth is called as a perithecium. Or no ascocarp, generally the spores are produced outside, is known as a naked assay. So naked assay producing fungi, the best example is Taphina deformans. Peach leaf curl is caused by Taphina deformans, right? So such kind of fungi they produce this kind of naked assay. So gymnothecium and pseudothecium, or gymnothecium is also, it is resembles to clistothecium, whereas pseudothecium is resembles to perithecium. So there's no name change only, nothing else, but you remember four kind of sexual fruiting bodies: clistothecium, apothecium, perithecium, and naked assay. Clistothecium means completely closed ascocarp. This is how it look like. Then you can ask me how the spores release out. When it attains a maturity, the pressure will be created inside, so that automatically bursts and the spores will be released outside. So it is completely closed ascocarp, clistothecium. Perithecium means partially opened ascocarp with a narrow or small mouth is called osteal or slit. So that is called perithecium. Completely opened ascocarp. So we can see the ascocarp is completely open. It is called apothecium. No ascocarp. That is called naked assay. Naked means no, right? So this is Taphina deformans that comes under. And these are all the different types of hemathecium, or sterile like structures, are like what kind of what can I say, that hair like structures, or sterile hair like structures, based on their arrangement. There are a couple of hematheciums are there, or like sterile, I would say sterile structures also. First one is a paraphysis, hyphae originating from the base and grows between the asci. See, hyphae originating from the base. This is the hyphae. I will show you just this is a hyphae actually originating from the base. So just take a look into this is uh, where this hyphae is originating from the base. If the hyphae is originating from the base and grows between the asci, it is known as paraphysis. Example is Xylaria species. Paraphysoids, so resembles like pseudoparenchyma with septa. Pseudoparenchyma means they are connected one by one, side by side, like pseudoparenchyma cells. So such kind of growth, if we are observing inside the structure, this is known as paraphysoids. We can observe in petal area. Periphysoids means finger-like projections, were actually developed from the opening that actually growing in the reverse direction or downward direction. We can see here, this is the structure where these finger-like projections are growing a downward direction or reverse direction. They are growing. That is known as paraphysoids. Example is Nectaria. Periphysis means actually they grow towards the ostiole. We can see here periphysites, they actually grow towards the ostiole. That is known as periphysis. Example is gibberella. And the last one is pseudoparaphysis. The hyphae actually originate from the asci and grows downwards between the asci. Actually they grow a reverse direction downwards. That is known as paraphysoids. Example is pleospora. So these are all the different kinds of hemathecium or sterile hair-like structure that is produced in the fungi. It is also very important. Sometimes we don't know, who knows where the questions will come from. So having at least knowledge on the basic topics is a very important. So these are all one of the important aspects when it comes to the last minute preparations. And number of ascospores and produced by, generally all ascomycota group of fungi produces 8 number of ascospores. I told you, right? When it comes to basidiomycota group of fungi produces 4 number of basidiospores. Here there are some exemptions. Couple of fungi, for majority of the ascomycota generally produce 8 only, but there is some exemptions. We'll see that special fungi where the number goes beyond or below. And 2 ascospore producing fungi, phylactinia, and 4 ascospore producing fungi, neurospora, tetrasperma, 5 ascospore producing fungi, capnodium, and also known as the melliola, and 8 ascospore producing fungi. Almost all majority of the ascomycota group of fungi can produce 8 number of ascospore, and 16 ascospore is produced by hypocrea or rufa, which is generally called as a trichoderma, right? So trichoderma is known to produce 16 number of ascospore in an ascus. So 512 ascospores are produced by podospora. And here one ascospores, ascus means it is a sac-like structure where ascospores are produced. So here so far we have discussed ascospore. In an ascus, how many number of ascospore we have discussed right now so far. So now when it comes to ascus, for each generally the fungi produce only one ascus for fruiting body, something like that. So producing fungus, one ascus producing fungus, Podosperia leucotrica and Sparotheca panosa, these are known to produce only one ascus. So these are a couple of important ascospore producing fungi and the genera and the number of ascospore produces. But you remember majority of the ascomycota group of fungi can produce 8 number of ascospore, whereas a basidiomycota group of fungi can produce 4 number of basidiospores. These are all a couple of powdery mildew genera and their general characters. Generally, downy mildews, rust, powdery mildews, smuts, and all those things, various kinds of fungi are there, right? So here are the different powdery mildew genera and their characters. Generally, Erisife produces many ascus. Generally, appendages are myceliaid appendages. It is known to infect barley, cucurbits, black gram, and green gram. One important example is Erisife polygony, which causes a pulsus powdery mildew. Pulsus powdery mildew is caused by Erisife polygony. The next one is the Levelula, which produces many ascus, generally mycelial appendages, so infects chili and red gram. One such example is Levelula tarica, which causes chili powder mildew. The next one is Phylactinia, that produces many ascus, generally produces bulbous kind of appendages. So example is Phylactinia corlea, where it produces where it causes mulberry powdery mildew. I'm just simply explaining what are all the different powdery mildew genera are there along with their important general characters. So we may expect one or two questions like what are the kind of appendages that are produced by Levelilla or actually powdery mildew is caused by. So such kind of examples we may expect. And the next one is the next genera is Uncinella. These all are powdery mildew genera only. Okay. So Uncinella produces many ascus. Generally, Uncinella known to produce circinoid appendages or curved appendages or coiled appendages. So grapes powdery mildew is caused by Uncinella necator. Now it is name changed to Erisife necator. Sparrotheca generally produces single ascus, mycelial appendages. Sparrotheca panos are known to cause rose powdery mildew. Uh, Podospara produces a single ascus dichotomously branched appendages, so known to infect apple, pear, peach, and plum. And cucurbits powdery mildew, which is caused by Podospara xanthi, and apple powdery mildew is known to cause by Podospara leucotrica. And Microsphaeria produces many ascus, again dichotomically branched. Soybean powdery mildew is known to be caused by Microsphaera diffusa. These are all different kind of different powdery mildew genera. Now we will see types of powdery mildew. So there are three important types of powdery mildew are there: oidium type, oidiopsis type, Wöhler-Opsis type. So these all are like I'm just telling the questions may come from this kind of chapters or this kind of topics. That's why I have mentioned all those things. Not theoretically I'm telling, but if you understand the topic in detail about this theoretically, however they are asking, you can simply answer. So just understand the topic. That's why I've just placed it. So there are three types of powdery mildews are there: Oidium types, Oidiopsis type, Wöhler-Opsis type. So from this, especially these types of powdery mildew, it's been years. I saw plenty number of questions, especially at least two questions from this powdery mildew types. So symptoms, generally powdery mildew, Oidium type can produce symptoms on upper surface, where Oidiopsis on the lower surface, whereas Wöhler-Opsis produce symptoms on the both surfaces. Mycelium in Oidium type is hyphae, generally hyaline, septate and ectophytic. Ectophytic means the mycelium is present outside the cells, whereas in Oidiopsis, mycelium is present in endophytic. When it comes to Wöhler-Opsis, hyaline, septate, both ectophytic and endophytic, where the mycelium penetrate inside the cell and present outside also. Hastoria, generally epidermal hastoria is present. When it comes to Oidiopsis type, epidermis and spongy cells are present. When it comes to Wöhler-Opsis type, epidermal hastoria is absent. And conidiospores are club-shaped, non-septate, long-branched when it comes to septate in Oidiopsis type, branched and septate when it comes to Wöhler-Opsis types. Conidia is cylindrical and barrel-shaped chains, and single-celled chains in Oidiopsis, single-celled clavate and club-shaped when it comes to Wöhler-Opsis. See, here what kind of questions we expect? Yes, here they will give you hyaline, septate, ectophytic kind of mycelium is produced. Which of the following? Oidium type, Oidiopsis type, Wöhler-Opsis type. Sometimes they also give examples also. Hyaline, septate, ectophytic type of mycelium is produced. Which of the following fungi? So what are all the fungi that comes under Oidium? First of all, we must know. So the later slide represents yes, these are all the different kinds of example. Cleistothis is generally myceliaid appendages. Myceliaid appendages in a Oidium type. Again, the same kind of myceliaid appendages also can be observed in now Oidiopsis type. But in Wöhler-Opsis, bulbous type of appendages can be observed. So example is Erisife in Oidium, Levelula in Oidiopsis, Phylactinia in Wöhler-Opsis. These are a couple of examples: grapes powdery mildew, Uncinella necator, backgram powdery mildew, Erisife polygony, apple powdery mildew, Podospora leucotrica, rose powdery mildew, Sparrotheca panos. These are all comes under Oidium type. So when it comes to Oidiopsis type, Levelula tarica, which causes chili and PGNP powdery mildew, mulberry powdery mildew, Phylactinia corlea. we must understand something. Both ectophytic and endophytic mycelium, such kind of things I have mentioned. That's why if you see in the mulberry powdery mildew, where the symptoms can be observed in upper surface and the lower surface, there is a meaning for every single thing the describing here. So chains of conidia, we can see this is chains arranging the conidia chain by chain, but chains of sporangia can be observed in albuga. In albuga, the sporangia are arranged in chains, but in chains of conidia can be observed in the powdery mildew type of genera. Here the branches and all those things, ectophytic, endophytic, that was clearly represent these pictures. What kind of questions we will expect? For example, which of the following fungi, both ectophytic and endophytic mycelium, is present? Oidium, Erysife, Libellula, Phylactinae. Answer is Phylactinae, because Ovularapsis type example is Phylactinae. Examples also they may expect. Bulbous appendages can be observed in which of the following fungi. So such kind of things we may expect. So we must aware of all this the data, so that it will be easy to understand the question and answer according. Yes, to have a very easy understanding, I have given a couple of things here. So these are all generally based on clistothecial appendages. How the clistothecia appendages are present, how these mycelial appendages are present on the clistothecia. Based on that, it is divided. So this type of appendages on powdery mildew, generally powdery mildew are produced from this clistothecia. Okay, so it is divided into two: one ascus producing powdery mildews, two ascus producing genera. So one ascus, generally two types of there mycelioid appendages and dichotomously branched appendages. Here we can see mycelioid appendages. So Sparotheca is an example of mycelioid appendages, simply like a mycelium. This such kind of things will be there. When a second one is a dichotomously branched, whereas if the mycelium is branched like a dichotomously, so dichotomously branched appendages can be observed in Podospara. So this is how the Podospara will look like. I repeat once again. So here the types of powdery mildew, this are actually based on the clistothecial appendages. On the clistothecia, what is clistothecia? First of all, it is a completely closed ascocarp. On the completely closed ascocarp, don't worry about this. Here the asci are exposing outside. For our understanding, it is exposing outside. Once it attains maturity, it will burst and come outside. Just for understanding, I just placed this kind of pictures here. There is nothing to worry about it. Generally, clistothecium means completely closed ascocarp is known as clistothecium. So here the types of appendages is divided into two: one ascus and many ascus. One ascus means it produces only one ascus, so that is divided into two. In one ascus, mycelioid appendages are produced by Sparotheca, whereas dichotomously branched appendages are produced by Podospara. These are all the two. When it comes to many ascus producing circinoid or coiled appendages or rounded appendages, it is a very important. It is produced by Uncinella. We can see here Uncinella produces a circinoid or coiled appendages. Bulbous appendages are produced by Phylactinia. We can see here at the base, there kind of bulged or bulbous kind of appendages are there, that is produced by Phylactinia. Mycelioid appendages are produced by Erysife and Libellula. See mycelioid. So here be clear. Mycelioid appendages with a single ascus means Sparotheca. Mycelioid appendages with many ascus means Erysife, where in the Oidium, Oidiapsis, Ovularapsis type we might have observed, right? Again here dichotomously single ascus means Podospara, dichotomously many ascus means Microsparia. We must understand whether it is a single ascus or multi-ascus. Then we can go for what the kind of appendage. It is a circinoid, or it is a bulbous, or it is a dichotomously branched, it is a mycelial. So this is about the types of appendages of powdery mildew. It is also very important. So in the past couple of years, this type of questions also they're asking. So just have go through such kind of topics as well. Here we have mentioned in a in a exam format. So bulbous appendages, very simple. Bulbous means in a single ascus there is no such kind of bulbous appendages are there, right? So in bulbous appendages are actually found in the clistothecia Phylactinia. Phylactinia only produces bulbous appendages, right? So mycelium is partly internal and appendages of perithecia with a basal swelling. Again same only basal swelling, very simple. You just forget about whatever the question they are stretching or extending. You forgot about all the stuff, just imagine and concentrate the stuff. So bulbous appendages are swellings or some kind of bulged appendages. The terminology may changes, but the answer is ultimately Phylactinia. Appendages are bulbous at the base. Very simple. Okay, partly internal asci with simple types of appendages are mycelioid appendages. So they have simple type of appendages or mycelial type of appendages are generally observed in Levilulla. We must know whether it is a single ascus or multiple ascus, then we can go for answer, right? Mycelium is partly internal, perithecia is lacking, generally iodine type. Circinoid appendages are found in the Uncinella. Perithecia with several asci, appendages are coiled at the tips. So coiled appendages can be observed in Uncinella. Circinoid appendages, coiled appendages, or rounded appendages. Uncinella we have seen in the earlier slide, right? So perithecia with several appendages, dichotomously branched in the genus. Microsporium is gender. Perithecia with several asci, they have already mentioned. It is a single ascus or several asci, they have already mentioned. It is a several asci. So Microsporium only produces several asci with the dichotomously branched, right? So we must understand whether it is a single ascus or the kind of question they are asking, so that it will be very easy to answer. So these are all the different kinds of variations that we may expect in exams. Don't think that the same questions will come. Generally, I'm telling this kind of variations we expect. And comes to the next one is different smut genera. From the previous year questions also, we are expecting 1-2 questions from especially this smut. So generally, plenty of smuts are there, plenty of smut genera is also there, but we are going to discuss 4 important smuts in sorghum. So there are 4 smuts are there: grain smut, head smut, loose smut, and long smut. So grain smut is caused by Spatialotheca sorgi. Now it's called Sporosorium sorgi. So just replace the genera Spatialotheca with Sporosorium. Now all the Spatialotheca, now it is replaced with Sporosorium. Wherever the Spatialotheca comes, just replace to Sporosorium. Okay, Spatialotheca sorgi or Sporosorium sorgi. Head smut is caused by Spatialotheca reliana or Sporosorium reliana. Loose smut is caused by Spatialotheca cruenta or Sporosorium cruenta. Long smut is caused by Toliposporium ehrenbergii or Sporosorium ehrenbergii. Actually, this loose smut is caused by Spatialotheca cruenta or Sporosorium cruentum. So here we can expect general pathogen names or causal organisms also, and a couple of other things we expect, like in which smut where the entire inflorescence is infected. Here in the head smut entire inflorescence infected, the remaining all the not inflorescence is infected. Here in the site of ovary also we do observe very clearly, where grain smut, loose smut and long smut only infect ovaries, whereas head smut only the inflorescence infection, where it infects the entire inflorescence. Sora is small when it comes to head smut, very large, medium to long, all those things will be given. Yeah? And the other most important things we do consider is disease survival. Here grain smut is generally externally seed-borne pathogen, whereas head smut is soil borne and seed borne. Again, this grain smut and loose smut both are externally seed borne pathogens, but head smut is a soil and seed borne, whereas long smut is airborne pathogen. The survival years also given here. Grain smut generally survives more than 10 years. Sometimes in exams it is mentioned that 13 years or 12 years also. More than 10 years. You remember that. Based on the options, we can answer it, no problem. Okay, and head smut can survive around 2 years, 2 to 4 years, whereas loose smut can survive 4+ years, and long smut can survive 2+ years. So these are all the different kinds of smut genera in sorghum that actually causes diseases in sorghum. So these are very important. The previous years, like they like they give such certain kind of questions, like they will ask like what is the, whether it is externally seed borne, a grain smut of sorghum, or maybe loose smut of sorghum. They will ask like disease survival, how it is actually survival, externally seed borne, internally seed borne, airborne, or what is the spore viability, how many years it will survive. So such kind of questions were repeatedly asking from the couple of years. And this is also very important. And which of the following smuts of sorghum entire inflorescence is infected? Which of the following smut ovary is infected? All the smuts are infected ovary, but when it comes to head smut, it is inflorescence, it is entire inflorescence. It is a very dangerous, because entire inflorescence is converted into black smut balls. So it is a very dangerous when compared to other smuts. That doesn't mean other smuts are not dangerous. Comparatively, this is a very dangerous. Okay, so this is how the various smuts and their related information. And we have mentioned a couple of aspergillus, penicillium, and in general couple of fungi, and what their common name and what is the actual fungi that is related to. Okay, black mold fungus, generally Aspergillus niger. So if it is in Aspergillus, comes under the Aspergillus group. The common black mold fungus of Aspergillus is Aspergillus niger. Green mold fungus, Aspergillus flavus. How these common names come, you might have a doubt, right? Based on the colony morphology. If the colony morphology is a black in color, it is a black black mold fungus which is caused by Aspergillus niger. If the colony morphology is a green, it is a Aspergillus flavor. If the colony morphology of white, not completely white, a little bit brownish white, or I can say a little bit hyaline. Okay, so that is Aspergillus candida. If the colony morphology is a brown or dark brown, it is Aspergillus tamari. If the colony morphology is yellow, dark yellow, sometimes normal yellow also, it is Aspergillus acriacus. These are all the various kinds of Aspergillus species, that is common names, and they're based on colony morphology. Common name in the sense based on the colony morphology only the names come. Okay, when it comes to Penicillium, blue cheese fungus as per Penicillium roqueforti, whereas blue mold fungus Penicillium italicum, green mold fungus Penicillium digitatum, rot of apples is caused by Penicillium expansum. Based on this three also based on the colony morphology only. The rot of apples are generally caused by the fungi Penicillium expansum. In general, grey mold fungus means Botrytis cinerea, black mold fungus means Rhizopus stolonifer, white mold fungus means Sclerotinia sclerotiarum, peat mold fungus is Astropoderma. These are all a couple of what can I say, the questions we also expect, like the colony morphologies or the common names they may ask. This is also very important. The previous year question papers are in the for namesake only we just told it is a net preparation. So this the kinds of series or the kinds of questions we are preparing is not only meant for net but also meant for JRF, SRF, ARS, whatever the kinds of exams are there related to plant pathology. Our information is definitely helpful. So anyway, so these are all different kinds of questions we expect from this group. Next one is some extra adding information, like Fusarium wilt of banana is also known as Panama wilt. Why it's called Panama wilt? Because that is was first become epidemic in Panama or Costa Rica, Brazil, 1890s probably. That is caused by Fusarium oxysporum pharma species cubensis. Generally, this Fusarium wilt of banana is very severe when it is associated with a nematode called banana burrowing nematode, that is Rhadophilus similis. So in exam, what kind of questions we expect? So Panama wilt of banana is associated with which of the following nematode? A. Heterodera, or Globodera, Rhadophilus, such kind of questions, right? Okay. So this Panama wilt of banana is generally a complex disease that is generally associated with a nematode called banana burrowing nematode Rhadophilus similis. Panama wilt of banana is caused by Fusarium oxysporum pharma species cubens. Generally, application of lime at the rate of 2.5 tons per hectare can be is one of the management practice for the management of the disease. And one more important thing, there are 4 races of banana pathogens, that is Fusarium oxysporum was reported. Those 4 are destroyed. Plenty number of banana varieties. For example, if you take race 1, it is completely wiped out the grass Michael. Race 2, it completely wiped out Blago. It is a cooking banana variety. Race 3, it is almost wiped out Heliconia species. This is not actually comes under this banana, but still this is considered to be as a banana pathogen. Fusarium race 4 Cavendish, it is race 4 tropical race 4 TR4. So the race 4 nowadays it is getting too much importance. It's lots of damage it is causing, especially for the imported banana variety, that is a Cavendish. Whatever the banana, majority of the banana that we are eating for various kinds of other purposes, there is Cavendish banana variety. So now race 4 infects Cavendish. We don't know whether the Cavendish will exist in the future or not. So all the races that infected previous varieties are almost wiped out. Expect the gen banks are expect the some kind of depositions are there. Now they are not cultivated anywhere. So now the race 4 infects Cavendish. So in exams we may expect like a race 1 infect, race 2 infect, race 3 infect, or race 4 infects which banana varieties. So such kind of infects also we may expect. So here they mentioned TR3 do not produce chlamydospore. It is mentioned that tropical race 3 do not produce any chlamydospore. Generally, Fusarium is known to produce microconidia, macroconidia, and chlamydospores, right? But TR3, this Heliconia species infecting Fusarium, it is not producing any chlamydospores, it is reported. And Fusarium is actually a diversified pathogen, and it's a very dangerous soil-borne pathogen. And the wilt of couple of other pathogens like Fusarium causing disease I have mentioned here. And wilt of tomato is caused by Fusarium oxysporum pharma species Lycopersity. Wilt of cotton is caused by Fusarium oxysporum pharma species Vas infectum. Fusarium oxysporum pharma species cubensis causes generally wilt of banana, just now we discussed, right? So these are all a couple of important information about Fusarium. So these are all the different kinds of genera and the spore characters. Since you guys might have know very well about it. So generally alternaria is known to be both vertical and horizontal septations. We can call it as muriform septations. We can call it as transverse vertical and horizontal septa. There are various kinds of names are there. Muriform septations, vertical and alternate septations are some kind of various names will be there, right? So this is hypomycetes anamorphic fungi. These are all sunflower and sesame leaf blade is called alternata, and early blight of potato is caused by Alternaria solani. There are various kinds of disease that is caused by Alternaria. Botrytis generally bunch like grapes. If you see it, it will produce spores like a bunch like grapes. On the pictures, we can clearly observe bunch like grapes. Generally causes mold fungi, right? So Helminthosporium or Botrytis, we know very well the brown leaf spot of rice, Bipolaris varizae, or maize tersicum leaf blade, which is called Helminthosporium medis. This is how the spores is produced. Lots of septations are there in between. And cercospora like it is like some kind of multiseptated cylindrical kind of spore it will produces, and couple of septa will be there, around 4 to 12 kind of septa we can observe here, and generally causes groundnut early thicca disease, cercospora rachidicol and cercospora berkel, and all the things of disease that is caused by cercospora. And this is the continuation Phariosauropsis. This is how the spores look like. Generally causes groundnut thickle spot Phariosauropsis tucumensis. There is a new name, I think so. Fusarium. Generally, Fusarium is known to produce three kinds of spores: microconidia, macroconidia, and chlamydospores. Generally, sickle-shelled or sickle-shaped or shoe horse-shaped conidia it produces. Generally, cotton wilt is caused by Fusarium oxysporum forma species vas infectum. Chili Fusarium wilt is caused by Fusarium capsicii. Pyricularia, conidia are hyaline, pale grey, and biseptate. Generally, pyriform shape of conidia is produced by Pyricularia. Generally causes rice blast. Verticillium. Again, here we can observe some kind of chains of conidia can be observed here. Generally, Verticillium wilt of cotton is caused by Verticillium dahliae. Rhizoctonia, it produces right angle mycelium, right? Right angle kind of mycelium is produced by Rhizoctonia. Generally causes rice sheath blade Rhizoctonia solani, dry root of pulses, Rhizoctonia betatricola. And Sclerotium, it is a very hard resting structure or mustard shaped structure. For everything we have given pictures here. In the some kind of white color or some kind of brownish, or in the rare cases we can also see an orange color. But generally it's a brown color, reddish brown color or brown color mustard shaped structure that is produced by the pathogen during a hard in unfavorable environmental time in order to survive. So generally stem rot of groundnut which is caused by Sclerotium ralpsi. In Sclerotium only one species are there, Sclerotium ralpsi, that is the only one species. On the acervular imperfect fungi, which means all fungi which produces acervuli, so from where the spores are produced. Okay, so Coltotricum, first one is generally how we can observe Coltotricum. This is the conidia where in the central oil globule is present. So presence of oil globule at the center is the identification feature of this Coltotricum. Sugarcane red dot which causes Coltotricum falcatum. And Pestalozziopsis, generally Pestalozziopsis contains 5 cells, 3 middle cells and 2 outer cells. So generally causes coconut grey spot and guava fruit canker. And Pestalozziopsis which actually produces 6 cells, 4 middle cells and outer 2 cells, 6 cells. Guava leaf spot Pestalozziopsis cd. And Gliosporium, this genre is actually differentiated from Coltotricum based on the CTA and in the acervuli. So grapes anthraconos, which is caused by Gliosporium ampliophagum. And Ascochyta, Phomopsis, these are all the different kinds of spores and their general features. So we may, to have a knowledge, we may also differentiate the other spores, and these are all the general spores. There are plenty of other spores as well. Since we have mentioned only a couple of general spores. Philosticta, Macrophomina, Diplodia, Botodiplodia, Septoria, these are all the different kinds of diseases caused by and how it is actually look like. Now when it comes to different shapes of conidia, so how the conidia shape is look like? Sickle shaped conidia, it look like exactly sickle shaped or half crescent shaped or half moon shaped or shoe horse shaped. We can also say shoe horse shape, right? So sickle shelled conidia is generally produced by Fusarium and also Coltotricum capsicii, and flame shaped conidia it is produced by apple scab. Mushroom shaped conidia is produced by Claviceps fusiformis, this is mushroom shaped conidia. Rhamphodial shaped conidia which is produced by Philactinia. So these are all some shapes how actually the conidia is the shape of the conidia they are mentioning, nothing else. And vermiform or multicelled conidia is produced by cercosporum and cercosporum. Just now I told you cercosporum produces some kind of filiform shaped with a multisepted 4 to 12 kind, more than 15 also sometimes. Okay, pyriform or pear shaped conidia is produced by pyriculare. So pyriculare conidia is actually look like a pear shaped. So that's what it's called as a pyriform conidia, pyriculare conidia. Muriform conidia or muriform septations or multiseptations are generally found in alternaria. And filiform conidia, mycospheril and septoria, it look like exactly filiform. And this cercospora and cercosporum also look like filiform like, right? Different shapes of ascospore. So far we have seen conidia. Now we are going to see ascospores. Okay, spindle shaped ascospore. So spindle shaped ascospore is produced by Spermoptera gossipi and Erymothecium corley. So this is actually look like a spindle shape like this. So generally spindle shaped is like this. So such kind of symptoms where we observed rice blast, which is produces spindle shaped spots, right? So generally spindle shaped ascospore is produced by Spermoptera gossipi and Erymothecium corley. And hat shaped ascospore, generally the ascospore is like hat shaped. Hat shaped ascospore is produced by Ascoidea rubensis. Filiform shaped ascospore or long narrow shaped ascospore, which is produced by claviceps. Generally claviceps produces filiform shaped ascospore. So don't confuse. Before we have seen cercospore and all those things are filiform shaped conidia. Now they're asking filiform shaped ascospore. So it is claviceps. Okay. And filiform shaped ascospore, this Cochiliobolus is also known to produce filiform, but it is not actually the filiform. Okay, generally they are telling filiform, but it is actually related to that claviceps. Boat shaped or footprint shaped. If you look at this, it is exactly look like a footprint, right? So footprint shaped or boat shaped ascospore is produced by Venturia inaequalis, apple's cap, right? So slipper shaped ascospore is produced by Pleospora. It is exactly look like a slipper. So slipper shaped ascospore is produced by Pleospora. These are all the different kinds of ascospores and their shapes. Now regarding the wheat stem rust. So to understand wheat stem rust, we must understand some other things. What is autoecious? What is microcyclic? What is macrocyclic? Autoecious means all 5 spore stages are not present. Heteroecious means, sorry, just a minute. Microcyclic means all 5 spore stages are not present. Macrocyclic means all 5 spore stages are present. Autoecious means the fungus finishes the life cycle in a single host. Heteroecious means the fungi requires 2 hosts. Now we will see. Autoecious means only one host. Macrocyclic means all 5 spore stages are present, and which means the fungi finishing the life cycle, which consists of 5 spores in a single host itself. Example is BFSS. So these are all the list of fungi that can comes under this category of autoecious macrocyclic rust, where they finishes the entire life cycle in a single host. They are producing all the 5 spores, iridium, aecium, iridium, pycnium, aecium, iridium, telium, bestium, and everything. They are producing in a single host only. Examples are bean rust, which is caused by Euromyces appendiculatus, flax rust, Melonsora lini, sunflower rust, Puccinia helianthi, safflower rust, Puccinia carthami, castor rust, Melonsora ricini. These are all the different kinds of autoecious macrocyclic rust. Okay. Now autoecious demicyclic. So first of all, we must understand what is a demicyclic. Demicyclic means lack of iridial stage. So out of all the 5 spore stages, iridial stage is lacking. So lack of iridial stage is known as a demicyclic rust. So autoecious means the fungi completing the entire life cycle in a single host. So which means iridial stage is lacking, but finishing the entire life cycle without iridial stage in a single host only. Best example is rubus orange rust, which is caused by Gymnoconia picciana or Gymnoconia nitensis. Heteroecious means the fungus requires 2 hosts to complete the life cycle. Macrocyclic means all the 5 spore stages are present. Very important. The best example is wheat stem rust, which is caused by Puccinia graminis triticea, and also one more example, white pine blister blast, that is the Cronartium rubicola. Heteroecious macrocyclic rust, best example is wheat stem rust, because in wheat stem rust 2 hosts are present. That happens actually in the different hosts. Wheat primary, iridium and telium will happen. And when it comes to Barbary, which is completely different family, whereas Aecium and Picnic will happen, right? So which means 2 different hosts where all the 5 spore stages are happening in different locations and different hosts. That is a very complex life cycle. Of course you guys know very well. So anyway, heteroecious macrocyclic rust example is a wheat stem rust, and another example is white pine blister blight. And heteroecious demicyclic rust. Demicyclic means I told you already, lack of iridial stage is known as demicyclic rust. Heteroecious demicyclic rust example is COR, cedar apple rust, which is caused by Gymnosporangium juniperae virizina. These are all the causal organisms. So don't be confused about autoecious demicyclic rust means rubus orange rust. Heteroecious demicyclic rust means COR. Cedar apple rust. Okay, alternate host for cedar apple rust is apple. I repeat, I already told you, demicyclic means lack of iridial stage is known as demicyclic. Example is cedar apple rust and robust orange rust are the example. So just I am telling, both in autoecious and heteroecious demicyclic rust, one stage is lacking, that is iridial stage. So what are all the fungi that comes under demicyclic? They are asking cedar apple rust and robust orange. If they are mentioning particularly autoecious demicyclic and heteroecious demicyclic, we can tell autoecious demicyclic example is robust orange rust, whereas heteroecious demicyclic example is cedar apple rust. So these are all the couple of examples about rust, and these are all the spore stages of fungi. Various spore stages are here. Five spore stages we do study in wheat stem rust, right? Pycniospores, aciospores, iridospores, teleospores, and basidospores. Pycniospores is generally stage 0, aciospore is stage 1, stage 2 iridospore, stage 3 teleospore, stage 4 basidospore. Generally, pycniospores is known to produce one number of germ pores, whereas aciospores produces 6, iridospore produces 4, teleospore produces 2, basidospore produces 1. These are all the number of germ pores. Pycniospores are generally hyaline in color, whereas aciospores are generally yellow in color. So iridospores are golden brown in color, whereas the teleospores are black to dark brown in color. Again, basidospores are hyaline in color. And when it comes to the condition or nature, pycniospores and basidospores are haploid in nature. Remaining all spores are dikaryotic, whereas teleospores can able to perform as the dikaryotic and diploid. Okay, generally pycniospores and aciospores are happens in upper and lower surface respectively on the Barbary host. This Barbary is a alternate host, very important. Iridospores and teleospores generally happen on the upper surface, that is on the primary host, whereas the basidospore generally happens in the soil debris. This table alone is a very important to understand the rust very easily. If we understand this table, we can easily answer the questions regarding rust. So generally pycniospores happens in upper surface or lower surface. Generally pycniospores happens in the upper surface of Barbary. So imagine there is a Barbary. On the upper surface pycniospores are produced. On the corresponding lower surface, inverted cup shaped lower surface, we might have studied inverted kind of pycnidium can be observed in where? Aciospores inverted, because it is producing on the lower surface of Barbary. But both the pycniospores and aciospore happen on the Barbary only, but pycniospores on the upper surface, aciospores on the lower surface. We must understand this. It is a very important. Sometimes number of germ pores also they will ask. Pycniospores 1, aciospore 6, iridospore 4, teleospores 2, basidospore 1. So this is a type of this is about the spore stages and the information related to rust fungi. And generally rust produces iridospores. Iridospore is known as the repeating spores. We also known as the summer spores. The condition is n+n. The color of the rust spores, that is iridospores, are golden brown in color. Generally they are single celled. For the management of rust disease, we do recommend oxycarboxyn. The trade name of oxycarboxyn is a plant wax. When it comes to smut, smut produces generally teleospores. Teleospores are also known as the resting spores, also known as the winter spores. The condition is n+n, generally dark brown in color, black dark brown in color, which are bicelled. Teleospore generally bicelled, right? For the management of smut disease, we do recommend a fungicide called carboxyn, and the trade name of carboxyn is Vitavax or Vitavax. Here iridospore, why it is called repeating spore? Because they are repeating, continuously repeating, and they are the spores which are spreading. Teleospores are resting spores because they are not repeating, simply taking rest when the during off seasons, right? So and they'll germinate and produce the basidospores in the later stages. So generally rust are localized infection. Foliar parts only to infect. Very rare cases to infect flowers, less than 1% of the times. So when it comes to smut, it causes systemic infection. The main concentration is inflorescence organs. That's why it's very dangerous. That doesn't mean rust is not dangerous. Of course, both rust and smuts are deadly dangerous pathogens. So this is about various kinds of spore stages and the general information about rust and smut diseases. And the types of teleospore, it is also very important here. Single celled teleospore. The teleospore is a single cell. So a single celled teleospore is produced by Euromyces, Malomsora, Pacospora. So generally the type of teleospore which is produced by Malomsora, even though the Malomspora is produces crust-like teleospore, but the type of teleospore is single-celled only. Pacospora is also okay. So here we are just giving the types along with the example. Generally single-celled, where the teleospore is only one cell. Such kind of teleospore is produced by Euromyces, Malomspora, and Pacospora. And the Euromyces is very important one. Double-celled teleospore is produced by Puccinia and Gymnosporangium. They produce double-celled teleospore. Very important one is Puccinia. Among all the spores all the spores in the wheat stem rust, there is only one double-celled spore, that is a teleospore. Remaining all the spore, pycnospore, asciospore, iridospore, basidospore, all are single-celled except teleospore, which is a double-celled or bicelled. Multi-celled teleospore is produced by frag medium. We can see here there are multiple number of cells in iridospore, that is produced by frag medium. Turnip-shaped teleospore or kidney-shaped iridospore or hump-beaked iridospore is produced by hemilia. Hemilia generally causes leaf rust, right? Coffee leaf rust. So if you see here, the teleospore is actually turnip shaped, whereas the iridospore is actually kidney shaped. That's why it's produced two kinds of spores: turnip shaped or napiform teleospore, or kidney shaped iridospore, that is produced by hemilia, vastatrix or hemilia. Crust like teleospore, where the teleospores are combining one by one, adjoining, they look like a crust. So earth crust. So crust like teleospore is produced by melonsora, and parachute like teleospore, it look like a parachute, right? So parachute like teleospore is produced by ravinelia. These are all the different kinds of teleospores along this Paxenia. These are all the different kinds of teleospore along with the example. It's also very, very important. In the previous years, if you see, at least there is a one or two questions from this section. It is also very, very important. The next one is alternate hosts of rust disease and other plant pathogens that I've given here. Wheat stem rust, the alternate host is just now we discussed. Microcyclic means autoecious means only one host where the entire life cycle is finished. When it comes to heterocyclic, the pathogen requires two hosts, a primary host and a secondary host. Secondary host is alternate host. Alternate means which they are surviving on the secondary host, which not belonging to the same family of the primary host. Anyway, wheat stem rust, which is caused by Paxenia graminis triticale, primary host is wheat, whereas secondary host is barberry. Barberry is not belonging to the family of wheat. They are belonging to another family, that is barberries. Wheat leaf rust, which is caused by Paxenia regondita. The primary host is wheat, secondary host is Thalictrum species. Sorghum rust, Paxenia purpurea. Primary host is sorghum, the secondary host is sleeping beauty, which is we can call it as Oxalis cornicula. It is also a very important one. And pearl millet rust, which is caused by Paxenia striata, and the primary host is pearl millet and the secondary host is brinjal. Maize leaf rust, which is caused by Paxenia sorgi, primary host is maize, secondary host is oxalis. White blister rust, Cronartium rubicola, the primary host is currant, secondary host is pines. Cedar apple rust, which is caused by Gymnosporangium juniperae virginale, primary host is apple and the secondary host is also apple. Crown rust of oats, which is caused by Paxenia coronata, primary host is oats, secondary host is buckthorn, that is Rhamna species. So these are all the different kinds of alternate hosts of rust disease and their pathogens which is actually responsible for. And related to others, so before we have discussed alternate hosts, now it comes to collateral hosts which belong to the same families. So the disease generally disease, pathogen, primary host and collateral hosts have been given here. So the rice blast, which is caused by Pyracularia varizae, the primary host is rice, secondary host is Lersia exandra, both are belonging to the family of Poaceae. And blight of rice, that is bacterial blight of rice, which is caused by Xanthomonas varizae, Pathovar varizae, primary host is rice, secondary host is Lersium exandra in the world, Cyprus rotundus in India. Downy mildew of maize and sugarcane, Peronosplerospora philippinensis and Peronosplerospora sakari, which are maize and sugarcane are the primary hosts respectively. But when it comes to alternate hosts, sorghum halafens and Saccharum spontaneum are the alternate hosts. Downy mildew of or green ear of kambu, so which is caused by Sclerospora graminicola. The primary host is bajra, kambu or pal millet, and the collateral host is Echinocola species and panicum species. Sugarcane smut, which is caused by Sporosorium cetaminium. Now before it is Histologos cetaminium, now it is name changed to Sporosorium cetaminium. So primary host is sugarcane and the secondary host, that is collateral host, is Saccharum spontaneum. So these are all about various kinds of alternate hosts and collateral hosts along with their the information related. Now we'll see the different forms of rust based on the absence or presence of spore stages. So generally 0 stage is pycnial stage, or which produce pycniospores. Stage 1 is the acial stage, stage 2 iridial stage, stage 3 teleal stage, stage 4 basidial and basidial stage. Now based on the presence and absence, they have other names. For example, U type means U means true type. Generally U means true. U type means 0, 1, 2, 3 are present and 4 is absent, that is basidial stage is absent. Brachy type means 0, 2 and 3 are present but 1 is absent. Opis type means 0, 1 and 3 are present but 2 is absent. Hemi-type means 2 and 3 are present, 0 and 1 are absent. Microtype means only type 3 is present. Leptotype means only type 3 is absent. These are all the different types based on the presence or absence of spore stages in the rust. And these are all the list of different rust genera which causes diseases. Let's start with the Puccinia. It is one of the largest genera which causes general rust disease. Starts with the wheat stem rust, which is caused by Puccinia graminis ptitis, brown rust Puccinia regondita, orange rust or yellow rust Puccinia striformis, brown rust of sugarcane, which is caused by Puccinia melanocephala, orange rust of sugarcane, which is caused by Puccinia coni, hollyhock rust, which is caused by Puccinia heterospora, white rust of chrysanthemum, which is caused by Puccinia horrida, sunflower rust, which is caused by Puccinia helianthi, safflower rust, which is caused by Puccinia calcitripe ware centurica, generally called as Puccinia carthame. Sorghum rust is caused by Puccinia purpurea, kambu rust is caused by Puccinia striata. Maize rust Puccinia sorghia or Puccinia folispora also, mint rust Puccinia menthe, barley rust Puccinia hordic, oat crown rust Puccinia coronata. So these are all the different rust disease which is caused by the genera Puccinia, and other genera is also there. So from this what we can expect is just like causal organisms, so different kinds of causal organisms. So we may expect Hemilia, only one important disease was there in Hemilia, that is a coffee rust, which is caused by Hemilia vastatrix. When it comes to the genus Euromyces, these are all a rust genera only. Okay, so the Euromyces, bean rust, Euromyces appendiculators, and Euromyces fascioli, gram rust, Euromyces caesari, pea rust, Euromyces pisci, and Euromyces fabae, carnation rust, Euromyces dianthe, jasmine rust, Euromyces habsoni, alfalfa rust, which is caused by Euromyces striatus var medicanginis. When it comes to Melamsora, another genus of rust, linseed rust, Melamsora lini, popular rust, Melamsora medusae, castor rust, Melamsora ricini, willow rust, Melamsora epite. And Pacospora, it is another genus of rust, soybean rust, Pacospora pacarizi, cotton rust, Pacospora gossipi, grape rust, Pacospora uvitis. And when it comes to the next genera, Phragmidium, rose rust is caused by Phragmidium mucronatum or Phragmidium tuberculatum, raspberry rust, Phragmidium rubeoidea, blackberry rust, Phragmidium violacearum. When it comes to the genus Gymnosperangem, there is only one disease, important disease, the cedar apple rust, which is caused by Gymnosperangem chuniberry verizinane, Ravinelia, which causes a gooseberry rust, that is caused by Ravinelia emblica, and last one is a Cronatium white pine bristle blast, which is caused by Cronatium rubicola. So these are all the different kinds of rust genera which causes diseases in plants. So in questions in exams, like which of the following is a rust genera, or cotton rust is caused by, or maybe just mentioning the different kinds of rust diseases. So this is this table is very useful to just identify the different kinds of rust genera along with the disease caused by them. And generally we have discussed about ascospores, right? Each ascos contains 8 number of ascospore, and rarely other exceptional cases are so there. Generally each basidium, when it comes to basidium group of fungi, each basidium contains 4 number of basidiospores. There is an exemptions. 9 basidiospores are produced by a fungus called Phallus impodicus. 2 to 8 number of basidiospores are produced by a fungus called Exobasidium vexans, that is a T-bacillus blight, right? 8 to 16 number of basidiospores are produced by Tilicia caris. 14 to 30 basidiospores are produced by Tilicia controversa. 32 to 128 number of basidiospores are produced by Tilicia indica. So generally basidium contains 4 number of basidiospores only, but there is an exemptions. So these are all the list of exemptions that I have mentioned. Generally in fill in the blanks also we might have gone through this, like embryo infection. Which of the following disease is an example of embryo infection, which generally infects embryo? That's what it's called, embryo infection. If it is infecting floral pods, it's called floral infection. If it is infecting seedling infection, it is infecting shoot infection. So like that it is divided. So these are all the very important one we must consider. Embryo infection, otherwise called as ovariculous smut. Ovaries or embryos, all the stuff is same, right? So more or less similar the connection. Okay, embryo infection, also known as ovariculous infection. The best example is wheat stem rust, because the wheat stem rust infects or enters through embryo or ovaries. That's why it's called embryo infection. And floral infection means the best example is Kumbu smut, which is caused by Tolposporium penicillare. It is a best example of floral infection because where it infects the floral parts. Seedling infection, the best example is wheat flag smut, which is caused by Urocystis agropyrae, which is the best example of seedling smut. And the shoot infection, also known as Culmiculus smut, the best example is whip smut, which is caused by Histologocytaminium, now name changed to Sporisorium cytaminium. These are all the different types of smuts along with their modes of infection or where it is actually infecting. This is also very important, we must consider, because in the last 3-4 years I never see any questions related to this slide, but if you see in 2018 probably, yes, I wrote in 2018 there is a question from this slide, and along with in 2019 also there might be a question, I think so. So not about just net, to having the broad spectrum knowledge, these concepts are very important. It's a very few smuts are there, right? Smuts and rust are there, right? So it's better to go through this as well, and not only for net but also for various other kinds of entrance exams as well. Now these are all the different kinds of spore types or conidia types according to the Secadian classification. We know very well who is Secado. She wrote a book called Silum Fungorum, right? Secadian classification is a very famous systematis. So based on the cell number or based on the number of cells in the spores, they all are asexual spores only. So they have different names. If it is a single celled, we can take a look into this picture here. It is a single celled. Shape may be different, but the cell is single, right? So single celled or aseptate spores are known as amerospores. Two celled, which means one septum will be there. Two celled, which means one septum will be there, that is called didymospore. Multi-celled, multiple number of, which means multi-septum will be there, and multiple number of spores, or sorry, multiple number of cells are there. So it is called a phragmospore. Star shaped, because the spore look like a star, that's why it is known as the star shaped or cell-led spore. For example, starospore. Muriform septation, it is a very important. Both vertical and horizontal septa, the presence of both vertical and horizontal septa is known as muriform spore. The best example is alternaria spore, right? We can also call as generally called as dictyospore. Filiform or wormy shaped scolicospore, where we can see here like filiform. Helical or spiral shaped, because the spire is the spore is like a spiral, so that's what is known as spiral spore. Example is helicospore. Hand shaped spore with like, what can I say, like a finger like projections, or I can say the basal from the conidium multiple number of cells are divided. This type such kind of thing is called a chirospore. Let me come once again. Single cell is called amerospore, two celled is didymospore, multi-celled is called phragmospore, star shaped is called starospore. Muriform, which means both vertical and horizontal septa, is called as dictyospore. Filiform or wormy shaped, it is scolicospore. Helical spore or spiral called is helicospore. So these are all the different kinds of asexual spores, or we can also call as vegetative spores. We can generally call as asexual spores based on the kinds of septations. This is especially the classification based on the Secadian type, right? And these are all the list of monographs. We used to study the monograph of Fusarium was written by, or monograph of that was written by, like, so these are all I have prepared some kind of the list of monographs. We may expect sometimes. Generally, monograph of Pythium was written by G. Rangasamy. Monograph of Alternaria was written by Verma. Helminthosporium A.P. Mishra. Monograph of Downy mildew and powdery mildew was written by D.M. Spencer. Cercospora, monograph of Cercospora. In India, it was written by R.S. Vasudeva in 1953. In world level, it was written by A.C. Chep in the world level. So monograph of fungal genus Cercospora, world, just now we discussed Chep, okay. Monograph of Taphrina was written by A.J. Mix, 1949. Monograph of erysipelas, that is especially powdery mildew, that was written by U. Brand. Especially powdery mildews, and just concentrated on erysipelas only. Monograph of downy mildew of crucifers was written by Nashat Shaharan and Verma. Hypomycetes and allomycetes was written by C.V. Subramaniam. And genera of rust was written by M.J. Tirumulachar. And Pachyderman cephalosporium, again M.J. Tirumulachar. And monograph of Phytophthora, which was written by Irvine and Reboro. Indian wheat rust, E.J. Battler. Fusarium, it's a very important, repeatedly asked question. This was monograph of Fusarium was written by C. Booth in 1971. Here is also important, okay. And other couple of monographs I've given here. Monograph of Helminthosporium, A.P. Mishra, Smut, Common Venki, Verticillium wilt, Peg and Brandy, Fusarium wilt of Tomato, J.C. Walker. And this J.C. Walker is also known for phenolic theory, right? Catechol and protocatechol, due to the presence of two phenolic compounds, catechol and protocatechol, radianians are resistant to onion smut, which is caused by Coltrotrechum circinens, right? So this physiognomy of tomato, which was written by J.C. Walker, and physiognomy of cotton was written by J.P. Verma. And monograph of histovaginals, M.J. Tribulachar, Aphanomyces, double C squat, and Chinoporaceae, Kirk, Sclerotinia, Corn, Ketomitaceae, M.S. Chytridiales, Carling, and Hypoxalan, Miller. Ceratocystis and Ceratocystopsis, Upadhyay, Pleospora and Segregatus, Wemeyer, Labolemon, Labolbenaceae, okay, a little bit confused, like Labolbenaceae, Thaxter, Clavari and allied genera, Corner, Claveraceae in India, thinned, Latinus Pedger, monograph of Tipitate steroid fungi, Reed, and Cornell's India, Subramanian and Shaker, and Indian slimy molds, that was written by Lodi. So these are all the different kinds of rust genera, sorry, monographs, and their the scientists involved in. So sometimes from the past many years, it is only repeated questions are asking, especially monograph of Fusarium only. But we don't know. Maybe in the future, we don't know. Not all the time the same person is taking the question paper, right? So maybe the mind also may changes. So I've just given some couple of list of monographs for your references, and as well as other preparations as well. Coming to bacteriology, generally single-celled, I would say, like based on the morphology, there are two kinds of bacteria. There coccus, which means just like round kinds of things, and rod shaped, that is called, we can say, rod shaped generally. Cocca means a singular, and then cocca there are a couple of types are there. Monococcus means a single spherical bacterium, that is monococcus. Diplococcus, diplo, means two, just a pair of spherical bacteria. Two spherical bacteria is known as Diplococcus. First of all, coccus means spherical bacteria. First of all, we just remember that. And tetracoccus means we can see here a group of four spherical bacteria. These are group of four spherical bacteria is called tetracoccus. Okay. And streptococcus means a chain of spherical bacteria arranged in a row. We can take a look here. A chain of spherical bacteria arranged in a single row, not exactly in a straight line, even in bending also. Okay, like in a chain, right? Staphylococcus, cocci are generally formed irregular in shape, like I would say grape bunch, like you can see here, grape bunch, like are irregular in shape. And Sarcina, generally a cocci are arranged in a cube of 8. The first 4 spherical bacteria were there. On the back side, another 4 spherical bacteria just look like a cube, right? So a cocci are arranged in a cubes of 8, that is called Sarcina. So these are all based on morphology. So one type is coccus, that is generally round shaped or spherical in shape. The next type is rod shaped. These are all the different shapes of bacteria: spherical or ovoid or coccoid, micrococcus, rod shaped or bacillus shaped, generally known as straight rods, Escherichia coli and bacillus. I'm just on the left side, the shape of the bacteria. On the right side, what are the examples that belong to the shape? Spherical or ovoid or coccoid means micrococcus. Rod shaped or bacillus, it is Escherichia coli and bacillus. Comma shaped or curved rod, it's a vibrio. Flexuous rod, it's a cytophaga. Generally helical shaped spirillum, kidney shaped or coroniform shaped clavibacter. The fusiform shape example is Fusobacterium fusiformis, bidiformis bifidobacter. Star shaped ankylomicrobium, dichotomously branched gallionella, bat shaped hypomicrobium, coiled or spiral shaped spirochaete, coiled or spring shaped leptospira. So these are all the different shapes of bacteria. Along with the examples, there is nothing to worry about it. Majority of the plant pathogenic bacteria are rod shaped, that to gram negative rod shaped. Okay, majority of the bacteria are gram negative and rod shaped. Okay, these are all other shapes also there, but the majority of rod shaped, then nothing to worry about it. And flagella, we in the couple of slides before, we might have discussed this flagella in fungi, right? So the same types of flagella is also there in bacteria, but based on the number and based on the arrangement here, it may changes. Let's see. The flagella is generally locomotor organ. Now we will see what are the different kinds of flagella and the details along with the examples. Generally, A trichus. A means no. Trichus means flagella. Trichus means hair-like structure. That is generally flagella. Flagella is a hair-like structure, right? So A trichus means no flagella are present, or there is no flagella. Example is xylella. We can see this one. A trichus means there is no flagella. Example is xylella. Monotrichous or polar plazid. Mono means single. Only one hair-like structure will be present on any one end. Maybe not all the times it is need to be in the right side. Sometimes it may be also in the left side as well. Okay. So any one of the corner, one flagella will be there. Best example is Xanthomonas. Amphitrichous. Amphitrichous means one flagellum is present at both ends. So at both ends, one flagellum will be there. The best example is Pseudomonas species. Peritrichous. Peri means many. So peritrichous means the flagella is present all over the body. So there is no exact place or location. So generally the flagella will be attached all over the body. It is called as peritrichous. The best example is Irvinia. And also a couple of other organisms generally produces peritrichous flagella. Pantovia, Escherichia coli, and Clostridium. These are all the best example for peritrichous flagella. And Lopotrichous. Lopo means generally I would say a tufts of flagella or a multiple number of flagella are generally attached to the both ends. We can see here multiple number of flagella. So Lopotrichous this is. Example is Spirulina. And Cephalotrichous. Cephalo means head. Generally cephalo means head. So which means for head hair will be there only one side. In the meantime, in the Cephalotrichous flagella, multiple number of flagella is only present to one side. We can remember it that way. So the best example is Pseudomonas fluorescens. Circumtracheal flagella is agrobacterium. So on the for our understanding another computerized picture that I have given here. So these are all different types of flagella, and every year there is a question from this. You can just take a look into the last 10 years previous question paper. Every year there is a question from this. And if you want, you can also take a look into our channel. We have already published all the net videos almost from 2006, 2007, 2009, 2014, 2014 1, 14 2, 2018 1, 18 to 2019, 21, 23. It's been almost more than 10 plus videos we have published on nets and preparation as well. You just take a look into the various kinds of question paper. There is a question from this types of flagella, or maybe example, or maybe what it is actually called, like that. So there might be some framework or some kind of questions from this table as well. So just take a look into this. And genetic recombination and organisms. So in bacteria, majority of the plant pathogenic bacteria are reproduced by means of binary fission, but couple of other bacteria and recombinations also discovered, like I would say sexual, I can't say sexual recombination, yes, sexual recombination, not reproduction, recombination, that's right only. Okay, so there are three kinds of sexual recombination can be discovered in bacteria: conjugation, transformation, and transduction. I'm not going to explain in detail about all these mechanisms, but just tell you what kind of questions we may expect. Like conjugation was discovered by what is the organism the conjugation is discovered, or maybe transduction was first discovered in, like that. So based on the previous experiences, I have prepared a table here. Conjugation was discovered in an organism, Escherichia coli, that was discovered by Lederberg and Tatum in the year 1946. When it comes to transformation, transformation was discovered by Griffith in an organism called pneumonia in the year 1928. Transduction was discovered in the organism Salmonella, that was discovered by Lederberg and Zinder in the year 1952. If you just take a look into all the basic information came in this table itself. You just remember CTT, conjugation, transformation, transduction, that must match with the organism CPS, Escherichia coli, pneumonia, and Salmonella. That gives an exact order and answers as well. So this table is also very important. At least we must remember the basic kinds of stuff, so that it will be easy to answer if the question is in a normal way. So for the deep preparation, it will definitely take a time. Since it is a last minute preparation, we just put some most important kind of things only. And these are all the bacteriocins which are produced by the different organisms. Bacteriocin means it is a kind of proteins or a kind of substance that is produced by the bacteria, right? So here we have given what are the bacteria and what is the bacteriocin or protein kind of substance that is produced by the bacteria. So the Escherichia coli produce a bacteriocin called colicins. Erwinia keratovora, Pathovora keratovora produce a bacteriocin called keratovorin. Bacillus subtilis produces a bacteriocin called subtilicin. Pseudomonas aeruginosa produces a bacteriocin called pyocin. Agrobacterium radiobacterium produces two kinds of bacteriocin. We can also call as biocontrollers. Agrocin K84, agrocin K1026. This K84 are selling with the trade name of Galtrol, whereas 1026 is Nogal. And Pseudomonas produces a bacteriocin called Pseudobacter. So these are all the different bacteriocins that are produced by the different bacteria. So sometimes we may expect also. Now coming to stainings, so different types of stainings of bacteria. Generally simple staining. We have plenty number of staining. Generally we know gram staining that was given by Christian Gram in 1884, and what we know is general staining. There are plenty number of stains are there, like types of stainings are there, like if you for identification of flagella or identification of presence of endospore or capsules or various kinds of stainings are there. Here simple staining is generally recommended for the morphology and arrangement of bacteria. Whatever we are doing in the laboratory, generally simple staining, like morphology, whether it is a rod shaped or how the bacteria and arrangement, like in a coca or something like that. So basic arrangement we use this simple staining. Flagella staining, the name itself indicates, just use it to identify the flagella, number of flagella, how many number of flagella are there, what is the flagella, like type, like atrichus or monotrichus, such kind of things we can detect by using this flagella staining. Endospore staining used for the identification of bacterial endospores. We know very well this endospores are the bacterial resistant spore that is produced by the bacteria like Bacillus subtilis and Clostridium. So these two types of bacteria only where they produce specialized spores are called endospores. We can also say heat resistant spores. Okay, for the identification we can recommend endospore staining. For capsule staining, identification of capsule, acid fast staining is just to distinguish acid fast bacteria from non-acid fast bacteria. And gas mask stain, which is used for rickettsia and some kind of protozoal organisms, and cytoplasmic strains just demonstrate what all the kinds of internal organisms are there, like deposits are there, like maybe starch or glycogen or phospho, polyphosphates or hydroxybutirates and other kinds of substances. To have an idea, we will go with cytoplasmic inclusion staining. These are all the different types of staining which are we are employing in bacteria. These are all commonly used some kind of stains in general gram staining. So we'll start with the common mordant used in gram staining. That is the commonly used mordant in gram staining is tannic acid. As I said, gram staining was first developed by Sir Hans Christian Gram in 1884, just to differentiate the gram positive and the gram negative bacteria. So here the commonly used mordant in gram staining is tannic acid. So mordant generally means it is a kind of substance or like insoluble compound, and that actually fixes the color with the bacterial cells or components. So based on the cell wall thickness, or based on the cell wall composition, like peptidoglycan or mucopeptides, polysaccharides and lipopolysaccharides. If you see gram positive contains a majority of the amount that mucopeptides, around 85%, right? So if you to see gram negative contains just 3 to 12%, but the uppermost layer is polysaccharides, whereas mucopeptides are there, but it is there in the middle layer, almost in the middle layer in gram negative, whereas when it comes to gram positive it is there in the upper layer. So for first is two stains it fixes, and when it comes to gram negative it just washes out the polysaccharides on the upper layer. So that is how actually the reaction works. So here the primary stain in gram staining is a crystal violet 1%. Simple staining is methylene blue. Trapping or mordant agent is iodine solution. The last one is a decolorizing agent is 95% alcohol. The last one is a counter stain that is used in saffron. These are also the repeatedly asked question when it comes to exam. So this question, like a counter stain used in gram staining, this is a continuously asked question for a couple of years, and this slide is also very important regarding gram staining. And pigments in phytopathogenic bacteria, what are the different kinds of pigments that is produced by the bacteria, and what is the color of the pigment that I have mentioned here. The first one is Burkholderia glume. It is produces a toxin or pigment called taxoflavin. Color is not mentioned here. Catrobacterium flacum fascians pathover poinsettia. It produces a pigment called carotenoid. This carotenoid is actually yellow in color, and it is a water soluble pigment. Clavibacter michiganensis subspecies michiganensis, which is a carotenoid pigment, which is generally yellow in color and again same water soluble. Irvinia herbicola and Irvinia iridovera, which produces a pigment called zeaxanthin big diglucoside. It produces a pigment called yellow carotenoid. Irvinia chrysanthemi, it produces a pigment called indigo. The color is blue, which is a water soluble. Irvinia rafansati, and which produces a pigment called proferazamine. These two are that was asked very long years to now. Irvinia chrysanthemum indigodeon and Irvinia refansity proferazamine. Proferazamine generally a pink color pigment. Irvinia rubrifaciens, it produces a pigment called rubrifacin, pink color pigment. Pseudomonas aeruginosa and Pseudomonas fluorescens, which produces a pigment called pyoverdin, yellow color fluorescent pigment. Xanthomonas, very very important, produces a pigment called xanthomonid, which is yellow in color and water insoluble, it don't soluble in water. Pseudomonas aeruginosa, pyocyanin, it produces a blue color pigment. Pseudomonas cepacia, which produces phenazine, which is yellow color, green, yellow color or sometimes green or purple in color. Pseudomonas aeruginosa, pyorubrin and pyomelanin, which is a red color or brown color pigments. So these are all the different kinds of bacteria and the pigments produced. So in exams we may expect like what is the pigment that is produced by the xanthomonid, what is the color of the pigment, or Irvinia refansity producing a pigment is known as. Such kind of questions we may expect. Irvinia refansity producing a pigment is known as indigodein. So such kind of questions we may expect. Here are the couple of important antibiotics that is produced by the plant pathogenic bacteria, especially, sorry, not plant pathogenic bacteria. For the management of plant pathogenic bacteria, what are the different kinds of antibiotics we are exploiting in PDM, plant disease management, especially that is origin of streptomyces. So streptomycin is known to be the first broad spectrum antibiotic. The 80% of the majority of the industrial antibiotics are the antibiotic that we are using in plant disease management, that is originated from the streptomyces, right? So let's see what all the different kinds of antibiotics are produced by, what all the different kinds of disease that we can manage by using this antibiotic. So here antibiotics are two types, antifungal antibiotics and antibacterial antibiotics. Since it is mentioned as a streptomyces, we are going to see only bacterial antibiotics. Now we'll see streptomycin, cyclohexamide, that is produced by, both are produced by Streptomyces gracious, effective against the downy mildew of beans, which is caused by Phytophthora fascioli, Peronospora tabaci, Phytophthora infestans, Erwinia amylovora. Chloramphenicol is produced from Streptomyces venezuelae, neomycin is produced from Streptomyces fradde, kanamycin is produced by Streptomyces kanamycetes, amphotericin B is produced by Streptomyces nodosus. Oxytetracycline, that is a terramycin, is produced by Streptomyces remosus, generally recommended for fire blight of apple. Tetracycline, generally Anchromycium, Penicillium, polycycline, sticycline, terricin. These are all the different kinds of antibiotics produced from Streptomyces gracious or Streptomyces aureofaciens, generally very active against pataplasmal and spiroplasmal disease. Blasticidin, which is produced by Streptomyces graciochromogenes, that is especially active against rice blast. So in exams they will ask like streptomycin is produced from, or chloramphenicol is produced from, or tetracycline is produced from. Mostly they will ask origin, where it is actually originated from, what is the bacteria where it is actually originated. But the regarding management, very rare cases we do expect management, but most of the cases what I have observed is produced by, or like it is produced from, where it is actually produced from. Such kind of questions we do expect. And there are a couple of other antibiotics. Chlorotetracycline, that is Aureomycin, Streptomyces aureofaciens, cyclohexamide, Streptomyces gracious, nystatin, Streptomyces neurosi, hyamycin, Streptomyces pimpirina, erythromycin, Streptomyces erythraeus, oleandromycin, Streptomyces antibioticus, carbomycin, Streptomyces alstedi, novobiocin, Streptomyces niveus, Vira A, Streptomyces antibioticus, vancomycin, Streptomyces orientalis, cosmycin, Streptomyces cosgensis. It is a very very important, that is a very active against rice blast and fire blight of apple. So these are all the list of important antibiotics that too especially produced from Pseudomonas species and their origin sources. So from this we may expect sometimes. So mostly streptomycin is streptomycin sulfate is originated from a bacteria called Streptomyces gracious or Penicillium notatum. Now name changed to Penicillium chrysogenum. Not name changed. Now majority of the Penicillium is produced from Streptomyces chrysogenum rather than Streptomyces. Penicillium chrysogenum rather than Penicillium notatum, because this Penicillium chrysogenum produces 100 times more yield than the Penicillium notatum. That is the thing. Okay, anyway, these are all the different kinds of antibiotics that is produced by Penicillium. Streptomycin what we have size. Now we'll see the different kinds of antibiotics that is produced by penicillin. So penicillin is also known to produce some kind of certain kind of antibiotics, but in at present we are not using this penicillium antibiotics because almost all bacteria become resistant to the penicillium antibiotics. Anyway, we'll see what are the different kinds of penicillin antibiotics. Antibiotic penicillin is isolated from a fungus called Penicillium notatum and Penicillium chrysogenum also. And griseofulvin, it is isolated from Penicillium griseofulvum, Penicillium nigricans, Penicillium album, and Penicillium urticae. So this griseofulvin is recommended for powdery mildew of rose, Paratheca panosa, and Alternaria solani, Sclerotinia fructigena. So these are all the different kinds of fungi that can be managed by using this penicillium antibiotics. When it comes to the antibiotics that is produced by the bacillus, so bacillus is a biocontrol agent known, right? So it is also known to produce some kind of certain kind of antibiotics. We'll see what are all they. Bacitracin, eumycin, and bacillomycin that is produced from the fungus Bacillus subtilis and Bacillus licheniformis, which are gram positive, and gramicidin and tyrocidin, Bacillus brevis, Serexin and Biocerin, Bacillus cereus, Bacitracin and Licheniformis, Bacillus licheniformis. These are all are gram-positive bacteria. When it comes to polymyxin B, that is actually produced from a fungus called Bacillus polymyxa, which is a gram-negative bacteria. So these are all the other list antibiotics that is produced from Penicillium and Bacillus species. Now we'll see the classification of important antibiotics, that is secondary metabolites which are produced from, especially what exactly the antibiotics belonging to the group, which group they are actually belonging. Now we'll see. The first one is beta lactams. So beta lactams group, there are two antibiotics are there, penicillin and cephalosporin. Penicillin isolated from a fungus called Penicillium notatum, whereas cephalosporin isolated from a fungus called cephalosporin species. Both of these beta lactam are very active against the cell wall leakage and inhibits the murin synthesis. So coming to the second one, mucorolides, erythromycin and carbomycin. So erythromycin are isolated from Streptomyces halsteadi and Streptomyces erythraeus. Carbomycin is isolated from Streptomyces halsteadi. So these both macrolides, that is erythromycin and carbomycin, inhibits 15S ribosomal functions. Uh, Amyloglycosides, streptomycin, neomycin, and kanamycin. Streptomycin is isolated from Streptomyces griseus. Neomycin is isolated from Streptomyces fridae. Kanamycin is isolated from Streptomyces kannamycetes. So all this amyloglycoside fungicide which inhibits 30 ribosomal functions. So tetracyclines, that is oxytetracycline and chlorotetracycline. Oxytetracycline is isolated from Streptomyces rimosus. Chlorotetracycline is isolated from Streptomyces aureofaciens. Both inhibits 50S ribosomal functions. This polypeptides, polymyxin B and bacitracin. So polymyxin isolated from polymyxa, Bacillus polymyxa and Bacillus subtilis from bacitracin. So this they inhibits the destruction of cytoplasmic membrane and inhibition of murin synthesis respectively. So here, what kind of questions we do expect is, they do ask the penicillin belonging to the group, or maybe what is the mode of action of the penicillin, or streptomycin is a antibiotic or first broad spectrum antibiotics belonging to the group, or isolated from, or what is the mode of action of the neomycin. So such kind of questions we do expect. So this is about it. And other things, pollens from pollen groups, chloramphenicol, amphotericin, nystatin, ariofungin, and natamycin. These are all a couple of pollen antibiotics. So this chloramphenicol is isolated from Streptomyces venezuelae. This question was not in NET exam, maybe some kind of entrance exam. I saw this question couple of years before. Anyway, this chloramphenicol is one of the most important antibiotics. Generally inhibits 50S ribosomal functions and translocation of ribosomal functions. Amphotericin, Streptomyces nodosum, nystatin, Streptomyces norisci, ariofungin, Streptomyces cynomyces varitericola, and natamycin, Streptomyces natalensis. So these are all the different kinds of fungicide that comes under pollen group. And now we'll see the couple of suitable medias. What are the different kinds of suitable medias that is recommended for bacteria? Generally, for fungi, PDA medium is recommended. For bacteria, Na medium is recommended, nutrient agar medium. But among the standard mediums, we have a couple of suitable mediums or selective mediums for the very good growth and characters of the bacteria. We'll see what are all they. So the first one is the D1 medium. D1 medium is recommended for agrobacterium. or agrobacterium tumefaciens. Now it is called as Rhizobium radiobacter based on the 16 SR RNA function. So this crown gall of apple, crown gall of rose, that is caused by agrobacterium tumefaciens. Now it is called as Rhizobium radiobacter. So generally this tumefaciens, that is agrobacterium occurrence, or majorly infects Rosaceae family, apple, rose, and some other stone fruits as well. And D1 medium is meant for agrobacterium or Rhizobium radiobacter. D2 medium is meant for Corynebacterium. So Corynebacterium, now it is called as Rathibacter tritici. Corynebacterium tritici are called now it is called as Rathibacter tritici, which causes a wheat seed gall nematode, like some kind of yellow ear corcle disease, right? Yellow ear rot disease of wheat, which is generally kidney shaped gram positive bacteria. D3 medium is recommended for Irvinia. D4 is recommended for Pseudomonas. PD2 is a medium that is recommended for Xylella fastidiosa. We know very well Xylella fastidiosa, the first plant pathogenic bacteria to be completely genome sequence, which causes pierced disease of grapes, right? So it is known to be transmitted by glassy winged sharp shooter, Homolodesca coagulata, Homolodesca vitripennis. So those are all the scientific names of the glassy winged sharp shooter. Not only this glassy winged sharp shooter, there are other vectors like 3 or 4 other vectors also known to be reported to transmit this pierced disease of grape vine. So these are all a couple of suitable medias. Other media like SC medium, fastidious xylem limited bacterium, the sugarcane ratoon stunting. Again, in this FEB, fastidious vascular bacteria, we must discuss the groups, that is xylem limited and phloem limited. In xylem limited, again, it's two types, gram positive and gram negative. So coming to the gram positive, it is sugarcane ratoon stunting, which is caused by Lifesonia xylis subspecies xylis. This question was 3 times asked in the national eligibility test, if you take a look into the previous years. So sugarcane ratoon stunting, previously it is called as Clavibacter xyli subspecies xyli. Now it is called as Lifesonia xyli subspecies xyli. Xylem limited gram negative bacteria is pierced disease of grapevine or pierced disease of grapevine, which is caused by Xylella fastidiosa. So the next one is a phloem limited. In phloem limited, there is only one bacteria is there, gram negative, that is a Candidatus liberibacter asiaticus, which causes a citrus greening, as known to be transmitted by psyllids, Diaphorina citri. So this SC medium is actually recommended for fastidious vascular bacteria. And since I have mentioned phloem limited, Candidatus liberibacter asiaticus, which causes citrus greening. In Asia it is caused by asiaticus. In America it is known to be caused by americanus, and in Africa it is known to be caused by africanus. So it is known to be transmitted by psyllids everywhere. And a sex medium, which is recommended for Xanthomonas campestris, Pathovar campestris, which causes a black rot of cabbage. So black rot of cabbage is caused by Xanthomonas campestris, Pathovar campestris. It enters into the plant cells through hydatodes. Hydatodes are known to be the entry points of this Xanthomonas campestris, Pathovar campestris, and this Xanthomonas is known to produce a pigment called xanthomonidin, which is an yellow color pigment. Just now we discussed it, right? So different kinds of pigment that is produced by the plant pathogenic bacteria. So this Xanthomonas is known to produce an yellow color pigment is called xanthomonidin. And these are all our other couple of suitable medias: crystal violet pectide medium, Irvinia species, potato sucrose peptone medium, Xanthomonas species, mannitol glutamic acid tetrazolium chloride, Pseudomonas solanacearum, Kingsby medium, Pseudomonas fluorescens, arginine glycerol mannitol salt, actinomycetes, nutrient agar, almost all bacteria, BCBR VC Pseudomonas fluorescens, limabin agar medium, Pathoptera sojae, sugarcane, sucrose peptone agar, Xanthomonas, Pseudomonas, Irvinia amylovara, glycerol aspirin medium, Streptomyces, C3GH medium is meant for Spiroplasma. These are all the other couple of suitable medium recommended for the growth of various kinds of bacteria. And this table is very important, like mode of entry of plant pathogens. How actually not plant pathogens, especially I would say plant pathogenic bacteria, only bacteria. So we'll see the different mode of entry of plant pathogenic bacteria. So let's start with the hydatodes. Just now we discussed a black rot of cabbage, which is caused by Xanthomonas campestris, Pathovar campestris, and bacterial leaf blight of rice, which is caused by Xanthomonas varaceae, Pathovar varaceae. Here they do ask like black rot of cabbage enters into the plant cells through, or which of the following pathogen is known to enter into the plant cells through hydatodes, or maybe trichomes, or maybe stomata, or maybe some other kinds of things. So here simultaneously we do study two things. One is mode of entry. The second one is the causal organisms which actually causes the diseases. So we do expect any kind of things, maybe entry points as well as causal organisms as well. Okay. Anyway, generally hydrothodes, two important pathogens are there. That doesn't mean the remaining pathogens are not entering to hydrothodes. What we mentioned is couple of only important pathogens. Okay. Hydrothodes, black rat of cabbage or brown leaf spot of bacterial libellate of rice. Floral pods or nectar is a very important thing is fire blight of apple and pear known to be caused by Irvinia amylover enters into the floral pods. Lenticels, two important pathogens, potato scab and soft rot of vegetables. Potato scab is caused by Streptomyces scabis, soft rot of vegetables is caused by Irvinia carotovora. Trichomes, bacterial canker of tomato, which is known to be caused by Clavibacter michiganensis, subspecies michiganensis. And through stomata, there are a couple of pathogens like bacterial leaf state, which is caused by Xanthomonas varaceae, Pathology varaceae. Citrus canker, Xanthomonas citri, Pathology citri. Angular leaf spot of cotton or black arm of cotton or bacterial blight of cotton, all are same only. It is called as angular leaf spot, also known as black arm of cotton, also known as bacterial blight of cotton, which is known to be caused by Xanthomonas actinopodes, Pathology malvacearum, or Xanthomonas citri, Pathology malvacearum. Common blight of beans, Xanthomonas campestris, Pathology fascioli. Leaf spot of tomato, Xanthomonas campestris, Pathology vesicatoria. So these are all a couple of entry points. This is a very very important one, and this questions was asked multiple number of times in multiple exams. And couple of other things we have mentioned here. Through wounds, soft rot of vegetables and stalk rot of maize is known to be caused by Irvinia and known to be entered through wounds. Crown gall of stone fruits, Agrobacterium tumefaciens, various Penicillium species, Penicillium expansum, Penicillium metallicum, Penicillium digitatum are known to be transmitted by, entry through wounds. Through insects, so major we do generally studies plant viral disease only known to be transmitted by insect vectors, and a couple of bacterial diseases also known to be transmitted by insect vectors. We have mentioned only two here. Probably in the next slides we have mentioned other important insect vectors that is actually act as insect vectors for transmission of plant bacterial diseases. Let's see this insect bacterial wilt of corn known to be caused by Pantovia stivati. Previously it is called as Irvinia stivati, later name changed to Pantovia stivati. The Pantovia is the scientist who discovered it. To respect him, it was just name changed to Pantovia stivati. It is known to be transmitted by an insect vector called cornflea beetle or simply beetle, Chitocnema pulicaria. Next one is the bacterial wilt of cucumber, which is caused by Irvinia trachyphylla. It is known to be transmitted by two vectors, typed cucumber beetle, Diabrotica victata, Racalama victata, or spotted cucumber beetle, Diabrotica duodecipunctata, or Diabrotica undecipunctata. This is a very important. These two questions were repeatedly asked in many exams. Through root hairs, Rhizobium enters, and through nematodes, wheat ear-corkel nematode, Anguina tritici or Rathaibacter tritici that enters the nematode ear-corkel, and bacterial wilt of carnation, which is caused by Parabarcolderia caryophily, that generally enters through a nematode called Melodogene incognita. So these are all the different kinds of entry points. These are all very important. Every year there was a question. If you just take a look into the previous year net question papers, so for every question paper are so far the published all the model papers, we do have a pasted a list in the comment section, all the list that will take you or redirect you to the previously published videos. We also suggest you to just take a look into that videos as well for better understanding. Regarding FEB, fastidious vascular bacteria. So regarding fastidious vascular bacteria, I think we have already discussed. Anyway, let me tell you in detail. So this fastidious vascular bacteria is a kind of bacteria especially limited to xylem and phloem vessels. There are two types of fastidious FEBs, the xylem limited and phloem limited. This is also kind of bacteria only, nothing to confuse. So this is xylem limited, two types are there, gram positive and gram negative. So the example of xylem limited gram positive bacteria, that is sugarcane ratoon stunting, is the Livesonia xyli subspecies xyli, previously called as Clavibacter xyli subspecies xyli. When it comes to gram negative xylem limited, Pierce disease of grapevine, Xylella fastidiosa. It is known to be transmitted by insect vector called glossy winged sharpshooter. Homolodesca coagulata is the previous name. Now the insect vector name changed to Homolodesca vitripennis. When it comes to phloem limited gram negative are there. That is a Candidatus liberibacter asiaticus, which causes a disease called citrus greening, also known as Huan Longping. It is a Chinese name which actually represents yellow dragon. So it look like a yellow dragon because half of the fruit is green, half of the fruit is remaining yellow. So that's why it's called a Huan Longping disease, which is known to be transmitted by psyllids. Diaphorina citri. This is an African uh, generally causes. It is an insect vector for African greening. And regarding Ralstonia solanaceae, which generally causes bacterial wilts, right? So Ralstonia solanaceae, which having almost the widest host range, over 200 species it do infect. And the why it is named as Ralstonia, because the name was given by Ralston. So he is a scientist or researcher who worked on. And there are 5 races of Ralstonia is recorded in the world so far. It is a very important. You just take a look into 2023, 2021, 2019 question paper. There is a question from this slide. It's a very important. Not only this slide, every single slide is important, but these are all extra important, like a VVVIP, right? Okay, just a joke. Anyway, race 1 infect Solanaceae, which means like we can take all the Solanaceae crops, potato, tomato, all the Solanaceae crops, whatever it comes under. Race 2 infects Musaceae. Musaceae means musa, banana. Race 3 infects only potato. Of course, this potatoes also comes under Solanaceae, but race 1 infect all Solanaceae crop, race 3 infect only potato. Race 4 infects ginger, race 5 infects mulberry. So what kind of questions we do expect from this is like Ralstonia solanacearum, which causes various kinds of diseases, wilts or bacterial wilts or whatever it is. They do ask like race 1 infects or race 2 is known to be infects. They don't ask Musaceae. There sometimes they may also mention the specific crop, especially banana. And the majority of the disease I do observe this race 2 infect Musaceae or banana or Musa species. So these are all related to banana only, right? So race 2 infect banana, race 1 Solanaceae, race 2 Musaceae, race 3 potato, race 4 ginger, race 5 mulberry. So these are all the couple of important things we must remember. Just now I told you like Pierce disease of grapevine is caused by Xylella fastidiosa. This Xylella fastidiosa is also known to be causes various diseases like Pierce disease of grapevine, citrus variegated chlorosis, almond leaf scorch, phony peach disease, coffee leaf scorch, alfalfa dwarf, olive quick decline, oleander leaf scorch. All the diseases are known to be caused by fastidious xylem limited gram negative bacteria, that is Xylella fastidiosa. It is the first plant pathogenic bacteria to be completely genome sequenced by A.J.G. Simpson in 2000 with the help of Brazilian consortium. So among I cannot say this one is important, that one is important. All the diseases are important. Sometimes they do ask Pierce disease of grapevine is caused by. Sometimes they may be we do expect citrus variegated chlorosis, almond leaf scorch, phony peach disease, coffee leaf scorch. There are various kinds of diseases that is known to be caused by Xylella fastidiosa. This Xylella fastidiosa is the best example for xylem limited gram negative bacteria. Right? We have already saw, right? It is a kind of FEB. Okay. And these are all the couple of other information related to this Xylella fastidiosa, that is the first plant pathogenic bacterium. This is the first plant pathogenic bacterium to be completely sequenced as Xylella fastidiosa. In 2000, with the help of a Brazilian consortium, the scientist A.J.G. Simpson sequenced this Xylella fastidiosa. It is known to be transmitted by an insect vector called glossy winged sharpshooter. I told you, right? Not one, there are 4 other vectors also known to be involved. So what we know is a glassy winged sharpshooter. The scientific name of glassy winged sharpshooter is Homolodisca coagulata. Now it is called as Homolodisca vetripennis, and also known to be spittlebugs like Cercopis Woolenata and spittlebugs and frog hoppers are also known to be caused by spittlebugs, generally called as frog hoppers, also known to be transmitted the disease. And here the sharpshooters, all the glassy winged sharpshooter or whatever it is a sharpshooter, mostly it is belonging to the family Cicadelidae, and spittlebug is also spittlebug generally belonging to the family Cercopidae. And as I told you, not only one vector, and it is also known to be there are more than 3 or 4 vectors vectors are known to be transmitted to this vector, but majority of the vectors are glassy winged sharpshooters only. And other vectors like blue-green sharpshooter, Graphocephala atropunctata, green sharpshooter, Dracocephala minerva, red-headed, and Carneocephala fulgida, and glassy winged Homolodisca coagulata. These are all the different kinds of sharpshooters or different kinds of vectors which are known to be transmitted the this disease, that is a Pierce disease of grapevine or citrus variegated chlorosis, all are okay. This is about couple of other information about this Xylella fastidiosa. And generally bacterial food storage organ, since in the general plant pathology itself we have mentioned like a bacterial food storage organ. What is a bacterial food storage organ? Poly beta hydroxybutyrate, that is a reserve food material of fungi, right? So they do store food material in the form of glycogen or oils. So in exams, how do we expect is what is the bacterial food storage organ? Poly beta hydroxybutyrate or beta hydroxybutyrate or hydroxybutyrate, in any way they will ask. But we must remember PHB, poly beta hydroxybutyrate, is the food storage organ or food storage product of bacteria, where the food is stored in the form of glycogen or some certain times oils or other forms, but generally glycogen. Okay, this poly beta hydroxybutyrate is a lipid-like structure where they generally joins with ester-like linkages, and the granules are diameters around 2.2 to 0.7% in diameter, and the PHB used as a energy and carbon source for the either nucleic acid metabolism, and this PHB supplies the energy material for the nucleic acid metabolism. And some bacteria produce copolymers of PHB referred as poly beta hydroxybutyric hydroxybutyric acid acetate, PHA. So PHB means poly beta hydroxybutyrate, PHA means poly beta hydroxybutyric acetate. So some bacteria also produce PHA also. What we used to study is PHB, poly beta hydroxybutyrate, but some bacteria is also known to produce PHA, poly beta hydroxybutyric acetate. And the staining agent for bacterial food storage organ is Sudan black bee. What is the bacterial food storage organ? Poly beta hydroxybutyrate or beta hydroxybutyrate. What is the stain used to stain this bacterial food storage organ? Sudan black bee, right? So bacteria, couple of bacteria, not all the bacteria, will produce some kind of heat resistant spores are known as endospores. Endospores are highly resistant to heat. There are two important bacteria is known to produce this endospore, that is a Bacillus and Clostridium. Both are gram positive only. So in BSc agriculture, everyone starts with the BSc agriculture, right? B stands for Bacillus, S stands for Streptomyces, T stands for Corynebacter or Clavibacter. Organ also known as the Clostridium. These are all the only bacteria that comes under gram positive. Remaining majority of the plant pathogenic bacteria are gram negative only. These endospores are highly resistant to heat just because it contains an acid called DPA, dipicolinic acid, which is made up of calcium ions, that provides you high standards or high ability to withstand against the heat. Generally, this DPA, dipicolinic acid, contains 5 to 15% of the bacterial dry weight. So whenever the unfavorable conditions will occur, this endospore can survive during unfavorable time. Again, whenever the favorable conditions will occur, again it starts to be like a new bacteria. So this is how the mechanism works. Some G plus C guanine and cytosine content of different plant pathogenic bacteria. Maybe sometimes we do expect in a couple of years this kind of questions was also there. So actinomycetes, which produces which is having a 63 to 78% of the G and C content of DNA. Agrobacterium 59-63, Clavibacter 65-75, Dickeas 57, Irvinia 50-58, 50 to 58, Lifesonia 68, Pseudomonas ralston and Burkholderia 58 to 70%, Pantovia 53 to 60%, Xanthomonas 63 to 69%, Xylella 52 to 55%, Prokaryotes generally 30 to 75%, Pseudomonas aeruginosa 67%, and Rhizobium Meliloti 61.6% to 65.6%, and Gonorrhea 50 to 53%, Micrococcus luteus 70.7 to 75.5%. It is known to be the highest GC content of the bacteria. Among the bacteria, it is the highest GC content. Klebsiella pneumoniae 56-58, Azospirilla brasilensis 70-71, Escherichia coli 50 to 51%. It's not a 50.51, it's a 50 to 51%. Clostridium tetani 25%, Bacillus subtilis 40 to 43, and Campylobacter fetus 32 to 35, and Candidata salivary bacter around 36.5%. These are all the GC content. and some important bacteria and the scientists involved with their discoveries. So we know this various kinds of important bacterial genera like Erwinia, Agrobacterium, Pseudomonas. So who discovered this, or what is the scientist who named it, and what is the year of discoveries? From this section also, and previously we have lots of questions. Let's start with the first one, Agrobacterium. So Agrobacterium was named by Kohn, Kohn in the year of 1942. Acetobacter bigernic 1898, Acidovorax Williams 1990, Arthrobacterium Kohn and Dimick 1947, Azotobacter bigernic 1901, Bacillus Kohn 1872, Burkholderia Yabuchi 1995, Clavibacter Davis 1984, Clostridium Pramwoski 1880, Corynebacterium Lehmann and Newman 1896, Cartobacterium Yamada and Komagata 1972, Enterobacter Edwards and Hormachi 1960, Erwinia Winslow, Nocada 1920, Nocada Trevesian 1889, Pantovia Gavini and Margaret 1899. Sorry, 1989. So these are all some important bacteria. So let me tell you, this Agrobacterium, Bacillus, Clavibacter, Erwinia. These are all the important one that we do observe repeatedly in many exams. Not only this, couple of other things also there. Pseudomonas mugula, very important. Pseudomonas mugula 1894, Ralstonus and other couple of scientists 73 and 96 respectively. Rhizobacter Gatto and Frank 1988, Rhizobium Young and some other scientists 1989, Rhizomonas Van Braggen et al 1990, Rhodococcus Zoff 1891, Serratia Bizio 1823, Spiroplasma Davis 1972, Streptomycin Waxman and Henry C 43, Xanthomonas Dawson 39, Xylella Welseta 87, Xylophilus Williams et al 1987. These are all the couple of other bacteria and the discovered scientists along with the year. From this section also we have lots of questions in the previous exams. So going through is very important. From this section, Pseudomonas is important. Spiroplasma Davis, that was studied in other sections also. Xanthomonas Dawson, Streptomyces Waxman and Henry C. And I cannot say only this kind of questions only come, but generally I'm telling. So like, what are the probability areas we must look into? That's what we are trying to make a point. And these are all the different kinds of resting spores that is produced by the bacteria. So prokaryotes generally what we know is only fungi known to produce spores. Some kind of bacteria also known to produce, that is endospores only, nothing else. Some other kinds of things, conidia, some other kinds of things, very specific bacteria are there, I will tell you now. So different kinds of resting spores are Bacillus, Clostridium, Sporosarcina, Sporolacto, Bacillus, desulfomaculans, thermoactinomycetes. These are all the bacteria they produce a kind of spore is known as endospore, which means the spores are produced inside the bacteria. That's why it's called as endospore. And metallotrophic bacteria like Methylococcus, Methylobacter, Methylomonas, Methylospara, and Rhodomicrobium vinnelli. These are all the couple of bacteria. They produce a spore called exospore, which are the spores are formed outside. It's called exospore. Actinomycetes is known to produce conidia, which are produced outside only. Cyanobacterium, that is a blue-green algae. Aconites, it's a kind of spores. Myxobacterium, myxospores. Azotobacter generally produces cyst. Dellovibrioid produces a dellocyst. These are all some other different kinds of spores, an exemption kind of spores that is produced by the various plant pathogenic bacteria. And the kind of pigments here mentioned, I think this question was asked 1 or 2 times. Bacterial responsible for blue color in the milk, which means if the milk is contaminated with blue color production of pigments. If you just take a look into the outside, we can see the colonies, right? Some kind of moldy growths or some kind of things will be there. If it is in blue in color, it is known to be caused by Pseudomonas syncytia. If it is in red in color, it is known to be infected or caused by Serratia marsans. If it is a blue-green combination, it is called Pseudomonas aeruginosa. If it is in yellow in color, it is known to be caused by Pseudomonas cinzantha. If it is a brown in color, it is caused by Pseudomonas putrefaciens. These are all a couple of pigments that is produced produced in the milk. Blue color pigment Pseudomonas syncytia, red color pigment Serratia marsans, blue green color pigment Pseudomonas aeruginosa, yellow color pigment Pseudomonas cinzantha, brown color pigment Pseudomonas putrefaciens. These are all the most important things that we must take a look into. And now the phytotoxins that is produced by the Pseudomonas species. So what are the different kinds of phytotoxin that is produced by only Pseudomonas? However, we have saw the antibiotics that is produced by only Streptomyces. Now we're going to see the phytotoxin produced by only Pseudomonas. Pseudomonas is known to be the largest genera that causes plant diseases. This is the number one. If you just list out what are all the genera that causes more number of plant diseases, the Pseudomonas is in the list one. The second one is Xanthomonas, and other bacteria comes under later. So the Pseudomonas is known to cause more number of diseases. Now we will see what are the different kinds of toxin that is produced by the Pseudomonas along with the production organism and what is the chemical compound and mode of action. The first one is coronatin. Coronatin is produced by Pseudomonas syringe pathover Atropunctata, Pseudomonas glycinia, Pseudomonas macrocola, Pseudomonas macrosporum, which is produces a chemical agent, a biosynthesis organ is polyketins. So generally chemical class. So mode of action is mimics the methyl jasmonates, and corpectin, which is produced by Pseudomonas correlata, which is a lipopeptide, membrane activates, and fucopeptin, along with one more is persicomycins, which are produced by Pseudomonas fiscovaginae and Pseudomonas syringe pathover persicae. So generally lipopeptides and generally a fatty acid chemical group they belong to, and this is very important. Fasciole toxin, which is produced by Pseudomonas syringe pathover syringe, and is also known to be produced by Pseudomonas fasciolicola. So it inhibits ornithyl carbamyl transferase and inhibition of amino acid synthesis. Mode of action is also very important, and mostly the toxin and the production organisms only we are expecting, and sometimes we cannot expect other things also they may be asked. And rhizobiotoxin, which is produced by Pseudomonas andropogonis. The mode of action is hematocystin synthesis, and syringomycin. This is also very important, which is produced by Pseudomonas syringe pathover syringe or Pseudomonas syringe and pathover fiscivaginae, which increases the host membrane permeabilities. Syringopeptins, which are known to be produced by Pseudomonas syringe pathover syringe, ergosterol and spizoglioides biosynthesis actually do. So these are all the different kinds of phytotoxin that is produced by the organisms. And other things also mentioned here. Syringopeptins, which are produced by Pseudomonas syringe, pathover syringe, ergosterol and same sphingolipid biosynthesis. Tap toxin, Pseudomonas syringe, pathover tabaci, beta-lactam group inhibits glutamine synthesis. Tage toxin, Pseudomonas syringe, pathover tagetis, chloroplast RNA polymerase. Tolacin, pathover Pseudomonas tolaci, induces the release of calycin from LUVs, and viscocin, which is produced by Pseudomonas marginalis, Pseudomonas fluorescens, that is ferulinin, and taxoflavin, which is produced by Pseudomonas glumi. I think this was asked one time, I think so. Torpolone, which is produced by Pseudomonas plantari. These are all a couple of toxins and the organisms produces. Yes, as I told you, majority of the viral diseases are only known to be transmitted by vectors, right? So here couple of plant bacterial diseases also there, that is also known to be transmitted by insect vectors. Now we will see what are the list of important plant bacterial disease and their vectors, along with the scientific name of the vector. First one is the Pierce disease or Pierce disease of grapevine, which is caused by Xylella fastidiosa. It is known to be transmitted by an insect vector called glassy winged sharpshooter. The scientific name is Homolodesca vitripennis. Peach or pecan bacterial leaf scorch, which is also known to be caused by Xylella fastidiosa. Spittlebug and leafhopper. The scientific name is Calostrophaga archaeata, and Leperonia quadrangularis. Citrus variegated chlorosis, which is caused by Xylella fastidiosa. It is also known to be transmitted by sharpshooter, Homolodesca vitripennis, and Oncometodon nigricans and Graphocephala atropunctata. Sumatra disease of clove, which is caused by Pseudomonas scizzi, is known to be transmitted by spittlebug. Scientific name is Hindola striata or Hindola fulva. Bacterial wilt of corn, as I told you, very important. Pantivia what it is known to be caused by. It is known to be transmitted by corn flea beetle, that is called scientific name is Chitonema pulicaria. Bacterial wilt of cucumber, Irvinia trachyphylla, which is known to be transmitted by striped cucumber beetle or cucumber beetle. Diabrotica victata or Diabrotica duodecipunctata. black leg of potato, which is actually Pectobacterium keratovorum, subspecies keratovorum, which causes, and seed corn maggot is known to be transmitted the disease. The scientific name is Hylima silicula or Delia platura. The scientific names are a little bit, a bit difficult, right? And fire blight of apple, Irvinia amylovora, it is known to be transmitted by insect vectors like ants, honeybees, pollens, wasps, and flies, and couple of aphids and psyllids as well. And this olive knot is caused by Pseudomonas savastoni, is known to be transmitted by olive fruit fly, Dacus folii. Bacterial rot of apples, which is known to be caused by Pseudomonas melapthora, apple maggot or fruit fly, is known to be transmitted. Dacus folii and other species, Pomonella something. A ring rot of potato, Clavibacter michiganensis, subspecies michiganensis, which is also known to be transmitted by potato Colorado beetle, or green peach aphid and potato flea beetle. The scientific names are Mesis persica, Apitrix cucuminus, and Decimillinata. Some other scientific names are given here. Citrus canker, which is caused by Xanthomonas axonopodus pathoversity. It is rare cases, not all the cases. It is rare cases known to be transmitted by leaf miner. That is Phylloncteris citrella. The citrus canker is generally known to be transmitted by this infected twigs, right? So citrus stubborn Spiroplasma spitri, leaf hopper circumferus tennis, constant Spiroplasma concali, leaf hopper Dulbus medis, citrus greening Candidatus Liberibacter asiaticus, which is known to be transmitted by psyllids, Diaphorus citriaceus, Triazo erythrae african. Wilt of potato Pseudomonas solanaceae are known to be transmitted by nematode Globodera pallida, and cucurbits yellow vine disease, it is known to be transmitted by bacteria Serratia marsans, squash bug, that is Ananas tristis. Here this Serratia marsans, it is also known to be as a human pathogen, not only plant pathogen but also known to be as human pathogen. And bacteria and their genome sequencing information now we will see. I think in a couple of maybe 100 slides before, we might have saw genome sequencing and their organisms, right? Like first plant pathogenic, what is first plant pathogenic fungi, what is first plant pathogenic bacteria, what is first plant pathogenic virus, we might have saw, right? So here this is just a genome sequencing information just related to only bacteria. So what are the list of bacteria that was sequenced from top to bottom, and other things also there, but we have given some couple of important things only. The first plant pathogenic bacterium is Xylella fastidiosa, we know very well, genome size is 2.68, that was sequenced by A.J.G. Simpson in 2000. And the next one is the bacterial wilt of tomato, Ralstonia solanacearum, that was sequenced by C.A. Boucher in 2001. And citrus canker, Xanthomonas axonopodus pathoversity, and black rot of cabbage, that is Xanthomonas campestris, pathover campestris, both are sequenced by Anna Ciar da Silva in 2002. Bacterial speck of tomato, Pseudomonas syringae, pathover tomato, AR Collimer 2003. Sugarcane ratoon standing, Livesonia xyli, subspecies xyli 2004. Rice bacterial leaf blade, Xanthomonas varaceae, pathover varaceae, BM Lee in 2005. Rice bacterial leaf streak, Xanthomonas varaceae, pathover varaceae, that was sequenced by Salzberg in 2006. So these are all a couple of sequencing information regarding plant pathogenic bacteria. And coming to the next one is virus. Just couple of theoretical information. Generally, we know very well that DNA virus contains only one kind of genetic material, either DNA or RNA, along with a protein code. So majority of the plant pathogenic bacteria contains RNA as a genetic material. Around 78% of the plant pathogenic bacteria contains RNA as a genetic material, and virus generally moves from cell to cell through plasma desmata. So plasma desmata is actually smaller than that of plant viruses, but still the plant virus is moving with the production of a protein called movement protein. So with the help of movement protein, it mediated the movement through plasma desmata, because the plasma desmata is simply like a easy connection from one cell to another cell. So this is like a small pipe like thing, so easy connection to one cell to another cell, while the cell virus moves from cell to cell through plasma desmata. Generally it moves from 8 to 10 cells per day. So majority of the plant virus are known to be transmitted by insect vectors, and a couple of viruses are known to be transmitted by mechanical transmission as well. And the enzyme generally required for the multiplication of virus is RNA polymerase. I repeat, RNA polymerase 1 is the enzyme required for the multiplication of replication of virus. If it is a viroid, RNA polymerase 2 is recommended. And let's see the difference between virus and viroid, since we have not discussed about viroid. So anyway, I will tell you. So generally a virus is nucleic acid with protein code, whereas viride is a nucleic acid divide of protein code, because no protein code will be there, only nucleic acid will be there. So the scientist first discovered virus is a biogenic, whereas the scientist first discovered viride is a diner. The protein code present, whereas protein code is absent in viride, generally known to be transmitted by insect vectors, viruses, but all the virides reported so far is known to be transmitted by mechanical transmission or vegetative transmission. And it is called as nucleoprotein. It is a combination of nucleic acid and protein. It is called as RNA particle because there is no protein. Genetic material, either DNA or RNA, but never both, only one kind of genetic material. When it comes to viride, it contains only one kind of genetic material, that is RNA. Generally here capsid is present, capsid is absent, that is protein code and nothing else. Let's come to the final point. Examples are tobacco mosaic virus, that is known to be the first reported viride. Potato spindle tuber, first reported virus, tobacco mosaic virus. Okay, so potato spindle tuber viride is known to be the first reported viride disease. So these are a couple of general information about virus and virides. Now we'll see the difference between virus, viroid and prion. So virus, we already discussed that nucleic acid protein code. It is the only protein nucleic acid divide of protein code. Prions mean it is not like that of virus and virides. It is just like a infectious protein. I would say some kind of, there are a couple of proteins, or thousands of proteins are there in our brain, or maybe in our body, you can just consider. So out of that, one protein, if it is get malfunctioned, it is known as prion. It is an infectious or malfunctioned protein. So generally the scientist first discovered virus is biogenic, viride is Diner and Reimer, but prion was discovered by Stanley Prusiner. The genome of virus is DNA or RNA, whereas viride contains RNA. It is only protein when it comes to prion. It is not a DNA or RNA. So proteins generally present in viruses, so virides it is absent. Generally in prions it is present. The size of the viruses are ranges from 20 to 2000 nanometer, whereas virides are ranges from 40 to 140 nanometer. When it, if I want to tell you in a nucleotide by 246 to 401 nucleotides, generally this prion is 5 nanometer in diameter. The examples are tobacco mosaic virus, potato spindle tuber viride is a viride example, and bovine spongiform encephalopathy, that is generally causes mad cow disease, is an example of prion diseases. And let's discuss some suffixes, or how actually in taxonomic classification of fungi, mycota, mycetes, ales, and all those things we have discussed, right? Now we will see the taxa, like what is the viral taxa. If it's the order ends with the virales, family ends with the viridae, subfamily ends with the virinae, genus ends with the virus. So these are all the different kinds of taxa along with their suffixes. And important plant viruses and their genomes, like what are the different kinds of viruses having the genome of single stranded DNA. I will tell you all Gemini viruses: banana bunch top virus, maize streak virus, beet collie top virus, bean golden mosaic virus. These are all the couple of viruses that having a single stranded DNA as a genetic material. When it comes to double stranded DNA, it's a very important: cauliflower mosaic virus, rice tumoro bacilliform virus, banana streak virus, and sugarcane bacilliform virus. These are all the different kinds of viruses that contains double stranded DNA as a genetic material. Coming to single stranded RNA, tobacco mosaic virus, tobacco necrosis virus, tomato spotted wilt virus, potato mop top virus, turnip yellow mosaic virus. So that doesn't mean the remaining viruses are not there. So far there are 2000 plus plant viruses have been discovered, where we are giving couple of important things only. Okay, double stranded RNA, wound tumor virus, lettuce big bean virus, rice ragged strand virus, rice dwarf virus. These are all the couple of viruses which contains double stranded RNA as a genetic material. The next one is the difference between cytoplasma and spiroplasma. So if we go through this general differences, we'll have a basic knowledge about the pathogenic organisms or groups. So let's start with the cytoplasma. So order, cytoplasma belonging to Achiloplasmatales, whereas spiroplasma belonging to Mycoplasmatales. So family Achiloplasmataceae, Mycoplasmataceae. The scientist who first discovered phytoplasma is Doe et al. In 1967. The first reported phytoplasmal disease is mulberry dwarf, whereas spiroplasma was first discovered by Davis et al. The first reported or discovered spiroplasmal disease is constant. And the cell shape is generally protoplasma are spherical in shape, whereas the spiroplasma is spiral or helicoid in shape, and the size ranges from 0.17 to 0.25 micrometer in diameter, whereas spiroplasma size ranges from 0.1 to 0.24 micrometer in diameter. So generally plasma membrane, it contains no cell wall or something like that. It contains lipoprotein or triploid membrane. Again same, spiroplasma is composed of a lipoprotein and a triploid membrane, a triple membrane. Generally it doesn't contain any kind of cell walls. Instead of cell wall, it contains cell membrane. The two triple layered cell membrane, 3 layers are there. Okay, so genome, generally cytoplasma and spiroplasma contains both the DNA and RNA as a genetic material. Genome size is a very important. Genome size ranges from 530 to 1350 kb, and couple of articles it is mentioned that 1650 kb, 530 or 630 to 50, 1650, but regularly I have gone through various kinds of information. It is mentioned in the standard literature that the genome size of cytoplasmal organisms are ranges from 530 to 1350 kb. When it comes to spiroplasma, it is ranges from 780 to 2200 kb. And culturing where spiroplasmal diseases cannot be cultured in a artificial medium, whereas spiroplasmal disease can be cultured in a medium. So appearance generally, since it is not cultured, there is no appearance. Since it is cultured, spiroplasma look like a fried egg or poached egg. So however we are putting omelette, it look like a fried egg or poached egg like appearance like this. So plasmid is generally present in cytoplasma, whereas the plasmid is absent in spiroplasmal disease. So both diseases are it is sensitive to penicillin. Actually this phytoplasmal disease and spiroplasmal disease both are insensitive to penicillin, and both are sensitive to tetracycline. The best examples are mulberry dwarf and coconut lethal healing. A couple of other diseases also there, but the important things like other things also there. Mulberry dwarf is known to be the first phytoplasmal disease, whereas the constant is known to be the first spiroplasmal disease to be reported. So these are all important phytoplasmal disease and the insect vectors and the scientific names of the insect vectors. So little leaf of brinjal, it is transmitted by leaf hopper. So we need to remember something here. Majority of the phytoplasmal and spiroplasmal diseases are known to be transmitted by leaf hopper. Majority of the phytoplasmal and spiroplasmal diseases are only leaf hopper transmitted. So let's see. Little leaf of brinjal, leaf hopper, Hishimonas spicatus and Amarasca bagutala, sesame phyllodes, leaf hopper, Orochius albicinctus, grassy shoot of sugarcane, leaf hopper, Saccharosydnus saccharivora, and Rafflesia maedis aphid. And aphid species is Rafflesia maedis, but Saccharosydnus saccharivora is a leaf hopper only. Coconut lethal yellowing, plant hopper, Mendes scrotus and Apalaxis scrotus. Rice yellow dwarf, leaf hopper transmitted, Nephrotic virescens. Mulberry dwarf, leaf hopper transmitted, Hishimonas cellatus. European stone fruit yellow ciliary transmitted, Cocosella pruni, Peach X, Calodonus and Scaphotypus, and pear decline, pear Silla pericola, and tomato bigbird, leaf hopper, Orochius arginatus. Sandal spike, leaf hopper, Jassus indicus. Apple proliferation, which is a vegetative propagation. See, majority of the diseases are leaf hopper transmitted only. Okay, so majority of the phytoplasmal and spiroplasmal diseases are known to be transmitted by leaf hopper. Sometimes we do expect scientific names also. So these are the couple of spiroplasmal diseases. Citrus stubborn, which is transmitted by leaf hopper, Circliferous tenacious and Scaphophytis nitudens. Constant leaf hopper transmitted, Dalbulus maedis and Spiroplasma kunkeli. Periwinkle yellosis, it is transmitted by leaf hopper, Spiroplasma phoenici. These are all a couple of spiroplasmal diseases and their insect vectors. If you observe very carefully, majority of the diseases are leaf hopper transmitted. That's why I told you majority of the phytoplasmal and spiroplasmal diseases are known to be transmitted by insect vector, that is called leaf hopper. And the nucleic acid compositions of different plant viruses. Generally, plant viruses contains 5 to 40% nucleic acid and 60 to 95% protein code. As I told you, virus is a nucleoprotein, combination of nucleic acid and protein. So the composition, the amount may varies from virus to virus, but the majority of the plant viruses contains nucleic acid 5 to 40%, protein code 60 to 95%. So that's why it is mentioned here. All viruses contains 5 to 40% of nucleic acid. This question was also repeatedly asked. 5 to 40%, and the protein content is 60 to 95%. but there is an exemption called a couple of viruses. Tobacco mosaic virus, which contains 5% nucleic acid and 95% protein code, and turnip yellow mosaic virus, which contains 34% nucleic acid and 66% of protein code, and potato virus X and potato virus Y, which contains 60% of nucleic acid and 94% of protein code. Cowpea yellow mosaic virus 3133 nucleic acid, 67 to 69% of protein code. Cucumber mosaic virus, which contain 80% of nucleic acid content and 82% of protein code, whereas tobacco ring spot virus contains 40% of nucleic acid and 60% of protein code. This is an exemption, but we must remember something. Majority of the plant viruses contains 5 to 40% of nucleic acid and 60 to 95% of protein code. One exemption is there, tobacco mosaic virus, 5% of nucleic acid and 95% of protein code. Of course other viruses also exemptions, but the TMV is very very important. Hope you guys know why the TMV is highly important for plant pathology, sorry, especially plant virology, since it is however late blight is known to be evolution for late plant pathology. This virology, evolution of virology, this tobacco mosaic virus is known to be the main cause, right? It is also known to be the first reported virus, right? So these are all the different kinds of nucleic acid contents and protein codes. And the next one is just like some kind of definitions in plant virology. What is capsid, capsomeres, virions, and sometimes we may expect like just giving a whole definition and just giving the option, something like that. So generally capsid means protein coat or general outshell is the general outshell of plant viruses, maybe either DNA or RNA, generally it is composed of. So that capsid means protein coat, the outer layer. This outer layer is generally known as capsid. Capsomere means the protein subunits, or each subunit or the each subunit of capsid is generally called as capsomere subunit. Virion means matured virus particle is known as virion. Generally the virion starts to develop after 10 hours of inoculation. And vector means we know very well any agent or any organism that act as a carrier or that act as a transmitting some other kinds of organism, especially plant viruses, since we are discussing virology. So that is known as a vector. Any agent that transmits the disease is known as a vector. So majority of the plant viral diseases are known to be transmitted by aphids and whiteflies alone. Around 50 to 60% of the plant viral diseases are known to be transmitted by aphids and whiteflies. Here it's also very very important. In exams we do expect like which of the following vector is known to transmit majority of the plant viral diseases, or which of the plant viral vector is the major agent in transmitting of plant viral disease. Aphids and whiteflies are the two major culprits. And a nucleoplast capsid, the name itself indicates a combination of nucleic acid and capsid, that is called a nucleocapsid. Since we have already discussed the nucleocapsid of plant viruses, which means nucleic acid and protein content. It contains 5 to 40% of nucleic acid and 60 to 95% of protein code. The TMV is exemptional, which contains 5% of nucleic acid and 95% of protein code. So if we observe the previous slides very carefully, in everywhere I have mentioned the special case of TMV. Why that much special the TMV is for? I will tell you why the TMV is special for plant virology, not only for plant virology but also for plant viral studies. So TMV generally belonging to Tobamo virus. So before it is Tobamo viridae classification. Now it is comes under the classification of Virga viridae in the recent classification. It is a positive single stranded RNA which composed of 5% nucleic acid and 95% of protein code. The TMV contains very very important 158 amino acids, 2130 capsomere subunits, 6400 nucleotides. I repeat once again, 158 amino acids and 2130 capsomere subunits and 6400 or 6400 nucleotides. So the scientists who first told that the TMV contains 158 nucleic acid were Anderer and Suta in 1960, and it contains 130. This is how actually the TMV look like a helix. If you just we look at the outside, it look like a banana bunch. So generally 130 helix will be there, and each helix of 16 1 by 3, which means 49 subunits. If we calculate, we will get 2130 capsomere subunits. And generally the TMV molecular weight is 17,600 daltons. That means 17.6 kd or 17.6 kd or 17.06 k. That actually determined by a scientist called Knight. The genome size is 6.4 and the refraction index is 1.54. The TMV genome can be altered with by using an substance called ADP, adenosine diphosphate. The ATP is the energy generating substance, right? ATP, adenosine triphosphate, consists of 3 phosphates, whereas adenosine diphosphate has only 2 phosphates. And molecular weight is 39 million dalton, and nucleic acid molecular weight is 2 into 10 to the power 6 dalton. So TMV is a rigid helical, and the size measures about 300 nanometers into 18 nanometer. And TIP, thermal inactivation point, is 93, and the dilution endpoint is 10 to the power of 6. And these are all the basic information. Now I will tell you why the TMV is very important. Just take a look into how many discoveries are first associated with the majority of the prime discoveries, or the principal discoveries, or core discoveries, that is actually related to tobacco mosaic virus only. We'll see the first RNA virus to be completely genome sequenced is the tobacco mosaic virus that was sequenced by Golet et al. 1982, and it is the first recognized virus in the world, and the first inclusion bodies that is produced in a plant virus was noticed in TMV also, that was noticed by Ivanovsky, and the first cross protection technique that was developed by McKinney. Nowadays we are studying cross protection technique used to manage the citrus virus, but is originally developed for tobacco mosaic virus, and later the technique was it became very famous in citrus virus. And it is also the first identified phenotypic mutant of plant virus to be reported by same McKinney, and it is the first virus to be chemically purified or crystallized by Stanley 1935, Bowden 1936, and it is the first coat protein to be completely genome sequenced. That's why they told 158 amino acids. So just after sequencing the coat protein only, they came to know that TMV contains 158 amino acids, right? That was given by Anderer and Suta in 1960. And also the first antigen reported, first SAP transmission reported, first coat protein mediated gene sequence, gene resistance. It is also developed in TMV by Paul Abel and Beachy et al. 1986, and the first virus that showed to encode cell-to-cell movement protein. And as I told you, generally plant virus move from cell to cell through a small thing is called a plasmodesmata. Generally, this plasmodesmata is smaller than that of plant virus, but still the virus is able to move through by producing a movement protein. So the movement protein that encodes the cell-to-cell movement was also first discovered by TMV. In the TMV, that was the DEAM 1987, and first proved the specific antigen that is infecting the plants by Beal, and first serological studies of plant viruses also reported in TMV. You just take a look into how many discoveries are associated with the TMV. I would say it's like a mother of plant virology. The core things are the most important things, and the revolutionary discoveries are started with. And one more important thing is, along with the cauliflower mosaic virus, TMVs also act as a model organism for plant viral studies. So that's why TMV is very important to plant virology. And coming to the viride, I think we have already discussed the viride. The prime difference between virus and viride is virus contains nucleic acid with a protein code, whereas viride contains nucleic acid but no protein code, which means absence of protein code or divide of protein code. We would say it is a viride. So central conserved region CCR is actually, so here virides are actually divided into two groups. Only two families are there, Paspiviridae and Aviceniviridae. Okay, they both are classified based on the presence of CCR. CCR means central conserved region. In Paspiviridae, CCR is present, where in Aviceniviridae it is absent. In exams, we do expect like CCR is present in Aviceniviridae or Paspiviridae. In Paspiviridae, you can remember Pasp, it starts with the P, means present. Aviceniviridae, its A starts with the A, absent. You can remember in that way. Okay, so Paspiviridae CCR is present, Aviceniviridae CCR is absent. Ribosomal activity, lack of ribosomal activity can be observed in Paspiviridae, whereas ribosomal activity is observed in Aviceniviridae. Shape of the molecule is circular in Paspiviridae, and the same kind of circular we can also observe in Aviceniviridae. Secondary structure is like rod shaped, whereas branched in Aviceniviridae. Replication mechanism is also very important. In Paspiviridae, asymmetrical rolling circle mechanism can be observed. In Avicenna viridae, symmetrical rolling circle mechanism can be observed. Self-replication mechanism is site of replication, sorry, not self-replication. Site of replication is the nucleus when it comes to Phosphoviridae, whereas chloroplast in Avicenna viridae. The replication enzyme is DNA dependent RNA polymerase 2, whereas in Avicenna viridae DNA dependent RNA polymerase only. So rolling circle mechanism, only one rolling circle mechanism in Phosphoviridae, whereas in Avicenna viridae two rolling circle mechanism can be observed. And the cleavage product is host nuclear RNAs and hammerhead ribosome in Avicenna viridae. And examples of Avicenna viridae, what are all the different kinds of examples that comes under Phosphoviridae is, the most important one is the protospindle tuber viride. We know very well it is known to be the first plant pathogenic viride. First viride to be completely genome sequenced. We can't say plant pathogenic because virides only known to infect plants. Okay, so on coconut kadang, it is the smallest plant viride. And when it comes to Avicenna viridae, avocado sunblotch and chrysanthemum chlorotic motile vital viride. If you see here, coconut kadang, it is the smallest viride, which consists of 246 nucleotides, whereas the chrysanthemum chlorotic motile viride is the largest viride, which consists of around 399 to 401 nucleotides. So this is general difference between Phosphoviridae and Avicenna viridae, and we do expect maybe CCR is present in, or maybe what is asymmetrical rolling circle mechanism can be observed in which group of virides. So such kind of questions we do expect. And here viral genomes like monopartite or bipartite. So generally we will see what are the different kinds of such systems are exist. So generally viral genome consists of a monopartite genome, that is in TMV. So bipartite, which means two, Tobra virus, Nepo virus, alpha cryptovirus, beta cryptovirus, furrovirus, furoviruses, and fungal transmitting viruses are comes under furoviruses or furroviruses, and comoviruses. These are all bipartite. Tripartite, which means three, hardy virus, cucumovirus, ilar virus, pomovirus, bigomovirus, curiosa virus, and bromovirus. These are the couple of tripartite viruses. When it comes to tetrapartite, beniviruses, alpha mosaic viruses, and armia viruses are comes under tetrapartite. 5-partite stenoviruses and 12-partite phytorioviruses. These are all the different kinds of partites like monopartite, bipartite, and all those things along with the examples. And we'll see, this is also important one, like some important plant viruses, what their sizes, I would say, what is the smallest one, what is the largest one, such kind of sizes we'll see now. So satellite tobacco necrosis virus, that is SCNVR, tobacco satellite necrosis virus, is known to be the smallest virus. The size is 17 nanometer, which is a very smallest virus. It's a very, very important one. And I observed this question repeated not in it, but also not in it, but some other entrance tests. Okay, tobacco necrosis virus TNV, the size is 26 nanometer. Brome mosaic virus 28 nanometer. Copy mosaic virus 28 nanometer. Tobacco ring spot virus 28 nanometer. Cucumber mosaic virus 30 nanometer. Vica virus 30 nanometer. Tomato bushy stunt virus 32 to 35 nanometer. Alpha mosaic virus 30 to 57 nanometer. Cauliflower mosaic virus 50 to 52 nanometer. So these are the couple of important plant viruses. And it continues. Lettuce necrotic yellow virus 60 nanometer, potato yellow dwarf virus 90 nanometer, Rio virus 65 to 70 nanometer, wound tumor virus, this is also important, 70 nanometer, sugarcane mosaic 11 to 750 nanometer, bean common mosaic virus 12 into 750, plumppox virus 12 into 760, Badna virus 300 into 20, tomato spotted wilt virus 100 nanometer, which is actually spherical in shape. Okay. So these are the list of different viruses along with their sizes. So in exams, this kind of questions was asked in the previous years, if you just take a look into. So these are also important stuff that we need to take a look into. And indicator plants, these are all very important. As I told you, every single slide is important since it is the last minute preparation. I can't say this is important or that is important. Every single thing is an important. Just like the kinds of concept we must take a look into. Based on some kind of concept. Anyway, let's see common indicator plants for viral diseases. First one is a virus. So for almost all plant pathogenic virus, generally we use a goose foot. It is an indicator plant. The scientific name of the indicator plant is Chinapodium amaranticolor. Citrus greening sweet orange. Citrus sinensis scientific name, Citrus exocotosis, trifoliate orange, Citrus trifoliate, Citrus tristiza virus, Claxi lime or Mexican lime, the scientific name is Citrus arentifolia. Potato spindle tuber virid, indicator plant is tomato, the scientific name is Solanum rhicopersinum, Solanum berthami. Red rat of tea, which is the indicator plant is fish poison bean, which is the scientific name is Tephrosia vagari. Phytoplasma periwinkle Catranthus rogus. These are all the couple of indicator plants for the identification of various pathogens of virus and as well as bacteria. And inclusion bodies, so each and every pathogen produces inclusion bodies, simply like their identification, how we realize the presence of this indicator, presence of virus by using this inclusion bodies. So we can detect based on the shape of the inclusion bodies, we can detect the virus. So we'll see what are all the different kinds of viruses and the shapes produced by the plant viruses. So tobacco mosaic virus produces X-shaped inclusion bodies like this, and whereas cauliflower mosaic produces amorphous inclusion bodies, Zika virus produces granular inclusion bodies, bullet shaped inclusion bodies are produced by rhabdovirus, whereas potivirus produces pinwheel inclusion bodies, that is a cylindrical shaped inclusion bodies. So these are all a couple of inclusion bodies that is produced by the plant viruses. Now transmission of plant virus through various vectors. So since we have already discussed, this is a very important part, like majority of the plant viral diseases are known to be transmitted by insect vectors. At least definitely there is at least one question every year. You can just take a look into the last 10 years net question paper. At least there is a question from this section. So we'll see some basic important things: tobacco mosaic virus, cauliflower mosaic virus, potato virus X, tomato mosaic virus, cucumber mosaic virus, all known to be transmitted by sap transmission. So some of the viruses which I have mentioned above is also known to be transmitted by other vectors as well. So such kind of mechanism is bimodal transmission, which means they are transmitting by using other vectors also. Bi means two modes of transmission, very simple for our understanding. The next one is the cucumber mosaic virus, papaya ring spot virus, sugarcane mosaic virus, potato leaf roll virus, pea stripe virus, bean common mosaic, beet yellosis, potato virus X, S and Y, groundnut rosety, peanut initiation mosaic virus, alfalfa mosaic virus, and cauliflower mosaic virus, all are known to be transmitted by insect vector called aphids. And the scientific names are given here, like Aphis crassivora, Mysis persicae, Aphis fabae. These are all the various kinds of aphid vectors that is transmitting the viral disease. And P aphid, P initiation mosaic virus, that is known to be transmitted by P aphid. This is about some couple of vectors. And the continuation, citrus tristiza virus is known to be transmitted by brown citrus aphid or black citrus aphid. Toxoptera citrusida, Aphis gossopi, I think we have already studied this, right? So banana bunch top virus and cutte disease of cardamom. These two viruses are very famous, that is known to be transmitted by an insect vector called black aphid, that is Pentalonia nigronervosa. We do used to study banana bunch top is transmitted by black aphid, and one more disease, that is cutter disease of cardamom, is also known to be transmitted by black aphid. And rice dwarf virus, rice tunglo bacilliform virus, wound tumor virus, maize chlorotic dwarf virus, beet top virus, rice grass stunt virus, rice ragged stunt virus, and papaya bunch top virus. These are all known to be transmitted by leaf hoppers like nephrotics virus and Sagalia constricta, Nila parvata lugans. These are all various kinds of vectors involved in. And maize-streak viruses also. These are all known to be transmitted by leaf hoppers. Since we have already discussed, the majority of the plant viral diseases are known to be transmitted by aphids and leaf hoppers, right? And in continuation, sandal spike, that is a Phytoplasma, little leaf of brinjal, sesame, phyllodes, gracious shoot of sugarcane, all are also transmitted by leaf hoppers like Nephrotics virus, Sisimonas picetis, Orosius albicindus, Saccharosydney saccharivora. On the right side, just on the exact corner, I have given the scientific names of the vectors also. And coconut lethal yellowing and coconut foliar decay is known to be transmitted by a plant hopper, Mendes crudus, and rice stripe virus, brown plant hopper. Ladal striatus, tobacco leaf curl virus, cotton leaf curl virus, and tomato leaf curl New Delhi virus, tapioca virus, mung bean yellow mosaic virus, bendy yellow bean mosaic virus, cassava mosaic virus, bean golden yellow mosaic virus, African cassava mosaic virus, all are known to be transmitted by insect vector, that is a Bemisia tavesi. Tomato spotted wilt virus, groundnut bud necrosis virus, tobacco streak virus, tobacco ring spot virus, sunflower necrosis virus are known to be transmitted by thrips tabasi. Squash mosaic virus, beetles. Cowpea mosaic virus is also known to be transmitted by beetles. Cocoa solen soot, this is important. Cocoa solen soot and banana streak virus, both are known to be transmitted by insect vector called mealy bug, Pseudococcus nijalensis. Leaf curl virus, citrus greening virus, lace bugs and psyllids. and wheat streak virus, Rhigas mosaic virus, peach mosaic virus, shallow mosaic virus. These are all like mites. And so just we are just trying to put like a couple of important vectors forward that have an exam importance. Please go through all those things, and you can also add other things as well. These are all the other things, so I'm not going through all the things like and this is important, like potato virus X, potato mopta virus, wheat mosaic virus, lettuce big bean virus. Not couple of viral diseases are also known to be transmitted by fungi and as well as the nematodes. So these are all the couple of viral diseases which are known to be transmitted by fungi, and the nematodes is also very important. This is a grape fan leaf virus. This is the first nematode transmitted plant viral disease to be reported. This is transmitted by Ziphinema index. Tobacco rattle virus, tobacco ring spot virus, P. early browning virus is all are known to be transmitted by nematodes. Now we'll see. So far we have gone through different stuff, right? First viral disease to be reported by Bijernik, reported in the year 1898, and the disease is tobacco mosaic virus. First viral disease was reported by Diner, year is 71, potato spindle tuber virus. Phytoplasma Doe et al. 1967, reported first discovered by mulberry dwarf. First spiroplasmal disease Davis et al. 1972, discovered by Corn. Sorry, constant. RLO Winston Black 1972, that is a club clover leaf. These are all the first discoveries are associated with different plant pathogenic groups, and who discovered it, and the year of discovery, and what is the first organism to be discovered, or the first disease to be reported. So such kind of things also we do expect. Coming to epidemiology, first of all, we must understand this disease triangle. Generally, there are three components in disease triangle: environment, host, and pathogen. So, let's talk about J. van der Planck. He's generally considered as father of plant disease epidemiology because he gave lots of concepts related to plant disease epidemiology, like vertical resistance, horizontal resistance, vertifolia effect. Let me tell you what is vertifolia effect. This question was asked, I think, 2 times, not 1. Loss of horizontal resistance while breeding for vertical resistance is known as vertifolia effect, which means if you are a breeder, you are working on vertical resistance, so automatically other resistance, that is horizontal resistance, will be lost. So loss of horizontal resistance while breeding for vertical resistance is known as vertifolia effect. That concept is also given by J. van der Planck. He is known as father of plant disease epidemiology. And not only that vertical and horizontal resistance, he also gave various terms like simple interest diseases. The simple interest disease, also known as the monocyclic disease or slow epiphytic disease, or compound interest disease, also known as polycyclic disease or rapid epiphytic diseases. Vertifolia effect, area under disease progress curve. This is also very important concept. AUDPC, area under disease progress curve. And he is known to publish 5 books. What we know is just only one book, that is Important Plant Disease Epidemics and Control, that was published in 1963. And mostly this question is only asking if you take a look into the previous year questions. But let's have the other books as well. We don't know in later, and maybe in future we do expect other books as well. So let's see. The first book is the Plant Disease Epidemics and Control. It's a very important and very famous book that was written by J. van der Planck in 1963. Disease Resistance in Plant 1968. Principles of Plant Infection 75. Genetics and molecular basis of plant pathogens is 78. Host pathogen interaction in plant disease, that was published in 1982. These are all the 5 books. And he also gave a couple of other concepts like protein for protein hypothesis, strong gene weak gene hypothesis, and disease triangle. All these concepts is also given by J. van der Planck. Due to great contributions towards plant disease epidemiology, he honored as father of plant disease epidemiology. These are list of sudden epidemics or epiphytotics that happens in the history of plant pathology. First one is lead blight of potato, 1845-46, Phytophthora infestans in Ireland. Grapes powdery mildew, 1845-50, in which is caused by Recife nicator. It happens in Europe. 1870, coffee rust, Hemilia vastratrix in Sri Lanka. In 1878, grapes downy mildew, Plasma paraviticola, that happens in France. In 1904, coconut lethal yellowing, it is known to be caused by Phytoplasma in Cuba. In 1904, in the same year, chestnut blight, Cryphonectria parasitica in USA. 1910, citrus canker, Xanthomonas axonopodus pathovaricitri or Xanthomonas citri pathovaricitri, Asia, Brazil, USA, and Africa. 1916 to 17, wheat stemmerus, Puccinia graminestridaceae, that is USA. 1918, Dutch elm disease, Ophiostoma ulmi or Ophiostoma nova ulmi, in US and Europe. 1921 banana bunchytop, which is caused by banana bunchytop virus in New South Wales. And 1930 banana sigatoka spot mycospirilla musicola in South Africa, South America. In 1930 Dutch elm disease Ophiostoma ulmi or Ophiostoma ulmi again in Europe. So these are all couple of list of important epidemics or epiphytotics that happens in the history. And these are other continuation. 1930 Dutch elm disease Ophiostoma ulmi or Nova ulmi. 1930 to 50 Panama wilt of banana, Fusarium oxysporum pharma species cubensis, Central America. 38 to 42 red dot of sugarcane, Physalis tucumenensis, India, Bihar and Uttar Pradesh. 1942 brown leaf spot of rice, Helminthosporium varese in India. 46 and 47 wheat stem rust Paxina, Gramineous triticale, Madhya Pradesh, India. 1950 to 60 tobacco blue mold, Pernospora tabacina, Europe. 1970 southern corn leaf blight, Bipolaris maedis, USA. 1973 apple scab, Venturi inequalis, India. 1974 to 78 coffee rust, Hemilia vastatrix, Brazil. Rubber leaf blight, Microcycles ulius, Suriname and South America. Fire blight of apple, Irvinia amylovora, North America and Europe. These are all the list of couple of important epidemics. Epidemics in India. Now we'll see the list of epidemics that happens in India. First one is sugarcane red rot, which is caused by Collotrichum falcatum. Now we're going to see only the epidemics that happens in India. Sugarcane red rot, Collotrichum falcatum, Bihar, Punjab, Uttar Pradesh, year in between 38 to 39. Bengal famine, Bipolaris varese, Bengal and Orissa region 1942-43. Around 1.4 to close to 4 million people are known to be died because of just food starvation, malaria, and various other kinds of diseases because of malnutrition. Wheat stem rust Paxina, Gramineous triticale, major wheat growing areas in India 46-47. Rice bacterial leaf blight, Xanthomonas varese, Pathovar varese, Bihar 1963. Rice tungro virus, Tamil Nadu 1985 to 87. Rice blast pericular virus again in Tamil Nadu 85 to 88. So banana bunchytop, there is a variety called Virupaksha that was completely wiped out by this banana bunchytop disease, especially in hills, the close to Kerala and as well as a major banana growing areas of Tamil Nadu, especially Kerala Tamil Nadu border like Marthandam or the borders of Tamil Nadu and Kerala. The majority of this hill banana variety Virupaksi was completely wiped out this banana bunchytop virus. Now we will see there are four important terminologies or four important concepts in epidemiology: epidemic, otherwise called as epipytotic, endemic, pandemic, and sporadic. Now we will see one by one. Epidemic. So epidemic means disease occurs in a large population of area is known as epidemic. Example is wheat stem rust, lead blight of potato, sugarcane red rot, and powder mildew. These are all the important diseases. So epidemic means it is a sudden outbreak that happens in over a large population of area and spread to all over the world. That is called epidemic. Best examples are stem rust, lead blight of potato. Endemic means that is the disease actually confined to particular area or locality. So when it comes moderately year after year. So best example is potato wart, that is only confined to Darjeeling area. Pandemic means ubiquitous, found everywhere in the world. Wherever you go, the disease will be present. The best examples is lead blight of potato, coffee rust, and wheat stem rust. Sporadic, which means irregular intervals. The disease come irregular intervals at irregular localities. Examples is woodbat of rice, ergot of rice, fusarium wilt of cotton, and angular leaf spot of cucumber. These are all a couple of examples. So these four terminologies are very important, like epidemic, endemic, pandemic, and sporadic. In exams, we would expect which of the following disease is an example of epidemic, or which of the following disease is an example of endemic. Potato wart comes under which of the following: epidemic or endemic or pandemic. Such kind of disease we do expect. Pattern of diseases, disease epidemics or epipytotics. Generally, there are three patterns are there: monocyclic, polycyclic, and polyetic diseases. So monocyclic diseases are also known as the simple interest diseases. Polycyclic diseases are also known as compound interest diseases. Polyetic disease. Now we'll see the rate of inoculum. Before rate of inoculum, we will see monocyclic disease generally produces a curve called saturation curve. It's a very important. Exams we do expect this. Polycyclic diseases are also known as a sigmoid. Polycyclic disease produce a curve called sigmoid curve, a shaped curve. Polyetic disease produce a curve called bimodal curve. or bimodal curve, and it's a very important monocyclic saturation curve, polycyclic sigmoid, polyetic bimodal curve, and rate of inoculum when it comes to monocyclic is 0.02 units per day, polycyclic 0.1 to 0.5 units per day, whereas polyetic 0.02 to 2.3 units per year. So here monocyclic examples of monocyclic disease are smarts, root rots and wilts, whereas polycyclic diseases are powdery mildews, rust, leaf spots, late blight of potato and rice blast. These are all a couple of examples of polycyclic diseases. And polyetic diseases are citrus virus, Dutch elm diseases, fungal vascular wilts, and Mycoplasma yellow. So these are all a couple of examples. Now I will tell you what is monocyclic, which means the pathogen complete the life cycle in a single cropping season. One life cycle in one cropping season is known as monocyclic. Polycyclic means the pathogen completes a many number of life cycles in a single cropping season is polytic. Sorry, polycyclic. Polyetic means the pathogen requires more than 1 year, maybe 1 to 2 years or 2 to 3 years to complete one life cycle. So these are all the examples along with the disease curves. And these are a couple of forecasting models just to forecast the disease, or you just predict the disease earlier. So epidem, in exams they give forecasting model. It is meant for which disease, or maybe they will give a causal organism. Such kind of questions we do expect. Epidem is meant for early blight of potato and tomato, which is caused by Alternaria solani and Alternaria alternata. Epime, which is caused by southern leaf blight of southern corn leaf blight of maize, Helminthosporium maydis. Epicon is also same, southern corn leaf blight, Helminthosporium maydis. Cercos, blight of celery, Cercospora species. Mycos, blight of chrysanthemum, which is caused by Mycospirilla species. Epiven, important one, apple scab, Enchuria inequalis. Epivet, viral disease of potato, contact and effet transmitted viruses. Tom cost, early blight of tomato, Alternaria solani. Plasmo, downy mildew of grapes, Plasma paraviticola. Epidemic stripe rust of wheat, Puccinia glumarum or Puccinia striformis. Epipre cereal rust and aphids, Puccinia tritici. Blight cast that was developed by Krause et al. First forecasting model, I think this is epidemic, and first some other models whether it's a blight cast, something will be there. I think that will come in the next slides. So late blight of potato and tomato, Phytophthora infestans, blight cast. Simcas, late blight of potato, neg fry. All these are from blight cast, simcas, neg fry. All these are meant for late blight of potato. And in continuation, plant plus Indo blight class that was developed by CPR, Central Potato Research Station located at Shimla. Jugal Sakas, again same CPRI only. These all are meant for, especially meant for late blight of potato, which is caused by Phytophthora infestans. And Blastilius, Epiblast, Pyricularia, Leaf Blast, Pyreneo, Blast class, these all are meant for rice blast. SAS, strawberry disease, proplant cereal disease, Vegas vegetables, cotton dust, cotton, epigram, barley, powdery mildew that is caused by Plumeria graminis, pharma species order. So these are all the just a list of a couple of important forecasting diseases that is used to forecast the plant diseases. As we already discussed about vertical resistance and horizontal resistance, right? Now I will tell you the other names. Sometimes we do, they will ask like what are the other names of this vertical resistance, or what is the major gene resistance is known, something like that. So this vertical resistance are the other names, like vertical resistance also known as major gene resistance, also known as race specific resistance, monogenic resistance, oligogenic resistance, strong resistance, qualitative resistance, or gene resistance. These are all are the other names of vertical resistance. When it comes to horizontal resistance, minor gene resistance, race non-specific resistant, polygenic resistance, multigenic resistance, field resistance, quantitative resistance, small r gene resistance, durable resistance. These are all the other names of vertical and horizontal resistance. So sometimes they may ask indirectly, like by giving other names, what it exactly represents for. So like that we do expect. Some signaling compounds are given here actually. Salicylic acid, jasmonic acid, these are all. Salicylic acid is actually a compound that is produced during an SAR pathway, systemic acquired resistance, that is actually mediated by the salicylic acid, whereas jasmonic acid and ethylene is mediated by ICAR, induced systemic resistance actually. So these are all very important one, and these are all actually the layers of defense mechanism. So SAR is activated by salicylic acid, and whereas ISR, induced systemic resistance, is activated by jasmonic acid as well as ethylene. So this S-benzolar S-methyl is a systemic fungicide generally recommended for the activation of SAR. It is like a fungicide, but it is also like a induced systemic acquired resistance. And a couple of other things also, like DCINA, dichloro isoctonic acid, and nickel nitrate, benzothiazole are generally used as an alternate of salicylic acid. And generally, we know very well if the agents are transmitting through water, if the spores or some other kinds of agents transmitting through water, it is called hydrochory. Wind means anemochory, insect means entomochory, animals means zoochory, mans means anthropochory, and examples are given here. What are all the couple of pathogens or examples of pathogens that are transmitting through water? Erwinia amylovira, Xanthomonas campus, Xanthomonas malariaceum, Malvacearum, Plasmodium probaceae, and Phytophthora infestans are known to be transmitted through water. Wind, Puccinia graminestratus, Helminthosporium sativum, alternative species, and Istulago known to transmit through wind. Insects, Erwinia trachyphylla, Erwinia amylovira, Mysis parasicae, Mysis pseudosolana, that is potrolevrol virus, Xanthomonas stevati, Ceratostomella ulmi. These are all a couple of pathogens that transmit to insect vectors. Animals, field mice, strawberry, latent ring, and Arabic mosaic virus. There are a couple of viruses that is known to transmit through animals as well. And through man, part of the infestants, Insulina negator, that is grapes powdery mildew, right? Plasma paraviticola, grapes downy mildew, Xanthomonas oxynopotus, Pathology, citri. So these are all the couple of pathogens are known to transmit humans. Now coming to quarantine, there are a couple of amendments and all those things were there that we're going to discuss here. 1912 Plant Quarantine Act. Actually, the first Plant Quarantine Act was passed in USA, United States of America, with the name of Federal Quarantine Act in the year 1912. The same Federal Quarantine Act in India, it was enforced in the year 1914 with the name of DIP, Destructive Insect and Pest Act. In India, it was passed in the year 1914. It's a very important. DIPA Act was passed in the year 1914, Destructive Insect and Pest Act in India. 1946, India set up a Directorate of Plant Production, Quarantine and Storage located at Faridabad, Haryana. Now the Faridabad, that Haryana, DPPKS is taking care of the quarantine all over the India. 1966 Seed Act was passed. 1968 Insecticide Act was passed. 1968 Seed Rules was passed. 1971 Insecticide Rules was passed. And 1972 Federal Environmental Pesticide Act. 1983 Seed Order. 1996 Food Quality Product. And 2001 Plant Production of Plant Varieties and Farmers' Right. And 2002 National Policies. These are all the couple of amendments and policies and rules and regulations that was amended in quarantine. These are all the list of copper fungicides in plant disease management. Now we'll see what all the copper fungicides are there. Bodo Mitchell. These are very important. Every year there is a question from this. Bodo Mitchell, which was the scientist involved, is PMA melodot, which was discovered in the year 1882-85. The composition is 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, and 100 liters of water. That gives Bodo Mitchell 1:1:100. This was discovered by PMA melodot. Very important. This is also called as holy water. It's been almost 140 years since the discovery. Still we are using this fungicide. So the composition is 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of lime, and 100 liters of water. So the composition they do, sometimes they may change 5:5:500 or 0.5:0.5:50. But the general composition that exactly matches to 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of lime, and 100 liters of water. Bodo paste. So the Bodo Mitchell and Bodo paste composition is more or less both is same. Instead of 100 liters of water, we are using 10 liters of water and Bodo paste to make it as a paste-like substances. Burgundy mixture was developed by Mason in 1887. So the only difference between Bodo mixture and the Burgundy mixture is in Bodo mixture we are using 1 kilogram of lime. Instead of lime, we are using sodium carbonate or replacing the sodium carbonate in a Burgundy mixture. Burgundy mixture is also known as soda borax, also known as Mason lime. Chestnut compound that was developed by Beverly 1921. The composition is 2 parts of copper sulfate and 11 parts of ammonium carbonate. This is a chestnut compound. And a Chaubai tea paste that was developed by Singh 1942. The composition is 800 grams of copper carbonate, 800 grams of red lead, 1 liter of linolenic iodide or linseed oil. This is the composition. It's a very important. Bodo mixture melodot. burgundy mixture mason, chestnut compound beverly, chauvet sing. So Bodo mixture generally recommended for the management of grapes, downy mildews and anthraconosis, and Bodo paste is generally recommended for almost all kinds of trees that is actually applied wherever the wounds can be observed, especially citrus gummosis or coconut stem bleeding, such kind of disease we do recommend Bodo paste. And burgundy mixture we do recommend coconut stem bleeding, which is caused by chalra paradox, and chestnut compound recommended for pythium damping of diseases. Chauvet paste that is recommended for pink disease of rubber, which is caused by Cordyceps salmonicola, and stem canker of apple and pear. These are all the couple of diseases that we recommend this copper fungicide. Why it is called copper fungicide? Because their major composition is composed of copper. This is a general classification of fungicide based on the chemical nature. Okay, just based on the chemical nature. So based on the in sulfur there are two. Again it is divided into two: inorganic and organic. In inorganic, powdered sulfur, wettable sulfur, and lime sulfur will come. When it comes to organic, ferbum, zerum, thirum, zinab, manab, nabum, and vapum will come. Coming to the copper fungicide, Bodo mixture, burgundy mixture, cuprous oxide, and chestnut compound. These are all the couple of fungicide comes under. In mercurial fungicide, two kinds will be there: inorganic and organic. Inorganic, mercury chloride and mercurous chloride, whereas an organic, phenyl mercuric acetate and methoxyethyl mercury acetate. These are all the two organic. Quinone fungicide, chloranil and dichloran, whereas heterocyclic nitrogen compounds, captan and folpet will comes under. Oxathin, two important fungicides are there: carboxin and oxycarboxin. These two fungicides are known as the world's first systemic fungicide, oxycarboxin and carboxin, that was developed by Van Schemling and Marshall Kulka in 1966. Benzimidazole, binomial and carbendazim, whereas others PCNB, dodine, tin compounds and antibiotics all will comes under. So this classification is actually based on just chemical nature, based on the chemical pure of chemical nature. So this is, sometimes we expect like a chloranil comes under which group, like a quinone groups or mercury group or copper group. So like that. So this is also an important one. And these are all the common abbreviations of some common formulations. A means aerosol, A means active ingredient, AF means aqueous flowable, AS means aqueous solution, B means weight, C means concentrate, again CG means concentrated granule, CM means concentrated mixture, CS means capsule suspension, D means dust, DF means dry flowable, DP means dustable powder, DS means soluble dust, ECR, E means emulsified concentrate, EW means emulsified oil in water, FL means flowable, FS means flowable concentrate for seeds and flowable series. So these are all the different kinds of abbreviations. Just like if you guys go through, that will be very easy to understand. Just like abbreviations, like sometimes they may ask only abbreviations. They don't by asking abbreviations they may ask to extend the formulation name. For example, A means active ingredient. For example, DP means dustable powder, or for example SC means suspension concentrate, or SL means soluble concentrate. So like that there is also a chance of asking. And these are a couple of trade names of the fungicide. They are very important. Here the trade names we have given for not only for fungicide but also herbicides and insects all put together in combination. Alachlor, the trade name is the Lasso, Azoxystrobin Heritage, Benudalin Calarius, Benomil Benlate, Binapacryl, Ambox, Endosan. Various trade names are there. We have given only a couple of important one: Amorocid and Bitetrenol Bacar, Blastocidin, Boscalid Emerald, and Butachlor Machete, Captafol Folfet, Captan and Captain Killer, or Kittelsen Killer. Captain is also known as Kittelsen Killer because it was first invented by Kittelsen. That's why it's known as Kittelsen Killer. Carbendazim, Bovestin, Carboxin, Vitavax, Chloranil Spergon, Copper Oxychloride, Bletox 50, Blue Copper, and Fetolan. And dichlorpygon, dichloran botran, difeconazole score, dimethylamyl milcarb, ditahen, phygon, Dodemorph BAS 238F, Dodine Cyprex, Edifenfos ethazole, terrazole, etherimol milstim, ethyl mercury chloride serasan, phenaminosulf dexone, and phenarimol rubigan, fentin chloride brestanol, fentin acetate brestan, fentin hydroxide deuter, Ferbum, Coromet and Fermet, Fluchloralin, Besalin, Folfet, Phantom, Fusatil Aluminum, Aleti, Fibuconazole, Voronit, Gliodin, Glioxidine, Hexaconazole, Contef, Iprobenfos, Ketagen, Isoprothional, Fusivan, Carathane, Dinocap, Crisoxamethyl Aragon, Mancozeb, Dithan M45, Menab, Endofil M42, Menoxome subdue, menoxome again apron and subdue both. Mercury chloride cyclosan, mercurous chloride cyclosan, mercury chloride merfusan, metaloxyl apron subdue redomil protaf, metaloxyl mancozeb redomil apron subdue, methoxyethyl mercury chloride agalol and saracenvet, phenyl mercury chloride saracendrai, myclobutanol eagle and nabum chimbum indophil A40, nitrofen toke, neuramil trimidon, oxycarboxyn plantavax, PCNV, quintozine, penconazole topaz, uh, pen, uh, pencycuran monseran, and pendimethalin stomp. Just like studying all those things, so nothing else. There's like trade names only. And these are all the other list of trade names. I'm just giving PCNB, phenyl mercury chloride, propiconazole, again propiconazole, various kinds of fungicides or insecticides or herbicides along with the trade names are given here. Just go through. And these are all the LD50 values, lethal doses 50 values of some couple of agrochemicals. ICAR, the trade name is Temic, and LD50 value is 1. So if the LD50 value is the lowest, the toxic is that much more, like the lowest and more toxic. If it is a LD50 value is more, it is less toxic, like that. So cyclohexamide 2.5 mg per kilogram of body weight, nicotine 50, edifen phos 100, endosulfan 160, caffeine 250, copper sulfate 300, carbonyl 307, therum 780, propiconazole 1517, oxycarboxyn 2000, dodine 2000, azoxystrobin more than 2000, carboxyn Vitavex 3200, chlorothalonil 3800, and tubiconazole 4000, PCNB for phosphatidylumine. These are all a couple of important fungicide or insecticide, and along with some other things, along with their LD50 values are given here. Sometimes we may expect such kind of questions as well. These are all antibacterial and antifungal antibiotic, which means antibacterial means bacterial killers, antifungal means fungal killers. Generally, we'll start with antibacterial antibiotics, streptomycin sulfate, sold as agromycin 100 or streptomycin. I'm not telling all these things. I'm just telling antibiotic, and it's recommended for which disease. The streptomycin sulfate is recommended for majority of the plant bacterial disease, especially fire blight of apple and fear, citrus canker, black arm of cotton, all those things. Terramycin, Phytoplasma, spiroplasmal disease, especially this little leaf of brinjal and sesame phyllodes. Oxytetracycline produced from Streptomyces rhizosum. P-chex, coconut lethal yellowing and pear decline constant kind of disease. Chlorotetracycline, P-chex disease, coconut lethal yellowing, again pear decline, and all those kinds of diseases. We do recommend these antibiotics. Now we will see antifungal antibiotic. Ariofunginate is produced from Streptomyces cinnamomum varitericola, Tanjore wilt of coconut, citrus gummosus, apple powdery mildew, and apple scab. We can recommend this ariofungin. Griseofulvin produced from Penicillium griseofulvin, powdery mildew of beans, downy mildew of cucumber, early blight of tomato, Botrytis cinereus. This antibiotic is recommended. Cyclohexamide, powdery mildew of beans, burnt of wheat, rhizopus, and botrytis. Blasticidin, blasts, and rice blast. Antimycin, rice blast, early blight of tomato, seedling blight of oats. We do recommend this antibiotic. And casugamycin, rice blast. So sometimes they will ask which of the following antifungal antibiotic. This was actually asked. I think that there is an antibiotic. I think this casugamycin only. Actually. Yes. Which of the following antifungal antibiotic is actually recommended for rice blast? This casugamycin. It's a very important. Isolated from a bacteria called Streptomyces casuganensis. It is actually isolated from bacteria, but it is recommended for fungal diseases. Okay. Thiolitine is actually recommended for lead blight of potato. Endomycin recommended for leaf rust of wheat and fruit rot of strawberries. Bulbiformin isolated from Bacillus subtilis recommended for red gram wilt. Nystatin produced from Streptomyces norisii. Banana and bean anthracnose, downy mildew of cucurbits we can recommend. Urocidin, Streptomyces anandi, it is isolated from, recommended for cholera, recommended Helminthosporium, couple of species. These are mode of actions for some important systemic fungicides. Mode of actions is also very important in exams. In the recent times, they are concentrating more on mode of actions as well. So let's start with alphabet. Azoxystrobin or trifloxystrobin. The mode of action is mitochondrial electron inhibitor. Benomyl, mitosis and cell division inhibitor. Benzimidazole inhibits beta-tubulin biosynthesis. Captan is otherwise called as kitten cell killer because it was first invented by Kittelson in 1952. Here is also important. I think it was asked. Uh, Captan mode of action is inactivation of enzymes through interfere with their NH2 and SH group, carbendazim, benomyl and thiobendazole, chitin and nucleic acid biosynthesis inhibitor, and mitosis and cell division inhibitors, carboxyn and oxycarboxyn. These two fungicides are known to be the world's first systemic fungicide that was developed by Van Schemling and Marshall Kulka in 1966. It interferes with electron transport, and chloramphenicol inhibits 50S ribosomal functions. Cyclohexamide inhibits protein synthesis. Edifenphos IBS, that is phospholipid biosynthesis. Erythromycin inhibits 50S ribosomal function. Phosphatyl aluminum, hinozan and ketagen that inhibits spore germination and penetration of the pathogen. Hexagonazole SBI sterol biosynthesis inhibitor. Iprodin lipid membrane synthesis inhibitor. Mancozeb denaturation of enzymes and proteins by the reaction with the SH group. Metaloxel disrupts the fungal nucleic acid RNA synthesis. Penicillin inhibits synthesis of murin, that is a cell wall component, and polymyxin G destroys cytoplasmic membrane. Streptomycin inhibits the 30S ribosomal function, and tetracycline inhibits the binding of aminoacetyl tRNA with ribosomes. These are all a couple of fungicides and antibiotics and their mode of actions. Mode of actions also very important. And these are all mycoherbicides. Where it is exactly isolated from, and it is active against which weed and the scientific name of the weed in the crop. So this is also a very important one, like they do ask the trade name or compound name and isolated from, or maybe it is targeted against which weed, or what is the scientific name of the weed, or by giving the scientific name of the weed they will ask what is the product name used to manage this weed set, like that they used to ask. The product name is Coligo. It is isolated from a fungi called Coltotrichum griosporides pharma species Ashnemane. It is used to target against a joint witch weed. The scientific name of the joint witch weed Ashnema virginica. The divine is another bioherbicide isolated from a fungi called Phytophthora palmivora. It is active against milkweed, vineweed. The scientific name is Moronia adorata. In citrus orchards, it is recommended, whereas Coligo recommended rice and sorghum. Next one is a cassette, Alternaria cassicae. It is active against the Jimson weed and the sickle pod weed. The scientific name is Dathuria stramonium. It is active against soybean and peanut, where these weeds are prevalent. Bimol, Coltotrichum gliosporides, pharma species Malway. It is active against mallow and round leaf mallow, Malva pusilla, wheat, flax and lentil. Velgo, Coltotrichum cocoides. Velvet leaf, Abutylian theophrasti, crop is soybean. Lubova number 1 and number 2, Coltotrichum gliosporides pharma species, Cuscut and Coltotrichum gliosporides, Dodder and Engelsilk hevia. Scientific names are Coltotrichum chinensis and Coltotrichum australis, Aricaceae, Sericae in soybean. Stamp out Cylindrobasidium leavi, Australian wattle, Acacia marinaceae and Acacia pycnantha, native vegetation. Biocon Chondrosterium purpureum, blackberry, Prunus serotina, forest weeds. So these are all couple of biocontrol, that is bioherbicide products, and they are actually isolated from and they are targeted against. And the questions was previously asked from this section also. And the next one is the most important one, classification of fungicides based on their chemical nature and mode of action. So I think chemical nature we already seen. Now we will see the mode of action, and it is actually used against. So starts with copper based fungicide, Bodomycin, Burgundy, chestnut compound. These are all most important copper based fungicides. Generally it disrupts the function of enzymes and proteins at the electron transport. So these copper fungicides are especially recommended for downy mildews, powdery mildews and lead beta potato. And sulfur fungicide, wettable sulfur, elemental sulfur, and lime sulfur. Generally, it interferes with the respiration and inhibits the electron transport movement. Generally, the sulfur fungicides are very active against rust, smut, powdery mildews, apple scab, and blight disease. Mercury fungicide. The fungicide is mercurous fungicide, metal toxicity generally. So this I generally act as a bactericide and also used for some kind of seed treatments or other functions as well. Dithiocarbomates, mancozeb, thiram, zinab, and nabum, which generally denaturates the protein and reacts with the SH groups. And these dithiocarbomates are generally almost all kinds of pathogen. If you see this mancozeb, thiram, zinab, or nabum, the last three are generally seed treatment fungicide, and the first one is the mancozeb. Generally, almost all pathogenic groups in all crops we generally observe, right? Heterocyclic nitrogen compound, the best example is the captan. It is a chitin biosynthesis inhibitor. Generally, Oomycetes, Alternaria, Cercospora, Rhizoctonia, Fusarium kind of pathogen, we do recommend this captan. Phosphate, best, the one that comes under is not one, many are there, just important one is phosphatyl aluminum. Actually, it inhibits chitin biosynthesis and oxidation of phosphorylation, oxidative phosphorylation. So, Pythium, Phytophthora, and Plasmopara, Oomycetes group of fungi, we do recommend this phosphatyl aluminum. Guanidines, Dodin, inhibits the respiration and membrane function. Venturia and Xanthomonas are the best examples. Coming to the systemic group, carboxymides, oxazine group, carboxyl and oxycarboxyl. Generally, it inhibits the mitochondrial electron transport inhibitor and also act as a succinase, the dehydrogenase inhibitor. Pythium, Puccinia, Rhizoctonia, and Pyricularia are the examples. And dinitrophenols, dinocap, uncoupling of oxidative phosphorylation, powdery mildews. Strobilurins, azoxystrobin, picoxystrobin, trifloxystrobin generally inhibits the ribosomal function and also blocks the transport of electrons, and it is effective against almost all kinds of pathogens. This strobilurine group of fungicides are isolated from a fungi called Strobilurus tenacellus. They are new generation fungicides. Azoxystrobin, picoxystrobin, trifloxystrobin, other kinds, whatever it ends with the strobin, it is a new generation fungicide. Of course, these are also getting old nowadays. Phenylamides, metaloxyl, and binomial generally inhibits nucleic acid biosynthesis. Pythium, Phytophthora, like Oomycetes group of fungi, we do recommend metaloxyl, especially downy mildews, we do recommend this metaloxyl. And pyramidols or pyrimidine, etherimol and dietherimol generally inhibits nucleic acid synthesis, generally recommended for powdery mildews. Benzimidazoles, binomial, carbendazim, thiophenate, mitosynthesis and cell division inhibitor, generally all fungal pathogen except Oomycetes, we do recommend this one. Thiobendazole, etherimol, lipid and membrane synthesis inhibitor for we do recommend Pythium and Phytophthora. Organophosphates, edifenphos, iprobenphos or pyrozophos. Phospholipid biosynthesis, Pyricularia and Rhizoctonia group of pathogens we do recommend. So here three important things we do observe. The fungicide, which group it belonging. The second one, mode of action is absolutely, in these days they are asking various kinds of mode of actions of fungicides, and not only for net, to have the general information. We have plenty of fungicide, but many people might get confused about this, right? Which fungicide is recommended for what. So for general group of fungicide, for powdery mildew, sulfur, or for downy mildews, we can go with metaloxyl and oxycarboxyl or carboxyl. We do recommend for rust and smut diseases and also other diseases as well. Like this, to have the general idea, this table is definitely greatly helpful. And others are anilopyrimidines, cyprodenyl, and mepaniferixim, pyrimethanol. These are all these fungicides are generally methionine biosynthesis inhibitor and lytic enzymes. Alternaria, botrytis, venturi, and verticillium. MBI, carboxamide, and cyclopropane, which are melanin biosynthesis inhibitors, and pyricularia and burkholderia. Generally, antibiotics like streptomycin, blastocytoines, casgamycin, and validamycin. Mostly protein synthesis inhibitors are ribosomal synthesis inhibitors. Xanthomonas, Irvinia, Pyricularia, Cercospora. Generally, this streptomycin is very active against almost all plant pathogenic bacteria and include Pseudomonas, Xanthomonas, Irvinia. Blasticide in cascomycin are a very active against pyrochlorine and cercospora, and validamycin are pyrochlaria, rhizoctone, and botrytis. They are very active against. These are all couple of systemic and non-systemic fungicides and their mode of actions and their active against. These are examples of the plant diseases are believed to be introduced into India from other countries. So we have lots of diseases. We used to study that it is introduced from there, it is introduced from here, like that we used to study, right? Now we are going to study only India. What are the different kinds of diseases that it introduced from various countries into India? So the first one is coffee leaf rust, which is caused by Hemilia vastatrix. It is known to be introduced in India in 1879 from Sri Lanka, probably 1870 to 79 in between. There is an exact stuff was not available. Like some literature it is mentioned that 1870 and some are mentioned that 1879. Okay, let's consider it as 1879 from Sri Lanka. In it is recorded 1876 in Sumatra, 1878 in Java, 1889 in Philippines. Slowly it spreads from all over surrounding countries, right? So late blight of potato, Phytophthora infestans. The year of record in India is 1883. It is believed to be introduced from Europe. Rust of chrysanthemum fuccinea Haryana, 1904 Japan and Europe. It is introduced. Wheat flax mud, it is a very important Eurosocrates. It is known to be introduced from Australia in 1906, where this wheat flax mud in Australia is around 1880s and 1890s. This is a very dangerous disease, and it is known to be introduced in from Australia to India in 1996. Downy mildew of grapes, Plasmopara viticola, Europe 1910. Powder mildew of crucifers, Photospora xanthi, Sri Lanka 1910. Downy mildew of cucurbits, Peronospora cubensis, Sri Lanka 1918. Downy mildew of maize, Sclerospora philippinensis, Java 1912. Paddy blast, Magnopotha graecina, Southeast Asia 1918. And the black rot of crucifers, Xanthomonas campestris, Pathover campestris, Java 1929. Foot rot of rice, Fusarium moniliforme, Southeast Asia 1930. Bacterial leaf blight of sorghum, Pseudomonas syringae, South Africa 1934. Powdery mildew of rubber, Idium heaviae, Malaya 1938. Black shank of tobacco, Phytophthora parasitica varnicotianae, Holland 1938. Fire blight of apple and pear. This is the doubt I have. In the previous couple of things we might discuss, right? Fire blight of apple was not there in India. It is here. It is mentioned. Fire blight was introduced into India from England in 1940. I don't know whether it is already existed or like even though it is existed now it is not prevalent. I don't know what it exactly means, but the data was like a little bit confusing. Anyway, let me come to know, guys. If you guys know, or else I will refer and tell you. Crown gall of stone fruits. Anyway, fire blight of apple and pear is caused by Irvinia malai. It is known to be introduced into India from England 1940. Crown gall of stone fruits, Agrobacterium tumefaciens or Rhizobacterium radiobacter 1940 England. Banana bunchita, caused by banana bunchita virus Sri Lanka 1940. Crown gall of apple and pear, Agrobacterium tumefaciens is it is believed to be introduced from England in 1943. Potato wart, Syngactrum endobaticum, Netherlands 53. Onion smut, Eurocystis sepulae, Europe 58. Blight of paddy, Philippines, Xanthomonas varese, Pathover varese, 1959. And these are all couple of pests that is actually introduced into India and other countries as well. First one is grape phylloxera vitifolia. It is believed to be introduced from USA to France in 1860, where grapes downy mildew is generally grape. This phylloxera root of it is also one of the reason for grapes powdery mildew that happens in that is believed to be introduced from USA to France in 1860s. And the Mexican boll weevil, Anthnomus grandis, Mexico and Central USA to America in 1892. Pink bollworm, Pectinophora gossipiella, India. It is believed to be introduced from India to other world countries in 1892. European corn borer, Ostenta nubilialis, Italy, North America 1916. Colorado potato beetle, Leptinotarsa decimillinata, USA. USA to France 1916, and pests are introduced to India from other countries. San John scale, Quadratus pediotus perniciosa, China, India 1879, and potato tuber moth, Thurmia operculella. I never heard of these, insect names and all those things. That's why I was a little bit taking time to pronunciate it. Anyway, Italy 1900. Woolly ephidmoth, Iriosoma Langerum, England 1909. Cotton cushion scale, Isseria purchase, Australia 1920. Golden system nematode, Globodera rostosinensis, Europe. And these are all the couple of pests that is introduced in India and other countries as well. Coming to the next one is the phanerogamic plant parasites. Phanerogamy means a flowering plant parasite. There are flowering plant parasites, and there is only 4 phanerogamic plant parasites are there: Cuscuta, Loranthus, Warabanki, and Striga. These are all the 4 important phanerogamic plant parasites. Cuscuta belonging to the family Convolvulaceae, Loranthus belonging to the family Loranthaceae, Warabanki belonging to the family Warabankaceae, Striga belonging to the family Scorfularaceae. In the recent classification, this Striga Scorfularaceae now changed to Warabankaceae. You guys can take a look into in the Wikipedia or somewhere else, but before Striga is actually comes under Scorfularaceae, now it comes under Warabankaceae. Generally, this cuscuta known to infect potato, onion, and ornamental crop. It look like yellow thread. So Loranthus generally infect mango fruits and picus. Warabanki is known to infect potato, brinjal, tomato, cabbage, cauliflower, and sunflower. And Striga known to infect tobacco, maize, sorghum, sugarcane, and millets. So here this Warabanki is more severe when it comes to tobacco also, I think so, because the tobacco known to produce benzobiron derivatives. Those are all it's like a germination kind of stuff for Warabanki. So these are all important phanerogamic plant parasites and their families and their crops infected. These are all the other names. These are all very important. Complete stem parasite Cuscuta, partial stem parasite Loranthus, complete root parasite Warabanki, partial root parasite Striga. You just remember, see every day. Just imagine if you are a student every day you are going class. You can remember, since you are not a good student. Example I'm telling. Okay, you don't know the spelling of class, so that's why you are writing as a class. CLOS. C stands for Cuscuta, L stands for Loranthus, O stands for Warabanki, S stands for Striga. You just match with the order complete stem, complete root, partial root, so that you will get the answer. Again I repeat, complete stem Cuscuta, partial stem Loranthus, complete root Warabanki, partial root Striga. So this Cuscuta is also known as golden vein because this Cuscuta look like a thread which is white color, golden color. That's what's called as golden vein. In Telugu it is called as something I actually forgot. Telugu it is called as Bangartiga, something like that. Dodder, Helvine, Hairweed, Lovine, Amarbel, Loranthus, Dendroptoi, Giant Mistletoe, and Banda, Warabanki, Broom Rape, and Tokra. Striga, it is also known as witchweed. These are all the other names. Sometimes we do expect other names as well. So for easy remembering students, as I told you, just remember the clause CLOS, Cuscuta, Loranthus, Warabanki, and Striga. Just match with the order complete stem, complete root, partial root. You will get the answer. Now when it comes to physiological disorders or nutritional disorders are also very important. At least there is a 2 questions every single year. These are all the standard ones. Every single year just go and take a look into there must be 2 questions at least. Rice Cara disease, little leaf of apple and grapes and stone fruits, citrus folio cellosis, white bud of maize or white tip of corn, and yellow streak of maize. All the diseases are known to be caused by zinc deficiency. Very important. Rice Cara, little leaf of apple, citrus folio cellosis, white bud of maize, and yellow streak of maize. All are known to be caused by zinc deficiency. This Rice Cara disease was first reported by Y.L. Nene and Padmanabham. Initially they thought that it was caused by some kind of disease. Later they identified this is a zinc deficiency. Just application of zinc sulfate can rectify the disorder. The next one is a pahala blight of sugarcane, citrus mottling, grey speck of oats, marsh spot of pea and sugarcane, speckled yellow of sugar beet. These all are known to caused by manganese deficiency. Pahala blight of sugarcane. It's a very important one. Marsh spot of pea also. And next one is a whiptail of cauliflower. These are golden. These are all platinum questions. If you see in UG level or wherever the general competition exams will be there, this rice carea disease or whiptail of cauliflower, such kind of questions are very most decorated questions. Whiptail of cauliflower and scald of French bean both are known to be caused by molybdenum deficiency. The next one is blossom end rot of tomato and fruits, not only tomato but vegetables. Blossom end rot of vegetables and fruits, spongy tissue of mango, black heart of celery, all are known to be caused by calcium deficiency. Top sickness of tobacco, dry and hot rot of sugar beet, internal cork and necrosis of mango, cauliflower and apple, cracking of young pomegranate, cracking of stem in celeries, white tip of rice, browning and yellow hollow stem of cauliflower, hen and chicken disorder of grapes, all are known to be caused by boron deficiency. The top sickness of tobacco, hen and chicken disorder of grapes. These are a couple of important ones that we must take a look into, caused by boron deficiency. And red leaf of cotton, sandran of tobacco, spongy tissue of mango, that is known to be caused by manganese deficiency. Before we saw magnesium, now it is a manganese. And black heart of potato, it's a very important one. It's a trademark question. Red heart of lettuce and cabbage, and brown heart of apple, that is known to be caused by oxygen deficiency. Coconut pencil point, that is a micronutrient deficiency. And bluish gray cluster in crop plants is known to be phosphorus deficiency. And rimfire or leaf scorching in plants, alfalfa little spot, cotton rust is also important. It is known to be caused by potassium deficiency. Straight head in rice, cork of apple, water core of apple, black end of pears, these are all due to imbalanced water supply. And black tip of mango, it's also very important one. This is due to boron deficiency, and also sometimes high sulfur dioxide concentration or toxicity of high toxic of sulfur dioxide concentration, also the reason. Tomato cat phase, low temperature. Frost injury of apple, again low temperature. Tomato sun scald, canker of linseed, blossom and rot of citrus, it is known to be caused by high temperature and exposing of sunlight. Red leaf of cotton and chlorosis in rice, buttoning of cauliflower, there is a nitrogen deficiency. Aloe heart of potato, excess amount of nitrogen. Citrus exanthema, this is important, dieback of citrus or citrus exanthema. Reclamation of votes and pear exanthema, these are all are known to be caused by copper deficiency. Dwarfing and yellowing of a cotton, sulfur deficiency. Internal brown spot of potato, moisture deficiency. Not a moisture deficiency because of excess moisture, I think so. Green netting of citrus, iron deficiency. Those are all are like nutrient deficiencies or disorders. Now we're going to see due to air pollution also, like due to air toxicants also, certain kind of disease was observed in plants. Now we'll see due to sulfur dioxide concentration, we do observe a disease in alfalfa, violet, conifers, pea, cotton and beans. And here air pollutant, and that is toxic at what concentration, that is important. Okay, the sulfur dioxide is toxic at 0.3 to 0.5 ppm. M means parts per million, ppb means parts per billion. Okay, so ppm. Nitrous oxide, so beans and tomatoes, it is concentrated toxic at 2 to 3 ppm. And hydrogen fluoride, it is highly toxic at even at lowest concentration, it is highly toxic, 0.1 to 0.2 ppb, parts per billion. Corn, peanut and tulips. And chlorine and hydrogen chloride, many kinds of crops at it is concentrated at 0.1 ppm. Ozone, it is also highly toxic, toxic at 0.1 to 0.3 ppm. It is toxic to tobacco, beans, cereals, alpha, petuna, citrus and corn, some other kinds of crops as well. And pan, this question was a little bit repeated last group. So far we have size inorganic, now we're going to see organic. Pan peroxyethyl nitrate, due to pan concentration there is a disease can occur in tomato called silver leaf of tomato, peroxyethyl nitrate. So in spinach, petuna, tomato, lettuce and dahlia also it is toxic, but due to pan concentration or toxicity there is a disease called peroxyethyl nitrate at the rate of 0.01 to 0.2 ppm, it is highly toxic. The last one is ethylene, 0.05 ppm. Generally it can cause toxic effects on many kinds of crops. So this is about various kinds of air pollutants that causes diseases. Now biocontrols. These are all the list of various biocontrols products and their generally used in plant disease management. So here we have given biocontrol agents and their trade names, along with it is active against which pathogen or targeted diseases. These all are important only. Ampeliomyces quisqualis, generally selling it with the trade name of AQ10. It is very active against powdery mildew of grapes. So for grapes powdery mildew, the biocontrol agent recommended is Ampeliomyces quisqualis. Now we are concentrating only biocontrol section. Okay. Candida oleophila, trade name is Aspire, Botrytis, Penicillium and Venturiana equalis. Trichoderma harziana, Binabiti, wilt, wheat tail diseases, root rot disease. We do recommend this. Fusarium oxysporum, Biofox C. See, generally Fusarium oxysporum is a pathogen, right? So from that also we do they're extracting some kind of biocontrol agents. Fusarium oxysporum and Fusarium moniliforme. Pseudomonas syringae, ESC10 and ESC11, Biosave 100, Biosave 110. They're extracting from this. So they are exploiting against Botrytis cinerea, Penicillium species, Mucor pyriformis, Geotrichum candidum, both are same. So and Pseudomonas chlororhaphids, the trade name is Pseudomen, leaf stripe, Fusarium species, spot blotch, leaf spot. These are all the various kinds of diseases we do recommend. Pseudomonas fluorescens, conquer, Pseudomonas thalassy, Coniotherium minitans. They are selling with the trade name of Coni and contacts, especially recommended for Sclerotinia, Sclerotiarum, and Sclerotinia minor. And Burkholderia sepiae, Denny, Rhizoctonia pythium fusarium, Bacillus subtilis, champion, companion Rhizoctonia pythium fusarium and Phytophthora disease it is recommended. Bacillus subtilis, epic, Rhizoctonia solani, fusarium species, alternaria species and aspergillus species, they are recommended against. So these are all a couple of list of important biocontrol agents and their targeted against and the trade names. And it continues. Bacillus subtilis, Kodiak, HV, generally hereafter I'm not going to tell about this trade names. Let's discuss the biocontrol agent and it is active against. So Bacillus subtilis, Rhizoctonia solani, Fusarium species, alternaria and aspergillus. Again the same Bacillus is also active against powdery mildews, downy mildews, cercosporally spots, early blights, late blights, brown rods, fire blights. And Agrobacterium K84, it is a very important. Agrobacterium K84, it is selling with the trade name of Galtrol. It is active against Agrobacterium tumefaciens, generally cause crown gall disease, right? And again Agrobacterium 1026, Nogal and Digal, again it's same Agrobacterium tumefaciens. And Agrobacterium radiobacter Norvac, it is also active against Agrobacterium tumefaciens only. Now Streptomyces griseoviridis, active against Fusarium, Alternaria brassicola, Phomopsis, Botrytis, Pythium, Phytophthora. Faciliomyces liliaceanum, generally especially recommended for nematodes. And Pythium oleandrum, Pythium ultimum, it is recommended against. Glycladium catenulatum, Pythium rhizoctonia solani, Botrytis, Didymellite is recommended. Glycladium virens, damping of root rot pathogens, especially rhizoctonia and Pythium pathogens. Thalaromyces flavus, Verticillium dahlia, Verticillium alboetrum and Rhizoctonia solani. Trichoderma harzianum, Rhizoctonia solani, Pythium species, Fusarium species, and Sclerotian ralph. These are all the different kinds of pathogen it is recommended against. So these are all the list of biocontrol agents. Same that is continuous with slight variations. This cucurbits of powdery mildew, Galvanomyces kikaracearum or Podospora xanthi. The biocontrol is the Teleopsis. Grapes powdery mildew, Ancilla nicata, Recipenicata, Ampelomyces quisqualis 10, Rust Puccinia, Darluca phylum. It is important one. What is the specific rust biocontrol agent? Darluca phylum or Verticillium lacanis also can be recommended. Grapes downy mildew, Plasma parviticola, Fusarium proliferatum. Cucurbits downy mildew, Pseudoperonospora cubensis, gastromitis, fire blight of apple, Irvinia amylovora. There is a biocontrol agent called Irvinia herbicola can be recommended. Apple scab, Venturia anaqualis, Candida oleophila, Athilia bombicina, and Chitomium globosum. Crown gall of Rosaceae, Agrobacterium tumefaciens, Agrobacterium radiobacter, and K84 and 1026 strains are there. Ergot, Claviceps macrocephala, Fusarium sambusarum can be recommended. For Womystis group of fungicide, Phytophthora, Pythium, and downy mildews, we do recommend this Chitomium globosum. White rat fungus, Sclerotinia sclerotiarum, the recommended biocontrol agent is Coniotherium minitans. I think we have already discussed, just a small elaboration here. On the commercial biocontrol agents, now we are talking about commercial biocontrol agents along with the target organisms and the trade names. Agrobacterium radiobacter K84, strain K84, trade name is Galtrol, and it is active against Agrobacterium tumefaciens. Now we are talking about only commercial products that are available outside. And Agrobacterium radiobacter K1026, Nogal, Agrobacterium tumefaciens. Bacillus subtilis strain GB34, Rhizoctone and Fusarium. Bacillus subtilis GB03, Kodiak campaign, Rhizoctone and Aspergillus, it is active against. Pseudomonas aureofaciens TX1, Biojet spotless, Pythium and Rhizoctone solani. Pseudomonas fluorescens A506, Frostban, Fireblight and Bunch rot. Streptomyces griseoviridis, Mycostop, soil-borne pathogens. Trichoderma harrygiana against soil-borne pathogen. Trichoderma harrygiana against soil-borne pathogens, as well as Botrytis cinerea. Ampelomyces quisqualis AQ10, especially powdery mildew of grapes. Now, couple of mushrooms, edible mushrooms, and their scientific names. In exams, like they do ask, like they're giving a name of the mushrooms, and they may ask scientific name of the mushrooms, or by giving the scientific names, they may ask us to identify, right? So it may happen. So Padistra mushroom or Chinese mushroom, Valvariella Valvaceae, Padistra mushroom or Chinese mushroom, hot weather mushroom, Agaricus bitrocus. White button mushroom, also known as European mushroom or temperate mushroom, Agaricus bisporus. Milky mushroom, also known as summer mushroom, Calocybe indica. Shiitake mushroom, Lentinus edodus. Oyster mushroom, there are various species are there. Pleurotus ozarkazu, Pleurotus astraeus, Pleurotus centripalliatus, Sclerotus jamar, Sclerotus florida. These are all the various oyster mushroom, generally Pleurotus species, or we would say. These are a couple of edible mushrooms, and continues. These are a couple of other jaw ear or black ear mushroom, Auricularia auriculata, Auricularia polytricha, winter mushroom, Flammulina velutypes, Rishi or royal Ganoderma lucidum, brown cap or giant mushroom, Stropharia rugoanulata, Caesar's mushroom, Amanita caesaria. So regarding Caesar's mushroom, we might have studied in 54 AD. Roman Empire, Claudius Caesar was murdered by his wife Agrippa by mixing the poison with Amanita pallidus. So why it is called Caesar's mushroom? Because this mushroom is the favorite mushroom of Amanita Caesar. So that's why it is known as Amanita caesaria. Then you can also ask some other question. Amanita is generally poisonous. Why it is, I mean, like how it is edible here? Not all the species are poisonous. Couple of species are edible also, like Amanita citrina, Amanita caesaria, like that. Couple of species are edible as well. And honey mushroom or bootless fungus, Armillariella milia, pore fungus or penny-bun fungus or sep fungus, Boletus edulis. So these are all a couple of important mushrooms and their scientific names. Okay, this continues as well. Cantarella mushrooms, Cantarellus sabarius, saffron milk mushroom or delicious Lactris deliciosa, delicious. Parasol mushrooms, Macrolapiota procera, pine mushroom, Trichloma calignatum, fairy ring mushrooms, Marasmus orideatus, bamboo sprouts, or some other kinds of like wild lady, flower of the king, king of the dried foods. So Dictyofera duplicata, termite mushroom, Termitomyces Clypeatus, and again Termitomyces eurizus, and false cumbi or gastrotomyces mushroom, Podaxis pistillaris, morels or guchis, Morchella esculata, Morchella deliciosa, truffles, tuber melanosporum. So these are all a couple of other mushrooms. The general temperature requirement of mushrooms, what temperature the general mushrooms are required. So there are a couple of 4 or 5 important mushrooms are there, I will discuss. Button mushroom, scientific name of button mushroom is Agaricus bisporus. The temperature requirement is 15 to 25 and the relative humidity is 85-95. And Padistra mushroom, Valvariella valvaceae, the temperature requirement is 30-35 and humidity is 85-90. Milky mushroom, Calocybe indica, 25-35 is the required temperature and the humidity is 75-85. Oyster mushroom, Pleurotus species, temperature is 20-30 and the relative humidity is 80-85. So these are all some temperature requirement of the mushrooms. And coming to this, generally majority of the mushrooms are comes under Basidiomycota group of fungi, except a couple of mushrooms. So we can also say costly mushrooms like Marchella, tubers, or truffles. So these are all actually comes under Ascomycota group of fungi. But majority of the mushrooms they comes under Basidiomycota only. Truffles are really valuable mushrooms because it took at least 10 to 15 years to get the first harvest. In the couple of areas, it is recorded that a couple of mushrooms like this truffles, most valuable mushrooms, 1 kilo costs around near 10 to 15 lakhs in some parts of the world. And let's see some couple of other names of the genuses or fungal mushrooms. Physis are generally called as cup fungi, whereas the tubers are called as truffles. In India, it is called gucci. Marchella generally called as morels or bell morels or true morels. Helvella generally called as saddle fungi. Since we have discussed the tubers, that is the truffles, these are all very important mushrooms, are very costly mushrooms. It is believed that if we have these mushrooms once, at least once in life, we won't get diseased. Like that, they used to believe. Uh, Anyway, let's see truffles. Again, these truffles are a couple of types. Tuber melanosporum, generally called as black or Perigold truffle or French truffle. Tuber magnatum, white Piedmont truffle. Tuber aestivum, summer truffle. Tuber uncinatum, desert truffle. Tuber indicum and Himalayensis are generally known as Chinese truffle. So there are different kinds of truffles and their common names. In exams, we do in exams there is a chance of asking like this, like summer truffle, Tuber aestivum, magnatum, uncinatum, or indicum, like that. So summer truffle is Tuber aestivum, or magnatum is a white Piedmont truffle, indicum is Chinese truffle. Like that, there is a chance of asking. And hallucinogenic mushrooms, there are lots of hallucinogenic mushrooms are there, but in the ancient literature it is repeatedly mentioned that this psilocybe mexicana and psilocybe semilancia and amanita muscaria is are having the best hallucinogenic properties. At the time they don't have alcohol or liquor companies the selling liquor, right? Even some Mexican people or Mexican Indo-Mexican people are still believing that after having these mushrooms or hallucinogenic mushrooms only they will get the message from the god. They still believe. And the psilocybe mexicana and semilancia is known to produce two chemical compounds, are psilocybin and psilocin. Probably these two compounds are the responsible for their hallucinogenic properties. And generally flesh of god, psilocybe is considered as the flesh of gods. Dead man's finger, Xylaria polymorpha. Candle snuff fungi, Xylaria hypoxylon. This also causes a disease called root rot of apple. And a dead mole's finger, Xylaria longipes. Invader of mushroom beds, Xylaria vaporaria. So these are all a couple of mushrooms and their representations or common names or other what it actually called. And some of common names of the mushrooms: self or coral fungi, clavaria or ramaria. See here, behind the common names there is a lots of reason, because it will produces selves or corals, so that's why it is named as uh, cell fungi or coral fungi. Why it's called a puffball fungi? Because it look like spores are produced in a puffing mechanism. It look like a ball and spores are produced in a puffing mechanism. That's why it's called a puffball. Earth star fungi. It exactly look like a star. That's why it's called a earth star. Like that, every fungi, the naming of every mushroom behind there is a reason. Okay. Anyway, I'm not going to explain all the reasons since we have very less time. Self or coral fungi, clavaria or ramaria. Puffball fungi, lycoperdon and clavaria. Earth star fungi, geostrum. Earth ball fungus, Scleroderma. Stinkhorn fungus, Pallas. Bud nest fungus, Nidula and Cyathus. Fly agaricus, Amanita muscaria. Caesar's mushroom, Amanita caesaria. Fire earring mushroom, Agaricus. Dye fungus, Pisolithus. Pretty-mouths, Calostroma. Hedgehog mushroom, Hydna. And continues. Pore mushroom or self fungi or bracket fungi, polyporous or ganoderma. Turkey tail mushroom, Trimatus. Bioluminescent mushroom, Armillaria melia. Orange peel mushroom, Peziza aurantia, Peziza aurantia and Alluria aurantia. Peat mold fungus, Peziza astrocoderma. These are all a couple of mushrooms and their common names. And important poisonous mushroom, here I mentioned so far we have edible mushrooms. Now we see important poisonous mushrooms. Fly agaricus, Amanita muscaria is known as fly agaricus. panther cap Amanita pantheria, dead cap Amanita pallidus, fool's mushroom Amanita verna, destroying angel Amanita virosa, psilocybe species hallucinogenic effects, and if you have a high concentration, these are also known to be toxic. Woolly milk mushroom Lactarus tormisosus, lilac bonnet Mycenia pura, Liberty cap Psilocybis similentia, Jack O'Lantern, Omphalatus illudens and Omphalatus olearius, and ghost fungus Omphalatus nidiformis, honey fungus Armillariella melia, because uh, this honey fungus is also known as bioluminescent fungus because of the production of light at the night time. Fenugreek milk cap Lactarius helvus, weeping bolete, Silis granulatus. These are all a couple of poisonous mushrooms. Now the important nematodes and their scientific names. There are a couple of important nematodes are there and their scientific names are given here. Wheat gall nematode or wheat ear corkel nematode or wheat seed gall nematode. That scientific name is Anguna tritici. This is also important. They will ask scientific names as well. Cyst nematode. The two species are there. Heterodera and Globodera. Both are cyst forming nematodes and generally causes cereal cyst nematode causes disease in cereals. South root knot nematode. Sorry, southern root knot nematode or cotton root knot nematode or root knot nematode. Melodogyn incognita. Root knot nematode means Melodogyn incognita. Dagger nematode, Ziphinema. Stubby root nematode, Trichodorus. Potato cyst nematode or potato root knot nematode, Globodera rostosinensis. Golden cyst nematode or golden eelworm or yellow potato cyst nematode, Globodera rostosinensis. Stem nematode, Ditylingus displaci. Reniform nematode or kidney nematode, Rochulenchus reniformis. Banana burrowing nematode, Rhadophilus similis. If you see in fusarium wilt concentration or fusarium wilt severity is more if it is associated with a nematode called banana burrowing nematode. It is also very important one. Other continuation, rice white tip nematode or leaf bud nematode, Phalangoides basi, root lesion nematode, Pralatainchulus penetrans, pin nematode, Pratalenchulus hamatus, citrus nematode, Tylenchulus semi-penetrans, stem and bulb nematode, Ditylenchulus dispaci, rice stem nematode, Ditylenchulus angustus, mushroom nematode, Ditylenchus myceliophagous or Aphelingoides composticola, red ring nematode of coconut, Redinocephalus cocophilus, false root knot nematode Nocobus, spiral nematode Helicotylenchulus. And these are all like kind of parasitism. So how this plant parasitic nematodes are parasitic on. So Angunas generally seed gall nematode. This is generally a migratory endoparasite. Bursa phalanchulus generally a red ring or wilt nematode. It is a migratory ectoparasite. Criconimella ring nematode, it is a sedentary ectoparasite. So Ditylingus stem and bulb nematode, it is a migratory endoparasite. Like they will ask a type of parasitism also. Okay, Globodera golden cyst nematode is sedentary endoparasite. Helicotylenchulus spiral nematode migratory ectoendoparasite. Heterodera cyst nematode sedentary endoparasite. Hirschmaniella rice root knot nematode, rice root nematode migratory endoparasite. So Haplolimus lance nematode migratory endoparasite. Melodogeny root nematode sedentary endoparasite, Pratylenchulus lesion nematode migratory ecto and endoparasite both. So Rhadophilus burrowing nematode migratory endoparasite, Rotalenchulus reniformis sedentary semi endoparasite, Scutellonema yam nematode, it is a migratory ecto and endoparasite both. Tylenchulus citrus nematode sedentary both sedentary semi endoparasites. So they will ask a type of parasite is ectoparasite, endoparasite, migratory endoparasite, sedentary endoparasite. Such kind of things they do ask because it was asked earlier. That's why I'm telling. These are all couple of list of plant viral diseases that are known to be transmitted by nematodes. Generally, majority of the plant viral diseases are known to be transmitted by insect vectors, but couple of nematodes also reported that act as a major transmitting agent of plant viral disease. Now we'll say I've given this couple of list of viral diseases and the nematodes involved in the transmission. Arabis mosaic virus. The nematodes involved is Langidorus Paralangidorus, Ziphinema and Ziphinema. These are all couple of species that known to be associated with Bromosaic, Langidorus, Ziphinema, and Carnation, Langidorus and Ziphinema diversicatum. And copy mosaic virus, Ziphinema, grape fan leaf virus. It is a very important one. It is known to be transmitted by Ziphinema grape fan leaf virus, Ziphinema index, and American other species as well. Grape vein binding virus, Ziphinema index. Mulberry ring spot, Langidorus martini. Tobacco ring spot virus, Ziphinema americanum and Ziphinema coxi. And tomato ring spot virus, Ziphinema americanum and Ziphinema bravicoli. Tobacco rattle virus, Trichodorus and Paratrichodorus species are known to be involved in. Now, nematode and fungal disease complexes. So far, we have seen the involvement of nematode in transmission of plant viruses. Now, we will see the involvement of or the association of nematodes with fungal, bacterial, and viral diseases. Since in the previous slides I have already mentioned that if the nematodes, couple of nematodes like banana burrowing nematode, if it is associated with the fusarium wilt of banana, the severity goes beyond, like more than the usual. So here there are a couple of complex diseases or associated diseases where their severity will be increased if it associated with the nematode. Now we will see what are the different kinds of associations. First one is the Melanogaini incognita. When it is associated with the fusarium wilt, that is a fusarium oxysporum pharma species, vas infectum in cotton, the severity is increases. And Rhadophilus similis in fusarium wilt or Panama wilt of banana, that is caused by fusarium oxysporum pharma species cubensis. And damping of disease of in green bean, that is Melanogaini incognita. And Phytophthora foot rot in pepper associated with Melanogaini incognita. And vascular wilt of tomato associated with Melanogaini javanica. Pomopsis blight of eggplant is associated with Melanogaini incognita. And fusarium wilt of watermelon is also associated with the Melanogaini incognita. And verticillium wilt of potato associated with the Paratylenchulus penetrans. And black shank of tobacco, it is associated with Melanogaini incognita in tobacco. So if these nematodes are associated with these fungi, their severity is increased. And now we will see the nematode bacterial association. So far what we have seen nematode fungal association. Now we will see the nematode bacterial association. Tundu or ear corcle, that is one of the phenomenal example. Tundu or ear corcle, that is actually caused by Clavibacter tritis, is associated with a nematode called Angna tritis in a wheat crop. Bacterial wilt of eggplant, Melanogaini incognita, Ralstonea solanacearum. And bacterial wilt of tobacco, Ralstonea solenis associated with a nematode called Melanogaini incognita. Bacterial wilt of alfalfa, that is caused by Clavibacter michiganensis subspecies insidiosum associated with a nematode called Melanogaini incognita. And Bacterial scab pseudomonas marginata, gladiolus associated with the Melanogaini incognita. Bacterial wilt parabarcularia caryophyll in carnation, that is associated with the Melanogaini and Helicotylenchulus. And cucumber wilt in strawberry, that is associated with the nematode called Aphelingoides rhizosombi and Aphelingoides frageri. And Kpha liked bacteriosis in garlic, which is associated with a nematode called Dietlingus dispersing. Wilt in potato associated with Globodera pallida. These are all nematode bacterial association or complexes. And just now we discussed in the couple of two slides before different kinds of nematodes that is act as a vectors in transmission of plant viral diseases. And generally in fungi and plant bacteria also they are actually associated and also helpful in the transmission of plant fungal and bacterial disease as well. And couple of viral diseases that are transmitted through nematodes are Ziphinema indica, grape fan leaf virus, tomato ring rot virus, tobacco ring rot virus, ring spot actually, tomato ring spot and tobacco ring spot, cherry raspberry virus that is known to transmit to insect vector called Ziphinema americana, tomato ring spot virus Ziphinema brevicoli, and Arabis mosaic virus Ziphinema diversicaudatum, Arabis mosaic virus, and along with the grape fan leaf Arabic mosaic virus, it is known to be transmitted by Ziphinema paralongatum. And raspberry ring spot virus, Langidorus elongatum, pepper ring spot virus and tobacco rattle virus, it is known to be transmitted by Paratrichodorus minor. These are the couple of nematode vectors that are helpful in transmission of plant viral disease or associated with the plant viral disease transmission. And these are all a couple of symptoms that are root infecting ectoparasitic nematodes and above ground nematodes generally. So if you see dagger nematode, these are all root infecting ectoparasitic nematode, needle nematode and pin nematode, ring nematode, spiral nematode, sting nematode, stubby root nematode, and stunt nematode. These are all the nematodes that actually infect root infecting ectoparasitic nematode. And above ground parts infecting nematodes like pinwood nematode, red ring nematode, seed gall nematode, stem and bulb nematode. These are all the couple of examples that were actually infect the above ground parts. And hot water treatments that are especially recommended for sugarcane seed-borne disease and other diseases as well. This is generally physical methods of plant disease management. Root rot, that is caused by Phaseolospora tucumenensis. Generally, the seed treatment or hot water treatment is 54 degrees Celsius for 8 hours, and Vipsmut Sporosorium cetaminium. This is 54 to 60 degrees Celsius for 10 minutes. Ratoon stunting, Livesonia xylem subspecies xylem, 50 hours for 3 hours. Wilt Fusarium moniliforme, 50 degrees centigrade for 2 hours. Downy mildew Pernoschlerospora sakari, 54 degrees centigrade for 1 hour. Leaf scald Xanthomonas albinians, soaking of cold water for 1 day, and then again treating with 50 degrees centigrade for 2 to 3 hours. Grasses shoot and white leaf both are caused by phytoplasma, 54 degrees centigrade for 2 hours for grasses shoot, whereas white leaf 54 degrees centigrade for 40 minutes. Sugarcane mosaic virus 52 to 58 degrees Celsius for 20 minutes for 3 successive days. And spike, which is caused by spike virus, 52 degrees centigrade for 1 hour. These are all a couple of hot water treatment for the management of sugarcane disease and other diseases as well. And coming to physiological plant pathology, phytoloxin, these are all metabolites which are produced by the plants after the infection of plant pathogens. So I'm not discussing in detail, I'm just discussing different kinds of phytoloxins that is produced by the plants. For example, rice produces a phytoloxin called sucronetin and mobilactin A and B for the pathogen called rice blast, that is a Magnoporthe griseae. For potato, it produces a phytoloxin called rishitin, stilbene, pytuberine, and lubimin. These are all the different kinds of potato pathogens that is especially produced, especially for pathogen called lead beta potato. Sweet potato phytoloxin is hypomearamon, tomato rishitin, Arabius thaliana camlexin, French bean and bean fasciolin and cavitol, and tobacco glutathione and capsidol, and pea pisatine, broad bean viverine and viverinic acid, pepper capsidol, cotton and rose gasipol, soybean glyceolin, red clover trifluoride and visitotol and macin, garlic allin. These are all the different kinds of phytoloxin that is produced by the plants. It's a very important one. The world's first phytoloxin to be discovered is P. That is pisatine. Pisatine is the world's first phytoloxin that was discovered by Muller and Borger in 1940 in the plant P. If they are these two scientists, Muller and Borger, while working with the Phytophthora system, so they discovered the world's first phytoloxin, that is a pisatine in pea plant. And other in continuation, broad bean, viveronic acid, carrot, isocomarin, alpha medicapin, sativin, chickpea or pea cysteine, macin. Panicin roots, xanthotoxin, papaya, denilone, oats, evilamin 1, 2, 3, orchids, arkinol and hircinol, almond, blueberries and grapes, pterostilbene, musa species, phenylphenolans and phenylphenolans and anigoraphones, something, whatever it is. These are all the different kinds of phytoloxin that is produced by the croplands. Coming to the PR proteins, these are all proteins that is produced by the plants where the infection of plants. There are 17 kinds of PR proteins have been reported so far. The PR protein 1, tobacco PR protein 1, the properties, PR1 is antifungal properties, PR2 is beta 1,3 glucanase. They will ask PR1 is meant for the properties of PR5. PR2 and PR5 was repeatedly asked questions. PR1 is antifungal, 2 is beta 1,3 glucanase, 3 chitinase type 1, 2, 4, 5, 6 and 7. PR4 chitinase 1,4, PR5 thamatin like protein. This is a wound hormone, you can also say wound. Okay, and 6 protein inhibitor, 7 endoproteases, 8 chitin type 3, 9 peroxidases, 10 ribonucleases like 11 chitinase 1, 12 defenses, thionin 13, and lipid transfer protein 14, 15 is oxalate oxidase, 16 oxalate oxidase like 17 unknown, it's not discovered so far. But when it comes to PR proteins, 1 and 5 was repeatedly asked. Sir, not 1 and 5, 2 and 5 was repeatedly asked. But we do expect other questions as well. And these are all the host specific and non-host specific toxins that are produced by. So that the name of the toxin is HV and produced by the pathogen. Here Victorian toxin. So these are all our host specific toxins. Victorian toxin, which is produced by Helminthosporium victoriae. HSC toxin, Helminthosporium carbonum or Cotylobolus carbonum. HS toxin, Helminthosporium sakari. HMT toxin, Helminthosporium medes resti. ACL toxin, Alternaria citri. AL toxin, Alternaria lycopersicii. AK toxin, Alternaria cucuchiana, AM toxin, Alternaria mali, PC toxin, Periconia circinata, PM toxin, Philostigma matis. These are all a couple of host specific toxins that is produced by the different plant pathogens. Now these are all host to non-specific toxins. Alternaric acid or ginol that is produced by Alternaria. Rhizopus produces fumaric acid, whereas Aspergillus sclerotinia and Sclerotium produces oxalic acid. Fumaric acid Rhizopus. Oxalic acid this is Aspergillus sclerotinia sclerotiana. Streptomyces produces taxotomins. Pseudomonas syringae pathovirus syringae produces syringomycin, syringotoxin and tage toxin. Again Pseudomonas syringae pathovirus syringicola produces fasciole toxin. Tab toxin, that is also known as wildfire toxin, it is a very important toxin that is produced by Pseudomonas syringae pathovirus tabaci. So they will simply sometimes they mention the toxin name and they will ask what is the pathogen that it produces. So then 10 toxin alternate tennis, Ophiliobolin, Cochiliobolus, Armithosporium, Pericularin, Pericularia varese, Amylovarin, Erwinia, Amylovara, Sarcospora, Sarcospora, Colrotrin, Colrotrigum. These are all a couple of toxin that is produced by the pathogen and the organism that produces. It is important. And some important toxins and their mode of actions. So the toxin name and the mode of action are produced by the pathogen. Here wildfire toxin is also known as a tab toxin that is produced by the pathogen Pseudomonas syringae pathovirus tabaci. Sometimes they will ask a causal organism or maybe mode of action. So this tab toxin is known to inhibit glutamine synthesis activity. Victorine Helminthosporium victoria that actually affects and damages the plasma membrane. Tage toxin produced by Pseudomonas syringae pathovirus it is inhibits chloroplast RNA polymerase. Fasciole toxin Pseudomonas syringae pathovirus fasciole that is known to affect membrane permeability and infects the pyrimidine biosynthesis and host lipid biosynthesis. Tab toxin Pseudomonas syringae pathovirus and the same inhibits glutamine synthesis. Pyrrolactin and syringomycin. Pyrrolactin is produced by Pyrrolaria varese, that is rice blast pathogen. And next one is syringomycin is produced by Pseudomonas syringae pathovirus inhibits membrane permeability. 10 toxin Alternaria tennis reduces the chlorophyll content and actually causes the stomatal closure. Lycomarasmin, Fusarium oxysporum pharma species lycopersiceti, which causes strepogenin deficiency and injures the permeability of the leaf surface, leaf cells. Generally Cercosporin, Cercospora zeamidis disrupt the cell membrane and loss the electrolytic leakages from the cell. Amylovarin, Erwinia amylovara, the mode of action is disrupt the cell wall permeability. Ophiobolinae, Helminthosporium varese affects the phenol metabolism. There are different kinds of toxins and their mode of actions. And R genes, which means resistant genes. So in order to protect from the avirulence genes or maybe effectors or elicitors are produced by the pathogens, so plant having some R genes, which means resistant genes. So these are all the couple different groups of major classes of R genes: NBLRR, NBLRR, CCTR, leucine-like kinases. These are all different kinds of resistant genes that is produced by the plants. So majority of the effectors or maybe elicitors was coded by NBLRR group of R genes. And these are a couple of major cloned genes are the resistant genes that were first identified. The world's first resistant gene identified is HM1, identified in maize, Helminthosporium maidis, that was discovered in the year 1992. PTO, tomato, 1993. RBS1, Arabidopsis, RPS2, Arabidopsis only, in 2001, 1994. And these are all the different kinds of HM1, PTO, RFO1, N and L6. These are all the different kinds of resistant genes that were identified in the plant. And same it continues. BS2 pepper, Peta rice, CF9, 2, 4, 5 in tomato, Cladosporium fulvum, Xanthomonas 21 rice, that is a bacterial blade of rice, Xanthomonas varese, Pathovar varese, RPG1, XA27, PAD2 rice. These are all the different kinds of resistant genes that are identified in plants. So these are all a list of couple of products and their sources and actually targets against. So ActiGuard, it is a source from benzothiazole, acebenzole or acemethyl and benzobarine derivatives and synthetic compounds. AQ10 Amphilomyces crucicollis and bicontrol acid and fungicide combinations actually here. Aspire Candida oleophila, mostly bicontrol rights. And Serende Bacillus subtilis, Yield Shield Bacillus pumilus, Messenger Erwinia amylovara, Oxycom, and these are all a couple of products and their source actually and their targeted crops and the diseases and the crops have been given here. TA plasmid, which means a tumor inducing plasmid, I am not going to discuss in detail about. Here it contains, generally the size is 200 kb and the tDNA region is ranges from 15 to 24 kb, and it contains 7 operand ranges, A to H, vir A, vir B, vir C, vir D, vir E, vir G, vir H. These are all the what can I say, some virulence regions in TA plasmid, tumor inducing plasmid in Agrobacterium tumefaciens. This is in continuation, and just a couple of basic information here. And IA auxins and AMP cytokinins are generally phytohormones that are produced during this extra overgrowth and all those things, where this pathogen actually forced to produce these growth hormones, where due to production of over amount of growth hormones only, where we are seeing the symptoms outside. The main culprit is the TA plasmid, tumor inducing plasmid. The name itself indicates tumor induction. It is just inducing the tumor. So due to this tumor inducing plasmid only, where we can see the symptoms. This bacteria is also the best example for natural genetic engineer, also called as farmer's friend, because if we remove this TA plasmid, we can use this bacteria as a carrier, like just carrying the genetic material, or like gene insertion, or in the genetic engineer. That's what it's called as a natural genetic engineer. So there are a couple of general information about this Agrobacterium tumefaciens. And functions of vir regions. This is a little bit important, because sometimes they may ask vir A is represent for vir B, vir D, that is represent for vir A is actually receptor activated phenolic compounds. So vir B and vir D for assemble the secretion systems, and vir C1, vir D1, D2, D4, E1, E2, E and G. These are all a couple of vir genes, and their functions have been given here. But just you guys just take a look into. And these are a list of DNA markers, RFLV. Most of the cases, year and references were not that much important, but at least nomenclature or expansion, like what is RFLV, restriction fragment length polymorphism, RAPD, random amplified polymorphic DNA, AFLV, amplified fragment length polymorphism. Such kind of expansions we do expect. ITS means internal transcribed, STS means sequence staggered site, VNTR means variable number tandem repeat, ASO means allele specific oligonucleotides. Like that, we have a couple of things here. ASPCR means allele specific polymerase chain reaction, APCR means asymmetric PCR, APPCR means orbitally primed polymerase chain reaction, QPCR quantitative real PCR. These are a couple of acronyms, are generally DNA markers. These all are DNA markers, along with their nomenclature I have given on the right side, and along with the year of discoveries also I have given the references who actually discovered this, and the same thing at continuation here. And we'll discuss about ELISA. At least there is a question every year from this ELISA. First of all, ELISA, what is ELISA? Enzyme linked immunosorbent assay, generally called as ELISA. It is a technique where we use it to detect the plant viruses, not only plant viruses but also used to detect the presence of human viruses as well. It is the most sensitive technique that is used for the detection of plant viruses. So generally it is there are a couple of enzymes used in ELISA. Enzymes used in ELISA are horse radish peroxidase and alkaline phosphatase. Sometimes in exams, like what is the enzyme used in ELISA? Such kind of questions we would expect. And the enzyme used is horse radish peroxidase and alkaline phosphatase. When it comes to substrate used in ELISA is the para-nitrophenyl phosphate and TMB, tetramethylene methylbenzene, okay, 3355 tetramethylbenzene. These are all the substrate used in ELISA. Don't confuse with the enzyme and substrate. Enzyme is horse radish peroxidase and alkaline phosphatase, whereas substrate is TMB and para-nitrophenyl phosphate. And the term ELISA was coined by, I repeat, coined by Eva Evangel and Peter Pellman, but the concepts and all the other things were discovered by, it is known to be discovered by Clark and Adams. And ELISA plate, actually ELISA consists of a plate which consists of 96 holes. Generally other 72 holes, 96 holes, 184, 386, the different kinds of requirements are available outside, but the most commonly used plate, ELISA plate, is consists of 96 holes. But this plate is actually invented by Guy Takatsu in 1951. The sensitivity of ELISA is 1 to 10 nanogram per ml. This is also important. It was repeated last exam as well. Where it comes to PCR, 1 to 10 picogram. So it starts with the PCR. P means picogram, like that you can remember. And generally ELISA plate is made up of a polystyrene plate. The plate polystyrene plate. And the most sensitive technique for the detection of plant pathogens also ELISA. Generally, as I told you, it contains some 96 wells. Most commonly used plate is 96 wells, and other wells is also available. And each each pore size consists of 0.4 micrometer or microliter. Actually, it's not micrometer, it's actually microliter. Now let me come once again. Enzyme is ARS production and alkaline phosphatase, and the substrate is paranitrophenyl phosphate. The sensitivity is 1 to 10 nanogram per ml, and the plate is made up of polystyrene plate. The technique was discovered by Adams and Clark in 1977, and ELISA plate consists of 96 wells, and the pore size is 0.4 micrometer, a microliter actually, not micrometer, microliter. And coming to the fathers in the field of plant pathology, what we know is just one or two maybe father of plant pathology in India, Anthony Berry, India, E.J. Butler, father of plant pathology, Anthony Berry, general, right? So here couple of other fathers I just have compiled, okay? Father of botany, Theophrastus, zoology, Aristotle, biology and natural history is also Aristotle, father of microbiology, Antonio Luinoc, father of modern botany, Linnaeus, father of taxonomy, Carolus van Linnaeus, antiseptic surgery, Joseph Lister, father of immunology, Edward Jenner, enzymology, James B. Sumner, cytology, Robert Hooke, parasitology, Francisco Reddy. These are all our fathers only, fathers and their particular field. Father of plant pathology, father of modern plant pathology, father of experimental plant pathology, founder of plant pathology, father of modern experimental plant pathology, all are related to Anthony Berry only. Father of modern plant pathology in India, E.J. Butler only. Father of mycology, P.M. Michelli, but when it comes to India, father of mycology, it is E.J. Butler only. So which means father of plant pathology and mycology in India, both are E.J. Butler only. Father of forest plant pathology, Robert Hartig. Father of forest plant pathology in India, K.D. Bakshi. Father of tropical plant pathology, H.M. Ward. Great grandfather of phytopathology, H.M. Matthew Dutullet. Founding father of systemic mycology, or founding father of modern taxonomy of mushroom, it's a CH person. Linnaeus of mycology, or father of systemic mycology, is E.M. Fries. The both CH person and E.M. Fries are the students of Linnaeus, Carolus van Linnaeus. Father of modern myxomycetes, Alexopoulos. Father of hypomycetes, T.S. Sadasivan is an Indian. Father of plant bacteriology, E.F. Smith. Father of Indian plant bacteriology, M.K. Patel. Father of hypomycetes, Sadasivan. Father of plant bacteriology, E.F. Smith. In India, father of plant bacteriology, M.K. Patel. I think we have discussed it. Father of bacteriology, modern bacteriology, founder of bacteriology, Louis Pasteur. Father of bacteriological techniques, because majority of the techniques that we are using in laboratory, that is given by Robert Koch. That's why we used to respect him with the name of modern bacteriological technique, father of modern bacteriological technique, and also the founder of modern microbiology as well. Father of antibiotics and father of American soil microbiology, Selman Waxman. Father of Indian antibiotics, M.J. Tirumlachar. Father of modern geomicrobiology and founding father of microbiology and father of soil microbiology, Sergei Vinogradsky is in Russia. Father of plant virology and father of microbial ecology, both are M.W. Bijernik, the person who discovered the virus. Okay. Father of plant virology in India is S.P. Rai Chaudhary. And founding father of plant virus epidemiology, Mitchell Truss. Mother of plant virology and serology, Helen Alice Purdy. Mostly we used to study only fathers, but there is only one mother of plant virology and serology, Helen Alice Purdy. Father of epidemiology, J. Vanderplank. Father of Indian epidemiology, S. Nagarajan. Father of nematology, S. C. Bastian. European nematology. Generally, father of nematology, N. A. Cobb is also considered as American nematology, right? When it comes to father of Indian nematology, M. M. Alam is generally considered as father of Indian nematology. Father of molecular plant pathology, Noel Keane. Father of biocontrol agent, S. D. Carrot. Father of modern microbiological technique, Valentine Relay. Father of Indian biocontrol is A.N. Mukhopadhyay. Father of soil solarization, Jacob Cotton. Father of soil solarization in India, Satish Lodha from C.S. Zedari. Father of seed pathology, Paul Neergaard. Father of Indian seed pathology, D. Suryanarayana. Father of seed testing is Frederick Nobe. Father of biotechnology, Karl Lerke. He is generally an Hungarian engineer actually. And father of soil fumigation, Edmund Thinard. Father of green revolution, Norman A. Borlaug. In India, M.S. Swaminathan. Father of mushrooms, H.P. Singh in India actually. Father of Indian rust, K.C. Mehta. Father of Australian plant pathology, Daniel McAlpine. Father of algology, F.E. Fritsch. Father of modern pycology or algology, William Henry Harvey. Father of Indian algology is M.O.P. Iyengar. Father of plant and insect mycoplasmology is L.O. Kunkal. Father of Indian phytoplasmology is G.P. Rao. Father of lichenology, Eric's Acreasus. Father of lichenology in India, D.D. Ashwati. Father of helminthology, Rodolphi. Father of Indian fungicide, T.S. Thind. Father of modern experimental optics and the giants of the Klepper of optical, Sir David Brewster. Founding father of plant pathology in Europe, Van Tubev. Father of host plant resistance, Painter. Father of genetics, Gregor Johann Mendel. Father of modern genetics, T.H. Morgan. Father of plant physiology, Stephen Hales. Father of Indian paleobotany, Birbal Sahani. Father of microscopy, Malfigini. Father of PGPR, J. Klepper. Father of remote sensing in India, P.R. Pishroti. These are all the different fathers and their belonging field. And couple of important terms and their given scientists. The term cell was given by Robert Hooke in 1665. The term mushroom was given by Nicander. The term lichen theophrastus. Powdery mildew, actually Linnaeus. Biology, J. Lamarck. Ascus, spore, verticillium, alternaria. These all terms are given by Nice in 1817. Bacteria. The term bacteria was given by Ehrenberg in 1838. Protoplasm, Purkinje 39. Cytoplasm, nucleoplasm, Strasberg 75. The term enzyme was coined by W. Cooney. Symbiosis and sex hormones, Anton de Berry. Not only these two, many other terms also coined by Anton de Berry on 1879. Mitosis of Fleming. Mycorrhiza and Aprosori may be Frank. Mosaic, Adolf Meyer. Virus, Bijanic in 1897. Virus, the term virus was given by Bijanic. Chromosomes, Waldeyer. Nucleic acid, Altman. Mutations, Hugo de Vries. Plasmodesmus, Strasburger. Rhizosphere, Hildner. Chemotherapy, Paul Ehrlich. Meiosis, Moore. Farmer and Moore. Biological control, Van Tube. Hypersensitivity is Stockman. Bacteriophage, Stuart Herrelli. The term bacteriophage was given by Herrelli only in 1917. Export is Goldstein, Bacteriocin, Gracia. Cross production, McKinney, Inaclone potential horsefall. Heterokaryosis, Anson Smith, Phenotype, Richard Goldschmidt. Prokaryotes and eukaryotes, Churton. Non-persistent and persistent, Watson, Roberts. Phytoloxin, Muller and Borger. Seed pathology, Paul Nirgard and Mary Nobel. Phytoanticipin, Mansfield. Antibiotics, Waxman. Pneumocorin, hydrochloric, Gauman. Avirulence in HH floor. Infection chain, Gauman, Plasmid, Lederberg. Vivotoxin, Diamond and Wagner. Fungistasis, Dobbs and Hinson. Phytopathometry, EC large. Acquisition, PDS, Sylvester. Lysosome, Christian Didouve. Philosphere, Last and Reen. Incubation potential and CSA, ST Garrett. Semi-persistent, Sylvester. Suppressing soil, Menzies. Xenophore, RIS. Bacterial chromosome is known as Xenophore, right? That was given by RIS 1961. Systemic acquired resistance, Ross. Bimodal transmission, Chaffant and Chapman. Doliporeceptor, Allier and Moore. McAleer and Moore. Horizontal vertical resistance, Vanderplank. Disease triangle, simple and compound interest diseases, vertifolia effect, all are Vanderplank conic. Lendicity, Nelson. Kerosene fungus, ND. Cryptosomes, cryptogram, Gibson, Harrison. Laterogenic disease, Horsefall. Man-made epidemic, PR Day. Molecules like organisms, Maramos. QTL means quantitative triloke. Zelderman, Rhizobacteria, Suslon Klapper, Hybridoma, Leonard Heisenberg, Toxins, Ludwig Brewer, Induced systemic resistant, Van Loon, Elicitor, Noel Keane. So the above all are a couple of terms and their discoveries. Now this is discovery of plant pathogens. What is the plant pathogen? Who first discovered it? The first plant pathogen, first bacteria was discovered by Antonio Leeuwenhoek in 1676. That virus was first discovered by M.J. Bijanic in 1898. Mycoplasma nocardan rocks 1898. And rickettsial-like organism Taylor Ricketts 1905. Flagellate protozoa Lefant 1909. The bacteriophage was first discovered by Twart and Herrelli individually in 1915 and 1917. L form of bacteria, Kleineberger Nobel. She just honored, gave the name L form because they are working in a Lister Institute. Joseph Lister, he is also a very big microbiologist as well as human biologist. General microbiologist, I think so. Kleineberger Nobel, she's working in a Lister Institute. To honor the Lister, they gave the name as a L form of bacteria in 1935. Lambda phases is the Lederberg, Delovibrio slope, mycoviruses Hollings, satellite viruses. The term was given by, it was first discovered by Cassanis. Cyanophages, Safferman and Morris 1963. Pytoplasma Doetel 67. Viride, Diner and Reimer 71. Spiroplasma Davis et al. 1972, Viruside Randless 1981. The preens was first discovered by Stanley Prusiner in 1982. For that he got Nobel Prize in 1993 probably. These are all the different plant pathogens and discoveries. and whatever we have discussed almost for 5.5 hours, all the information was actually a compilation, just like abstract that they just drafted from a couple of areas from my book, A Vision into Plant Pathology. This book is not only meant for net, ARS and all those things. Whatever the plant pathology exam related to JRF, SRF, net, different kinds of IPS, UPSC related exams, whatever the exam related to plant pathology, this is like a small pocket encyclopedia for especially for students. So this book is helpful. If you guys are interested, just take a look into. For further more details and guidance, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.